Welcome everyone to our first ever Global Advocacy Summit for 2022. I'm Kat Bryant-Knudsen, the founder and CEO of the Speak Foundation, the voice for limb-girdle muscular dystrophy. At today's summit, you're going to hear from individuals who are living with limb-girdle muscular dystrophy. You're going to hear from industry on clinical trials that are going to start over the next 2 years. And finally, you're going to hear about the regulatory drug process in Europe, internationally, and in the USA. Our first session is our European advocacy session, and I'm pleased to introduce Rob Camp. He is the director of the International Patient Collaboration for EUPATI. He'll be discussing the processes and structures that patients can get involved with in Europe. And then we also welcome Aleksandra Leijenhorst. She is the chair of Stichting Spierkracht, and she's the coordinator of the LGMD CAB in Europe, which is also an international organization involving South Africa and India. And we welcome them today. Thank you guys for being with us.
Thank you very much, Catherine. And thanks to the Speak Foundation for inviting us from Europe to be here today with you and to talk a little bit about patient advocacy in Europe. Later on, we'll do a deeper dive into community advisory boards, but right now I just want to give an overall picture of what's happening in Europe. So again, thanks very much to the Speak Foundation for putting together this whole LGMD Global Advocacy Summit, and let's go forward. Great. So just to help us all clarify what is Europe, according to the Council of Europe, Europe is made of 46 countries. So it's a relatively diverse group of autonomous nations who oftentimes work together. If we think about the European Union, we're actually smaller than 46 countries. We're 27 countries right now. And so the EMA, which is the regulatory body for Europe, basically functions for 27 countries in the European Union, plus 3 EEC countries, which are Iceland, Liechtenstein, and Norway. For example, in this map, if you look just for an example, Ukraine and Turkey are not part of the EMA, although even countries that are not taking part in the EMA listen very closely to what happens, what comes out of the EMA, and that somewhat guides them in their future decision-making processes. And I'd like to talk to you a little bit today about patient advocacy as much on a local level, what you do sort of in your local group. I'm from Barcelona, so I started working in a group in Barcelona in HIV almost 30 years ago. Then I went national, started working on a country level, and finally I'm now working international. And I was working with Euordis for many years, putting together the CAB program from what we did in HIV many years ago. I did want to mention, I just want to bring up quickly that sort of, if you look at the UK here, which is this island sort of in the middle of the map, so to speak, the UK used to be a really important part of the EU. It's now no longer the case because of this word called Brexit, that since January 1st, 2021, they no longer take part in EU decision-making processes. And they are no longer part of things like the EMA. The EU was basically put together to be a free trade group between the 28 countries. It's now a free trade group between 27 countries, and the UK has to negotiate trade issues with the European Union from now on. But more importantly for us as patients is, how does that affect people living in Europe who would like to go to the UK for treatment, or vice versa, people in the UK who want to come to Europe for the same reason, for treatment reasons or therapeutic reasons. basically now you need to jump through quite a few hoops. The last bullet point here is the electronic healthcare card that we can use to go from one country to another. If I would like to go to France, I just make sure I have my electronic healthcare card and go to France to do so. Now, for anyone from Europe to go to, for example, Newcastle, there's a lot of issues that you have to go through, and that includes making sure that you know who will be paying for the treatment and how before you go to Newcastle. So it's much more complicated, and you need to figure that out before you start the process. There are ways of doing it. It's just not as easy as it once was before 2021. So that's just somewhat of an aside. So what I wanted to mostly focus on today is the European Medicines Agency, the EMA, which is a sister body, let's say, to the FDA in the United States. They're the regulatory body who makes decisions on many things, on approvals of drugs for new molecules that have never been seen before, but also including generic drugs. In 2020, I have another slide, they approved 96 drugs in total, I believe 39 of which were new molecules for the year. Of course, one of their main important issues, of course, is the safety of the citizens of the European Union, and they take that very seriously. Public health is a major factor for the European Medicines Agency, the EMA, and we saw that in 2021 and 2022, that they had a series of, I was going to say televised, of video conferences with the world regarding the approval of vaccines as well as of treatments for COVID-19, which is something fairly new to the EMA, to do televised meetings. The meetings were fascinating, and they're all online, and you can go look at any of the meetings that have already happened. And they did spend a lot of time just on what I said, on safety and on public health issues. They also work on approving products for outside the EU. Normally that means for countries in the developing world, countries for example, in Africa, or sometimes in Latin America or Asia, countries that don't have the same access to regulatory authorities, regulatory bodies as we do in the North. They're really concerned with early access, and they speak with all companies. When companies come to the EMA saying, we'd like to talk about our approval, our development plan for this product, the EMA always brings up early access. Will you have an early access program? How will it work? Are patients involved in the design of this early access program, et cetera, et cetera, et cetera. So it's really important for them that companies know that they will need to know whether or not they will have an early access program with their development program. The EMA spends a lot of time on rare diseases. Actually, in 2020, 50%, a little more than 50% of new drugs that were approved, new therapies that were approved in the EMA were approved for rare diseases. So that's a really important part of the work that the EMA does. And of course, like all agencies in the world, there are also negative opinions. That is, not everything is approved. They do offer an option to sponsors to pull their application before they get a negative opinion, but there are some times when they do finally give a negative opinion. I just want to go briefly through patients' engagement at the EMA. EMA was founded in 1995, just a couple years after the founding of the EU. EMA started in 1995, and right away in 96, they started meeting with patients as far as what do patients need, what do patients want, how do patients see this European approval process. So right away, patients started getting involved in the EMA. In the year 2000, patients were even starting to go on the different committees, like the orphan medicines product committee, etc. I'll go into that in the next slide. In 2005, they started writing a framework of how will we as an agency work with patients as we move forward. And also, just because I'm remembering it now, I think it's really important because this is a body, an international body, made up of the heads of medicines agencies on a national level. Everything that they approve on the EMA level then sort of trickles down to a national level. So what first happened at the EMA, for example, in the year 2005, is now starting to happen on a national level because it was some of the same people, for example, in Spain, who worked on making sure that patients were involved in regulatory decision-making in 2005 that are now including patients in the same decision-making on a national level, for example, in Spain. In 2014, there's a whole department made now of public engagement where patients are really front and center of why this department exists. And just 2020, patient engagement in crisis management was another issue that came up. And that was obviously because of COVID-19, but now there's a plan of how to work in crises moving forward. The EMA has, if we look at any regulatory system, there's pre-submission phase, then there's the evaluation phase, which is after phase 3, and then post-authorization. So we see that here patients are involved in all 3 of those really big areas, from being in the comp here on the left, which is the Orphan Medicines Product Committee, which is the committee that deals with orphan medicines, rare diseases. So that they're involved in pre-submission. They're involved, of course, in evaluation here in the middle, and then they're involved as we move forward in post-authorization. And you can see patients are involved in the second circle. You can see the human medicinal product, which is all drugs for human use that goes beyond the, let's say, 50% orphan drugs, and is all drugs that patients are involved in as well. In the middle, I just wanted to highlight PRAC, which is the Pharmacovigilance Committee, which again is over time watching the profile of the drug, the side effects. Is it safe? Is it safe to use in a wider population than what was studied? So in all of these places, patients are involved with the EMA in the decision-making. This is really the same slide. I just wanted to highlight here this very first line that says patients can represent their community in the management board. The management board is actually the highest level within the EMA where real policy decisions are made. And there are patients in that management board committee. So again, just to emphasize that patients are really in all parts of EMA. And maybe here on the left again, just to say that patients can represent their community, patients can represent their organizations, or they can be individual experts. Many times at the time of evaluating a product, patients maybe had not been involved in evaluating the drug at all. So then the EMA looks for people with the disease or living with the disease or families of patients with the disease and say, does this make sense to you? Does this work for you? And they come in as individual experts to say, yeah, this looks okay, or not, as the case may be. So I think that, again, EMA really has to be lauded for the work that they do. Very quickly, I just wanted to say here that to work, and I just mean to be involved with the EMA, there are 35 patient and consumer organizations here on the right who have been pre-approved by the EMA, and there's an application process that you have to go through, but who then are called on fairly regularly to give advice to the EMA. And I know it's not very clear, but the very last the very last logo here is the World Parents Project of muscular dystrophy from the Duchenne group, and they are an eligible patient organization, and they have been for many years. And also, I just think it's interesting that there are actually more patient organizations involved as advisors than doctors' professional organizations that you see here below. Which I'll also show in another slide, that there are more patients involved than healthcare professionals at the EMA. And so just where patients can really help out, I think that the EMA has seen that face-to-face meetings are really important, but also opinions that come in writing from patient organizations. And that's, again, where community advisory boards might come in, where if we agree or disagree with a product development plan, we as a community advisory board can write to the EMA and say, this makes a lot of sense, or this could be better. And EMA really wants to hear and listens to those opinions from the community on specific products. Of course, like I said, patients are involved in committee meetings, et cetera. And on the right, one thing, the training day, they have a training day every autumn. This year it will be in October, where they spend time on giving I think I have another slide on giving specific advice, on giving training on how to understand, for example, clinical trials better. And I just threw this in because I recently got a note from the EMA saying that I was going to be removed as a patient expert if I didn't fill out a form. So I quickly, on Monday, I tried to do it. It was almost impossible to figure out. It's kind of complicated, or I'm not that technically savvy, which is also very true. But I sent a message quickly to the patient advisor at the EMA, said I don't understand how to re-up to be a patient expert, and they very quickly showed me how to do it. It took me 10 minutes, and now, once again, I'm registered as a patient expert, as an individual. So again, it's fairly simple to do, and if you can't figure it out, there's someone at the EMA who will walk you through it, and it's a very, once they help you, it's a very straightforward process. And just, I think, to finish up here on the EMA, I just wanted to show that where patient advice really is important is, for example, in the public assessment report that they publish for each product. It's written in lay language, and it's written so that the normal public can understand what exactly is going on with how the drug works and how the drug should be used in real life. There's also other things, of course, that patients are involved in, like evaluations and packaged leaflets and so forth. But I just wanted to show here what they call the EPAR, the public assessment reports, where patients are really central to making sure it's understandable. So sort of back to patient advocacy directly, I think that there are 3 levels of patient advocacy that we need to keep in mind, at least in my experience over the years. This is how I've sort of grown to see it. And I think that it makes sense to look at it this way, that there's patient education on a local level, on a national level, and on an international level. Locally, obviously, how you can help educate is just provide support to people who are living with the disease. Nationally, the same thing, but also raising public awareness, advancing research on a national level, improving quality of care, and maybe even addressing some political decision-making that might affect care and research on a national level. In the EU, most health decisions are still nationally controlled. So it's really important especially for care, to work on a national level to make sure that patients' voices are part of the discussion. And of course, internationally is all of the above, but also it includes something called collaboration. That is, we need to start looking at other cultures and other ways of thinking and other people beyond our own small society, and And I think that it's really, really wonderful to start connecting on a much larger level, on an international level. And just the end, sort of the same thing, just if, as a patient advocate just starting out, the idea would be to work at the level you're most interested in. I work internationally mostly, but I still work on a national level. I'll show later on some EUPATI Spain slides where we do patient education in Spain that is centered on Spain. Now it's sort of a little more complicated because Spanish is such a big language, and actually more people from Mexico look at our website than people from Spain. So maybe we need to start thinking about international, even for national issues. But anyway, so for local work, maybe you can speak with someone who's recently got a diagnosis, who maybe needs to better understand what exactly it means, and do they have genetic testing, and what will genetic testing help them with if they get it. So things like that we can do on a one-to-one, on a local level, I'd say. And for example, I put support groups here because support groups were really helpful in the 90s when we started in HIV, and I see that it's still really important, at least online through Facebook groups, etc., as simply as support for people who are living with LGMD. Nationally, again, we go through maybe educating the public a little more about the disease, expressing the importance, again, of screening and early detection, and specific topics we also need to be concerned about, for example, insurance access. Although in most EU countries, treatment and therapies and care are taken care of by the national health system. In some countries, there still are insurance companies who will take care of it, but you still have to go through sort of an insurance process. For example, in Germany, that happens quite often. And again, making sure that there are no disparities in care, that maybe rural versus urban, making sure that everyone has the same access to care, job discrimination, communicating with the media, etc. And of course, now media also includes blogs, podcasts, vlogs, etc. So I think that media is an important area that we should not forget about. And finally, international, I put the LGMD CAB logo here because I think internationally we work on a lot on education about clinical research and reducing barriers to participating in research, etc. So there's lots of places where we can do work as patients. So moving on to training a little bit, moving on and going back to EMA for 1 second. EMA offers, like I said, training every year, face-to-face, except for the last 2 years. It's been online. But they offer training for patients and healthcare professionals. And last year, they haven't put the agenda together for 2022, but for 2021, the agenda was basically built around how patients can contribute to understanding clinical trials, especially meaningful clinical trial endpoints, the feasibility of clinical trial design, etc. So all those things, the EMA is really trying to help us understand how we can make a difference by collaborating with the people who are designing these trials. And so the same, something similar will happen in 2022. If it's face-to-face, it will be a full day, not just a half a day meeting. So for training, again, I have a couple slides on a couple different initiatives in Europe that are actually global in the sense that anyone can access these. They're all free and they're all online. The one from EUPATI is they're centered in the Netherlands, but in fact they offer training, what they call here toolbox and training in 13 different languages, including English. So it's I can't pull down this is just a slide, but in all the the major languages of Europe, plus Japanese, I believe Korean, and many other languages as well. They also have what they have here called national platforms, which are national versions of EUPATI. EUPATI, like I said, is in Netherlands, and they put this training program together. But nationally, for example, in EUPATI Spain, we do what we need to do for our patients in Spain, which may coincide or may be slightly different from what they do at a central level. Right now, there are 23 national platforms. So it's already in many more countries. EUPATI was originally a project of the European Commission, and it was originally designed to be in 7 languages. It's already now in 13 languages in many more countries than it was originally planned for. So I'm sorry, I should have waited for this slide, but this is where we do national training. If you see here in the second and third bullet points, the countries that are involved with national training, and we do in Spain, for example, both mini courses, workshops, and training initiatives in Spain that mostly depend on EUPATI information. And this is all free access. Just go to eupati.eu and you can see more of what's available. I just wanted to mention here that at EUPATI Spain, we will have on the 20th of May, which I know is the day of the summit, but here in Spain, we'll be offering a roundtable on the ethics of clinical trials, because May 20th is the global International Day of Clinical Trials, commemorating the first clinical trial that happened in the 18th century. And so every year we do a roundtable on clinical trials. This year, the focus is on the ethics of clinical trials. And because, and I say this somewhat humorously, everyone in the world now is a specialist in clinical trials because of having followed the COVID clinical trial situation over the past 2 years. We just ask people on the street, what do you think of clinical trials? Do you think they're ethical? Would you enter a clinical trial? Why wouldn't you if the answer is no? And it's a very exciting sort of hour and a half that we talk with people. Also, we invite people from the National Regulatory Authority, we invite an ethicist, and we invite someone from the industry as well as a patient to talk about their versions of the ethics of clinical trials. Back to training, EURORDIS, of course, is sort of the mothership, if you will, of rare diseases here in Europe. And EURORDIS has many, many programs. And I just wanted to spend a minute on the training programs that they offer. All their training programs are open and free online, but they also do have or will have face-to-face again once everyone hopefully soon gets back to being able to do so. But we have a summer school, which is based on regulatory authorities' decision-making processes. That is an 11-month course, and at the end of the summer school, there's a face-to-face meeting, usually held here in Barcelona, but it's been online for the past 2 years. There's also a winter school, which is focusing more on innovation in therapies and translational science. That's a really wonderful program, and I believe Aleksandra later, who will speak, is also a part of the winter school. There's also a leadership school and a digital school. So it's a really quite full program, and it's all focused especially on rare diseases. EPF is the European Patients Forum. They also have a training program. Their training program, of course, doesn't repeat what other trainings do. But for example, here, the EPF does work on fundraising, patient engagement, even a course for young patient advocates. So again, I think that putting all of this together, it really becomes a really wonderful tool of not only learning, but how to implement so that you can make decisions, again, either locally, nationally, or internationally. So changing speed a little bit, moving out of training and into other things that patient advocates can be involved in is the innovative health Health Initiative Project. I equate the IHI to the NIH in the United States. So the NIH, I believe, has a budget of approximately $3.2 billion per year. I think the IHI has a budget slightly smaller at 2.4 billion euros. What I just wanted to mention is that they basically put together calls for projects from anyone, and anyone can apply to do a project in specific areas that the IHI says are important for public health overall. So for example, I just looked this morning on projects, and that one of the projects that's still open for 2022 is how to identify the molecular response of non-response to treatment in autoimmune diseases, inflammatory diseases, and in allergies. So anyone can look at the call, decide if they're interested, put together a consortium of people. Of course, like most European projects, the more countries represented, the better. But even one country can begin the process. And here, although the UK can't be a leader in any of these projects, they can be a member of the consortium in projects related through the IHI. So this is something that you all can look at online. It's really fabulous, and I think they'll be having a second call in 2022 later this year. Just to remind us all that we can be involved as patients in the R&D life cycle, everywhere from even before there's a drug in hand, all the way through the development process out the other side after hopefully approval if all goes well. So for example, the story I like to imagine is that someone here on the left in yellow, maybe even before they have research priorities, they have a dream. They're an investigator in a company. They wake up one morning and say, "Eureka, I've thought of the answer." And before they call the CEO or their boss at the company, my fantasy is that they would call patients and say, "Does this make sense to you? Or what's lacking in my understanding of why I think it's so great?" And then they start working together with patients all through the process, all through animal studies, in vitro studies, phase 1, 2, 3. Hopefully, if approval goes well, even beyond when drugs are approved. For example, with gene therapies now, I believe in the EU, it's either 12 or 15 years that drugs need to be followed after approval, and patients need to be involved in all of that process at every point along the way. Again, Aleksandra later will be talking more specifically on what are community advisory boards, but I just did want to throw in here, because I think it works well with the previous slide, is that community advisory boards, we can review clinical trials before they get up and running. We can monitor some ongoing trials. And regarding patient priorities, we can stay informed about interim results if there are interim results. For example, quality of life and patient-reported outcome measures are also really important. And just sort of, not to just simply read the slide, but the final bullet point about adverse events reporting, I think that we can be involved more in making sure that adverse events are reported and reported well, and then something is done about them. I just wanted to give a couple slides on successes that we've had in community advisory boards as patient advocates in other disease areas. Aleksandra later will talk about LGMD, but I wanted to show what's already happened in other diseases. In Duchenne, for example, they've already changed the number of biopsies in 3 different studies. They've reduced the number of tests and questionnaires in other studies. They've reduced the length of placebo. Rather than being 1 year, it's much, rather than being longer than 1 year, it's 1 year in one study, 6 months in another. And it's because they, as patients, came to the table with the sponsor and said, "This is too long. We need a shorter placebo arm." And depending on the situation, it's not a one-size-fits-all issue. the company can say yes, no, or maybe to all of this. Sometimes the company needs to go home and think about it, but sometimes at least the discussion is started in a face-to-face meeting with the company. And for example, randomization ratio of how many people get the new drug versus the placebo, they've also been able to change in some cases from 1-to-1 to 2-to-1 in one case, and even in one case, 3-to-1. So 3 people get the new drug and only 1 person is on placebo. And also many companies now share their data after individual data with the individuals after the study. Cystic fibrosis, quickly. Cystic fibrosis came up with a great plan to how to follow these drugs and these companies over time. So they invented a tracking system called the Tracker. And the Tracker focuses on clinical studies, access, communications, and patient support and quality of life. And so in those 4 areas, working with the companies, they decide in clinical studies, what will we work on together? How will we do it? What milestones do we want to see? And will it be positive or negative? And what's the timeline for that? So all of that, they've worked out sort of in an Excel sheet, if you will, and just over time, see if in fact they've made a difference in the success of clinical trials in cystic fibrosis. They've also worked on many informed consent forms over the years. As maybe many of you know, informed consents sometimes are impossible to understand, and they work with companies to make them more reasonably understandable for the patient who will actually be in this study. Like I said, they're doing evaluation as well of how effective they've been with companies over time. And we'll have more information on that, hopefully fairly soon. Final success story is a small one from cystinosis. Cystinosis had their first CAB meeting in November of 2019, and the company said, patients who enter our study need to stop their previous medication 60 days before entering our trial. And everyone in the community advisory board said, impossible. This is the only drug that we have that works for our kids. They can't stop the drug 2 months before starting this trial. This is a no starter for us. So the company was surprised. They went home. They had internal meetings, as would happen, and they wrote back in 10 days and said, you know what, there's really, we don't need to stop the existing treatment before day 0 of the new trial. So your kids can continue on what works for them until day 0 of the trial. And so even in the very first meeting that this community advisory board ever had with a company, there was a win. And I think that's really something that we need to remember, that even small changes can be really significant for patients in the trial and hopefully for the success of the trial as well. I think I'm coming near the end. I hope I haven't gone too over time. But here in Europe, once we get drugs approved from the EMA, we don't get access immediately. In Germany, mostly they get quick access. But everywhere else, they need to go through a process called HTA. And HTA is Health Technology Assessment. It's usually country by country because, like I said earlier, health is still a national issue in the EU. It's not an international issue, except when possibilities like this come up. Both UNETA on the left and HTA International on the right try to combine HTA bodies on national levels and make them more international, so that what we do and decide in Spain doesn't have to be repeated in Italy, or doesn't have to be repeated in France, or doesn't not agree with what happens in Portugal, etc. But where everything is more coordinated. Because right now, just as an example, in Spain, the median time for access to new therapies is about 2 years after EMA approval. So, for example, we get a drug that's approved for cystic fibrosis in November of June 2020. We actually don't get access to the drug until HTA goes through their process of evaluation of value until almost 2 years later. So EMA works on risk and benefit, HTA works on value. So there are two separate items, and until both of those are resolved, we usually don't get access to products in Europe. Both of these organizations are trying to make that easier, and again, patients are involved in the steering committees of both of these initiatives. These are my last two slides. You'll be happy to hear. The first one is just how more we can work as patient advocates. We can fundraise on a local, national, or international level. Plan fundraisers, including walks and runs. We can donate money, et cetera, et cetera, et cetera. Or we can, and in rare diseases, many times it's rare disease organizations who raise funds for development of products. So I think it's also important that, somewhat differently than in many other disease areas, in rare disease, patients and patient advocates can be involved even earlier in decision-making about products because they have been part of fundraising for the research to move forward. And finally, public policy, again, on European level, a national level, and a local level. It's really important to keep in touch with politicians and legislators to testify whenever possible at government hearings, et cetera, to make sure that our voices are heard regarding any policy changes or any policy initiatives that might come up that involve LGMD. And I know this seems like a lot. I've gone through 30 slides, and it seems that patients, I suggest, need to be involved everywhere. And I just remember one time reading someone in HIV saying that if we don't define and take charge of doing it, someone else will decide for us. So we really need to be at the table helping to make the decision, maybe not making the full decision, but being there at the table to make sure that our voice is heard at the table. And for example, the pressing needs of people living with LGMD may not be heard if someone with LGMD is not at the table. Research volunteers in LGMD trials need to be protected. All of this is really complex. Even the word community is complex. How big is the community? How do we define community? And of course, the CAB, for example, advocates for very specific things in trials. For example, we could advocate for extended clinical hours, where one night a week, a hospital is open later, one night a week for people who are living with the disease, if that's something convenient for people with LGMD. We look for those quote unquote innovative solutions. And of course, we want to make sure overall that the design and conduct of clinical trials is more transparent. So I think I will end there. Yes, so I end there. So again, thank you very much to Catherine and to the Speak Foundation. And I hand it to Aleksandra now.
Thank you, Rob. It's a big privilege to have you as a trainer. I'm Aleksandra Leijenhorst, Chair of Stichting Spierkracht, Coordinator of the LGMD CAB, and Ambassador of the Carefree to School Foundation for Kids with Chronic Diseases in the Netherlands. Today, I want you to introduce to the LGMD CAB Community Advisory Board. I thank the Speak Foundation to give us a place and priority in the Summit 2022 as they value our LGMD CAB. I have been involved in the neuromuscular world for 17 years now, and I am really hopeful and grateful. I have a vision: when we work together, we can be the change. Everyone who lives with LGMD, we have the same goal and need to work shoulder to shoulder as one family. So let me take you on our CAB activity. What is a community advisory board and why are we in this CAB together? CABs were originally created in the 90s for HIV, AIDS, hepatitis and breast cancer to balance the fact that until then medical science had evolved with little to no input from people being studied. CABs change that and help industry and other researchers to design, carry out and communicate on studies that are more rational and inclusive from a patient point of view. At the LGMD CAB, we meet with companies to give them, the members, combined expert knowledge and advice to optimize clinical trials for better and faster results for people with LGMD. Everything that can accelerate treatments or a cure through market and bring them to the patients without leaving anyone behind. The CAB is operated by patient advocacy leaders and facilitated by Eurordis Rare Disease Europe and EUPATI. We are supported by Rob Camp, who provide us with trainings for more than a year. The LGMD landscape is changing, and we include all types worldwide, as we also want global access, and there are many differences in many countries to overcome. I hope we all learned from COVID-19 that we need to think globally and not per country. We are independent, autonomous, and an international board of trained patient representatives, including parents, people with LGMD themselves, and leaders of the LGMD member organizations from 12 different countries across the world. LGMD and patients are represented from Scandinavia to South Africa and from India to the United States. To give you a face to who we are, I want to meet you, the members of the LGMD CAB. Not all of them, just a few. I introduce you Kate Adcock from the United Kingdom.
Hello, I'm Dr. Kate Adcock, and I'm the Director of Research and Innovation at Muscular Dystrophy UK. At Muscular Dystrophy UK, we bring together more than 60 rare and very rare progressive neuromuscular conditions that affect around 110,000 children and adults in the UK. And this, of course, includes all forms of limb-girdle muscular dystrophy. As a charity, we support high-quality research to find effective treatments and ultimately cures. And we ensure that everyone has access to the specialist healthcare and support that they need, the right help at the right time, wherever they live in the UK. And we provide a range of services and resources to help people live as independently as possible. And we drive to get faster access to emerging treatments for families in the UK. And it's because of our interest in research and getting treatments to people living with conditions such as limb-girdle muscular dystrophy and our expertise that I can bring in working across several conditions, that I'm proud to be a member of the Limb-Girdle Muscular Dystrophy Community Advisory Board to support the UK and indeed the global community access clinical trials and ultimately better treatments.
Thank you, Kate. I want you to meet Jitika Khanna, our member from India.
Hi, this is Gitika Khanna, representing Indian Association of Muscular Dystrophy, India, and I'm a patient of LGMD2B since last 25 years now. I'm one of the oldest patients here in India. And in IAMD, what we do is rehabilitation of patients, along with providing them relief, like providing them with different aids that they require to manage the disease. And then we provide counseling especially during the Corona period. We did a lot of physical counseling, we did a lot of video counseling. And apart from that, we also advise on the physiotherapy. So the entire motive is to maintain the muscle mass as long as we can. And I'm very, very grateful to LGMD CAB for providing me the insight to the clinical trials, especially how they are conducted and the intricacies and that are involved, funds that are involved, the patient inputs from the patient that are involved. And I very much look forward to meeting a lot of new companies through them, so we can provide a cure and a treatment in the near future for all the LGMD as well as other muscular dystrophy communities here in India and in the world. I also wish to extend thanks and gratitude to Speak Foundation for letting us raise awareness and work for patient advocacy for LGMD. Thank you.
Thank you, Jitika. Back in Europe, I want you to meet Stefania and Liana from Italy. Stefania speaks Italian and Liana translates everything.
Hello, I'm Stefania. I'm 45 years old and I have a limb-girdle muscular dystrophy, type R3 or 2D. I'm a psychologist. I work at the hospital. Department of Muscular Disorders, and I am the National Vice President of WILDM, the first Italian association born to assist people carrying any type of muscular dystrophy. 2 years ago started an LGMD group, which now includes about 100 individuals with different subtypes. In trying to find the best way to support our community and to raise awareness, we had the great opportunity to get to know EURORDIS and the LGMD CAB. We have become members of the CAB after having received a specific education through courses and masterclasses. We strongly believe that knowing how other nations deal with limb-girdle muscular dystrophies is important for the exchange of experiences and personal feelings. For our community, especially for Europe, we would like a common way of total caring and a common regulatory policy for the approval and distribution of specific treatments. Our dream is that a drug treatment is found to slow down at least the progression of the disease. The best will be a gene therapy to cure the defect and bring improvements. Also, our best wish is that in this process, companies pay attention to the point of view of the LGMD community. The CAB may help to make all this happen. Together we are stronger.
Thank you, Stefania and Liana. I also want you to meet Jessica from Spain.
Hello, my name is Jessica Martin. I'm a nurse at the Maternal and Child Health Department. I'm also the person responsible for the quality and safe patient care at Mataró Hospital. I have 2 children affected by LGMD2D. They are currently 10 and 7 years old, and they were diagnosed 5 years ago. When we received the diagnosis, we didn't find any association in Spain that represent the people affected by this disease, nor any place where we could find assessment or support. So, in 2018, I found Proyecto Alpha together with my husband and our relatives. Proyecto Alpha is a non-profit association that represents family and people affected by LGMD. Our goals are creating a registry of affected people in Spain with genetic confirmation, carrying out activities to give visibility and raise awareness about LGMD, and giving support to the research projects. One of the most important activities we have carried out is the documentary Never Stop Moving, which talks about LGMD. It shows the daily life of 3 families in Spain. Also, the song is entitled as the documentary Nunca dejes de moverte. Never stop moving. More information will be available in the next LGMD magazine. In January 2022, our association started collaborating with Sant Joan de Déu Barcelona Children's Hospital, which is a neuromuscular disease expert center. This collaboration focused on a natural history study about the sarcoglycanopathy. It correlates functional skills with serological and histological markers to know better the evolution of the disease. I'm thankful for being part of the LGMD CAB since 2020 and for sharing knowledge with my colleagues from other countries. Together, we work to make sure that LGMD patients' voice and necessities are covered, listened to. We know that this knowledge can improve the experience of people who will participate in future clinical trials. I would like to finish my speech with the mission statement that reminds me that all I do is worth it. We work forward with the hope of finding a cure so that the muscles never stop moving. Trying is 99% of the success. Shall we do it together?
Thank you, Jessica. The USA is very big, so we have two representatives from the USA. We have Ralph Janis and Donovan Dekker. Donovan, can you tell us about your experience with our CAB?
Hello, my name is Donovan Dekker. I am one of the representatives from the United States on the Community Advisory Board in Europe. I've served 6 years on the Muscular Dystrophy Coordinating Committee at the National Institutes of Health. I am also a member of the Patient Advisory Board for the Speak Foundation. I've had limb-girdle muscular dystrophy for 44 years. I have 4 sisters with limb-girdle muscular dystrophy 2D, plus 2 nieces with limb-girdle muscular dystrophy 2I. 2 of my sisters have died from the disease. One thing I look forward to working on the CAB in the future is working with the pharmaceutical companies and researchers building a trial and helping patients find the trial. In 1999, I was the very first person in the world to do human gene therapy for any form of muscular dystrophy with Dr. Jerry Mendell at the Ohio State University Hospital. I believe I can help patients, parents, pharmaceutical companies, and researchers understand what it's like to be involved in a trial. I also hope to educate limb-girdle muscular dystrophy patients that they need to do genetic testing to find their gene defect before they can be involved in any research, or if a cure comes along for certain gene defects, a person needs to be tested. This should be completed shortly after their diagnosis. While over 30 different gene defects have been identified so far, many still need to be identified. The training I've received because of being on the CAB has helped me understand all the different issues that have to be completed on getting a drug or treatment approved in other countries. Learning how other countries work on diseases can only help benefit everyone. Thank you.
Thank you, Donovan. I think you are an example on how patients can be the change. The LGMD CAB facilitates discussions with sponsors of clinical research. This could include a pharmaceutical or biotechnology company, but we are also happy to meet with a hospital or university research team. We can discuss latest developments and challenges related to medical research from the patient's perspective. And the discussions are held in a neutral setting and are structured around the principles of confidentiality, transparency, sharing, openness, optimization of research and development, and a joint action plan as an outcome of the discussions. We can give input on a variety of topics, thanks to the expertise of our members, patients outreach, education on research, clinical studies and their design, criteria of participation, informed consent forms and processes, compassionate use programs, retention of participants, and reporting on results. The CAB members offer their expertise by providing advice before a clinical study starts and ensuring that clinical studies are designed to take into account the real needs of patients. This could mean the CAB members offer their expertise in how the clinical trial is run, how the patients will be able to best adhere to protocols, or how to ensure continued access by the medication after the study ends. We can also help companies think about the needs of patients to help with recruitment. Input is also given on disease-specific topics, such as clinical endpoints and how they are measured, meaningful patient-relevant outcomes, the PROMs, diseases registries and their common elements or factors that are meaningful to patients when measuring health and social outcomes. And our representatives always speak on behalf of the patients in their country, never for themselves or for their kid. Ultimately, we want the best outcomes for people living with LGMD. More than ever, patients and the patient perspective are seen as a way to make medical research and clinical studies more impactful, possibly quicker and certainly more efficient. We meet regularly and have identified what we see as a success. These slides give an overview of how many CABs we have in Europe. We have 9 CABs, and 111 CAB members. We have engaged patients, researchers, and companies together. You can get involved too. You are the expert. You are living with the disease. Time of waiting is over. We have to act. Sharing your data is important. Know your mutation. You own your data. The data is yours, and the CAB urges you to register in the database of Treat NMD. It is a safe registry, and companies can request anonymous data to help design studies or contact participants via the registry to take part in trials. Always be aware that you stay the owner of your own data. Visit My Journey or Our Tomorrows and get enrolled in the natural history studies. Look for trials at clinicaltrial.gov and be active. The LGMD CAB is interested in engaging with all researchers at the earliest stage possible. Please contact us for an information pack at cabmembers.lgmd@gmail.com. And remember, you are not alone. We are in this together till LGMD can be treated.
Thank you to our European advocacy partners for that wonderful session. We now are going to introduce our international updates. We have Mandine Casado, who will be talking to us about AFM-Téléthon. And we also welcome Carles Sánchez Riera, who is a PhD and researcher for Sapienza University of Rome. They're going to be sharing international updates for LGMD and advocacy. Thank you guys and welcome.
Hello everyone. So I am here today to present you the LGMD Peer Support Group, which is the French patient group, part of the AFM-Téléthon Association. So the AFM-Téléthon have a big strength, which is that she regroups only patients and their parents, their family, in order to defeat the disease. So it's a patient organization which bringing together volunteers and employees, and all the volunteers are patients or families of patients. The goals of this association is to conquer the disease based on a strategy of general interest. And we have the chance to be supported by French TV with a popular support through the Téléthon, which is the annual fundraising event in France. So this association has three missions. Firstly, to cure, so research and development of therapies. Secondly, care patient and their family. And thirdly, the communication in order to raise awareness about neuromuscular disease. So firstly, to cure the disease. So it is important to keep in mind that AFM-Téléthon have 3 research laboratories. So the first one is named Geneton and is especially on gene therapy. The second one is eStem, which is about the stem cell therapy. And the third was the Institut de Myologie, which is based on the myology research. Associated to these 3 research laboratories, the AFM-Téléthon has the first French industrial pharmaceutical facility dedicated to the production of gene and cell therapy for the rare disease. It is important to note that there is a big scientific council with a lot of expert members in the association. And I think it's also important to keep in mind that the board of directors is composed of patients and their families. One of the best achievements of the association is the creation of the biotech company dedicated to the limb-girdle muscular dystrophy and named Atamio Therapeutics. So this company was launched in 2020, and the aim is to develop gene therapies for various limb-girdle muscular dystrophy. One of the great achievements is the clinical trials for FKRP-related LGMD. We will begin in some months in 2022. There is also hope for 4 other clinical trials we are in the pipeline of this company. So for the gamma sarcoglycanopathy, for the kalpainopathy, for dysferlinopathy, and for alpha sarcoglycanopathy. The second mission of the association is to care. So there is a lot of patient support, especially with regional services in France, with the creation also of the peer support group like the LGMD support group and with facilitator. The aim is really to have an advocacy for the rights of people with disabilities. It's also important to keep in mind that the AFM-Téléthon participate to the creation of the rare disease platform and to the creation of respite home in order to allow patient and family to have holidays. And the third mission is the communication. This communication is especially done through a social network like Facebook or Twitter, but also through the website of the association or the website of the support group. And the TV show is also an important event to communicate to all the people in France. Now I will speak a little about the LGMD peer support group. So this group was created in 2018 and it regrouped 15 people all around the France. All these people are patients or family when people suffering from LGMD. The different activities of the LGMD support group is firstly to support patient and family. So we want to inform about LGMD, about recent research, development, and the clinical trials ongoing. For this, we have developed a helpline, social network, blog, patient, and patient support meeting, especially in region in France. And all this activity allow us to have a peer-to-peer exchange. So we are LGMD expert patients. So we work closely with the clinical professional and with researchers to help them to better understand the disease and to make progress in research, medical care, and also social care. And finally, we have an important level of interaction with the international. So in order to develop synergy and partnership with international association, we participate in national and international events. And the aims of all your effort is really to share the expertise of patients living with LGMD and the expertise also of the association of the French Association of Muscular Disease. Thank you very much for attending this session.
Hi, I'm Carles, and I'm going to present an update of the projects of GFB. GFB is a patient association that in the past funded the preclinical studies of beta-sarcoglycan gene transfer, and we also participate in different natural studies of the beta-sarcoglycan sarcoglycanopathy. But this is enough, it's not enough. And now we start the quality project. This quality project is a project about the quality of life of patients with alpha, beta, and gamma sarcoglycanopathy. And this project is funded by GFB exclusively. This project is structured in 6 parts. These parts are at the distance of 6 months each one, and each part consists in different questionnaires. As for example, the first part, we have the policy and the consent, the personal data, some questionnaires about the physical activities, the clinical questionnaires, and other kind of questionnaires. In the second part of this quality project that is already ongoing, we remove the clinical questionnaire as we already have it, and we add another questionnaire that is called PROMISE that allow us to evaluate the fatigue and the sleep disturbance or positive effect, among others. In this project, we have participation of patients of all around the world. We have 44 patients with alpha sarcoglycanopathy, 69 with beta, and 33 with gamma. We already have completed 210 questionnaires involving 146 patients all around the world. And the project is all developed in collaboration with Dr. Ivan Torrente, that is a neurologist of the Hospital Policlinico of Milano. And last but not least, we are building a network of patient associations. So from the moment, we have like 8 patient associations that are collaborating in this project. And I want to say also that the project is already recruiting, is still open. So if you still don't have participate, you can contact directly this email, that is secretariagfp@gmail.com. And another project where we are collaborating with is the project of Dr. Sandona. And she is focusing on sarcoglycanopathies as well. And whereas the healthy subjects are presenting a healthy, well-folded protein, in patients, we can find both the absence of the protein, or in 65% of the cases, we found a dramatic reduction of a protein because the defective folding of this protein. So what they are doing is a pharmacological approach where using the small moleculars called GFTR correctors that they tried in patient derived cells or mice, and they saw that it is possible to recover the protein of folding. And then this protein can well establish in the sarcolemma, recovering consequently the force and strength. If you want more information about this project, you can contact directly in these 2 emails that we have here down, the Professor Sandona, that is doriana.sandona@unipd.it, or the other email. Another project where we collaborate with is the project Muscle Diversity Project, where we want to understand why the muscles of the same subject degenerate at different paces. So for the moment, we are established a beta-sarcoglycan mouse model that is mimicking the pathology in humans. And as well, if you want more information about this project, you can contact or you can visit the page diversitamuscolare.it. And finally, we want to thank the people that are collaborating with us, and Dr. Ivan Torrente, Dr. Doriana Sandona, Kathy Bryan, me, and Beatrice, our president, Beatrice Bola. We thank also the staff members of GFP, Claudia Paniga, Chiara Galoni, Lucica, Ana Rega, Irina, and Serena, and the people of different patient associations that are collaborating with us. As well, the people that are helping us with the software and the data security and the legislation and all of that. So thank you very much.
Welcome everyone to our patient roundtable number 1. we have assembled individuals who are living with limb-girdle muscular dystrophy or are caregivers of individuals living with LGMD. We're going to be asking some provocative, interesting questions and getting real insight direct from patients about how they feel about clinical trials, drug development, and we hope that the insights we all share today will help industry and other stakeholders as they develop trials in the future. So first off, we're going to go around the room and introduce everyone. I am going to start with Melissa Grove. Why don't you introduce yourself and tell us where you're from?
I'm Melissa Grove from Dallas, Texas, and I have limb-girdle 2I R9.
Thanks, Melissa, and thank you for being here. We're going to go next to Carles.
Hi, everybody. I'm Carles. I'm living in Rome, and I am affected of a beta-sarcoglycanopathy.
Thank you, Carles. We're going to go to Catherine Noonan.
Hi, I'm Catherine. I live in London in England. I have 2 children with limb-girdle 2D or R3, one of which who took part in the nationwide Children's Hospital trial in 2016, and the other one who has naturally acquired antibodies.
Thank you, Catherine. And now we're going to go to Brandy Benton.
Hi, my name is Brandy Benton. I live in Duluth, which is in the Atlanta, Georgia area, and I live with limb-girdle muscular dystrophy 2A.
Thank you. And then last we have Josh Thayer.
Hi, everyone. I'm Josh Thayer. I live in Brookline, Massachusetts, and I have LGMD 2B/R2.
Thank you so much, Josh. Thank you all for being here and for willing to share with everyone, just answering all these questions. We have a great first question that we're going to present to every person on the panel. The first question is, what do you feel are the biggest challenges as a patient in understanding the rare disease development process? And this would be for drug development. We're going to start over here with Melissa.
Yes, I think for many of us, there hasn't been any treatment for so long that really we've just been all told to just wait and really not be proactive because there wasn't a lot that could be done. So I think it's a very complicated process. And so from going from being completely passive to having to be very active and learn a lot of information, learn a lot of science, I think that's where the big challenge is going to be.
Oh, good point. We all feel like we went to get our master's degree in some kind of science, don't we? Carles, speaking of scientists, we have a PhD over here. So Carles is a scientist and an individual living with LGMD. Carles, what's your take on this question?
Yes, for me, the major challenge is to understand that trials are not yet treatments. So this is a very important point because they are very important in terms of science, but it should be taken for what they are. They are trials. Therefore, the biggest challenge for physicians and industry is to keep the life of families uninterrupted as much as they can. So, and then they should apply corrective measures when this is not possible. These for me are the biggest challenge that are around the drug development process.
Good answer. Catherine, how about you?
I think the biggest challenge for me, Kat, is really coping with pace and understanding why everything just moves so slowly, particularly when you are living with or are the parent of children with a progressive condition. I have found that when I've attended conferences and I've been able to understand a little bit more about the technical challenges that farmers or universities are facing, that does help me to be a little bit more patient. And so I think that's one of the challenges, both for the community to educate themselves, to Melissa's point, but also for the farmers and universities to help educate the community, not just when they've got big end milestones to announce, but also with updates along the way about what challenges there are, what technical decisions or choices need to be made. And I think that would help us all find somewhere a little bit more patience with the progress.
Oh, I agree. And Brandy, I know you have a lot of insights here because you are in health administration. So share your thoughts on this question.
Yes, so my master's is in public health, so understanding clinical trials and just having the probably not as in-depth scientific knowledge as Carles, but understanding how things work and, presents less of a challenge. But as someone who has a community of others who are living with other types of muscular dystrophies, I know that the questions that come through are echoed here, the timing and just the process and understanding the process and understanding how things are developed, I think is kind of the medical literacy, the health literacy, how things are communicated in ways that people can understand is probably a challenge. I think there's some other ways that people have been able to communicate how clinical trials work that are more layman's terms or, very simplified, and being able to make the information more accessible, especially to those who are living with the specific muscular dystrophy or whatever the clinical trial is focused on. I think that's the biggest challenge, one of the biggest challenges.
Oh, good answer. And then I want to go to Joshua Thayer. He is in the field a lot because he works with Jane Foundation and he's a lawyer, but he also has an amazing scientific background. So Josh, what's your thoughts on this question?
So this sort of goes to Melissa and Brandy's point to a bit, which is, we're dealing with very complex areas of the law and regulatory law in particular, which is not easily accessible to most of us without spending a considerable amount of time studying up on how the regime works. And then the substance matter is science that, Carles with his PhD probably can follow better than most of us. And so you have both the legal regulatory regime and you're dealing with scientific topics that are highly complicated. And so it does take a lot of dedication for the lay patient to get a leg up on just understanding even the announcements that are made by research institutions and pharmaceutical companies. And that sort of gets me to the second area where, I think it's tricky to follow drug development. And that is where the development is conducted and by whom it's conducted, right? So you start in a university laboratory and they're doing basic research. They might discover a compound and start developing it. And they're going to want to protect the patentability of that compound. So they will operate in a certain amount of secrecy until they file a patent application. And then they'll make these impactful publications, which are these long articles that are, use all of this technical language. And then eventually when it's licensed to pharmaceutical companies to continue with clinical trials and regulatory approval, they have to follow certain rules with respect to informing all of the public when they inform anybody about anything that could impact the trading of their shares. So they have to protect their potential investors. And so it's really not a regime that's designed for easy access to ongoing access to emerging data, which can seem really unfair to patients.
Yeah, these are all such good points. And, you all have brought something so valuable in your answer. The next question we want to ask, and I think this is a good question, do you have any safety concerns or anxieties about potential clinical trial opportunities for limb-girdles? for any of the clinical trials coming up? If so, what are your reasons for concern? And what areas are you most concerned about? I think this is a good question to start with Melissa. Melissa is a licensed professional counselor. She's been in the field of giving therapy to individuals who are living with HIV, and she runs a multi-million-dollar counseling center, Legacy in Dallas, Texas. So she's in the field of counseling, and I'm sure you're familiar with some of those terms that we just mentioned: anxiety, safety concerns. Answer that question for us, if you don't mind first.
Well, what we're dealing with here is a lot like the early years of HIV, where there were new technologies and new approaches. And I've helped clients for 32 years go from virtually no treatment to very sophisticated treatments. And I think with any new treatment, there's an anxiety, of course. Is it safe? Will this hurt me? Will this make me worse? I think that also with this that we're dealing with personally, I have concerns with the gene therapy. This is a very new technology. I don't know if I necessarily want to be the first guinea pig in on this one. I may want to be in a later study and not in the safety study. My other fear is that if I do a gene therapy or something that uses a vector, will I use up my opportunity to get an improved treatment later? Will I ruin my shot? And so I think those are some of the biggest concerns for me. I don't want to get worse. I want to get better, obviously, or at least stay stable. So I think that's one of the concerns I have is with all these new technologies, what are the off-target effects? How is that going to impact me? Even a very slight change in my functioning, something that would be mild for a, quote, normal person, can be very debilitating to my day-to-day functioning. So I can't really afford to lose anything more. And so I'm hoping what comes along will make me better or at least stabilize me where I am. So those are some of my concerns.
Those are good responses. Brandy, what are your thoughts on this question?
Yeah, so overall, like just participating in clinical trials, I understand that researchers are required to, follow strict rules to make sure that participants are safe in that aspect. I understand, but just like Melissa, there's a risk associated with participating that you just don't have it. You don't know the outcome, right? And you don't know how it's going to affect your body, or if if there's a control group, all the things that go into the trial. I think what's even more concerning than safety is knowing that without people to do the trials or without people to participate, that we can't advance the research, and without enough diversity. I mean, whether it's people of color, people from different regions, people with different experiences, that it can affect as well. So I think the part that I don't know if it concerns me, or the area that I'm challenged in, is what am I doing to myself to participate, or what am I doing to help the community, more, to help advance the science that I'm believing in and that I'm hoping for. Like, I don't want to push it off on somebody else to do it and then me benefit if I could participate as well, but then still have my safety concerns. So it's a interesting kind of push and pull of what do I do if I'm concerned about some of the exact same things that Melissa said without repeating her, but also participating helps advance the research and it helps gets to a cure. So it's less about the safety concerns for me. It's about which one do I side with myself for now, or side for the advancement of science later for me and others.
Yeah, it is truly a balance. And then we have our own personal lives to attend to, and then circumstances, and, some of us have children, and it really be working jobs, taking off of work and, sacrificing there. How can we incorporate patients more into the process of drug development? We want to incorporate patients, and how can we do that? I know there were some great ideas that were shared with me by some of you. I think, Catherine, I want to start with you. Share with us some of your concepts and ideas that you had mentioned. These are great, I think.
I had a couple of things that I'd mentioned, Kat. I think the first one at quite a simple level is that I'd just really like to see patient representatives involved at every stage of the drug development. You know, we've talked about the communication barriers through legislation, but I think patients have got a lot of value to add in terms of outcome measures, in terms of communicating to the community in layman's speak. And I also think there's a lot of value in, for example, keeping patients involved after they've participated in a trial. It's really interesting that no one has asked me in the last 7 years for my feedback on participating over 2 years, being away from home for 6 months, spending over $30,000 to be part of a trial, but nobody's ever actually sat me down and said, okay, what went well, what didn't go so well, what could we have done differently, how could we make it work better, how could we encourage more people to apply? Nobody's asked me those questions, and I'd really like someone to pick up the phone and ask me. So that would be the first thing. The second thought that I've got is my children cost the UK government an enormous amount of money, whether that's in the health sector with their treatment and their therapy, whether that's the social care sector, housing, adaptations, equipment, specialist residential schools and education, and a whole host of other things besides. So that's the actual cost that they're incurring all the time. But then there's also a real opportunity cost for, you know, the part that they're not going to play in contributing to the economic outlook of the country, contributing to productivity and GDP. And to my knowledge, I'd love to be wrong, but to my knowledge, no one has costed the impact of this disease over my child's lifetime. And I have 2 children with the disease. And I truly believe that if we did understand that cost, governments, insurance companies, other funders of eventual treatments would have far more interest in putting in research money and actually getting involved upfront to try and get rid of this cost. Because as much as we all want a treatment to be better or for our children to be better, there is also an economic cost benefit analysis which has to stack up here. And I think patients have got a real opportunity to play a part in capturing where all those costs lie so that we can help people to do that. So that's where I'd really like to see patient involvement in the drug development process.
Oh, what a brilliant answer. Carles, I know you had some really good suggestions as well. Can you share yours?
Yes. So for me, to incorporate patients more into the process, we need to keep developing the work and the network of the patient associations. So for me, it is important that professionals get involved in the daily life of patient associations, because the process of drug development is very complicated. We said already, with laws, scientific reports, psychological outcomes. And for that reason, professional figures should take part as advisors, as members, or as independent figures in the patient associations to understand the process. But not just that. On the other hand, we need more. We need to give the professional tools to the patient associations so that the caregivers and the people, the affected people, could explain by their own point of view what is happening to them with the professional tools. Not the professional just explain it to them, if not the people, the same people that are affected, explaining with the professional tools what is happening there. And that will give us also the opportunity to be valuable for the society. Catherine now was explaining that we have a cost for the governments. But this cost, we can turn it back, or we can be worthy also with proposals, and these proposals should come from us, should come out from us. And this, for me, is the way also to be more involved.
That's a very, very good answer. And to your point, I think when our opinion is given, if we're asked to give an opinion and the community feels super strong on a particular opinion, I think it's so important that industry and the FDA really give a rationale for why our opinion or our feelings about it or our real strong advocacy efforts were ignored, because I think patients are kind of left with a question mark, like why was that not brought up, or why was that skipped over? I heard that from multiple patients. Why aren't these considerations being put into this trial? And I love the fact that we get this neutral opportunity here to share our thoughts and feelings. It's so wonderful. Here's another great question. What areas do you think should be further explored when designing trials for LGMD? And are there any takeaways as we move forward? Let's start with Josh Thayer on this question.
Thanks, Catherine. Yeah, what I would like to see further explored, and somewhat urgently so, are additional clinical outcome measurements. We're kind of still, I think, stuck in the past with walk tests and other functionalities that are often, if not unique to ambulatory patients, less prevalent among those of us who are further progressed in the condition. But in addition to that, each of the limb-girdle muscular dystrophies is in fact a distinct genetic disorder. And so not all of us progress at the same rate, and even more so, we'll have different patterns of weakness emerge as we progress. And that's actually even true within the subtypes, as well as across subtypes. So I would like to see the pharmaceutical companies themselves get more invested in this process of discovering, observing, quantifying, and publishing, and discussing with the FDA, how in a relatively short span of a clinical trial, we can get a nice sense of whether the drug is effective. And I think this ties in nicely with what Catherine Newham and Carlos were just saying, which is you can't do that without patient involvement. The patients really do know their own bodies. And the fascinating thing is that when you interview various different patients from completely different backgrounds, they'll all say this. And they'll often present very similar patterns of progression. We sort of feel like we're hearing our own story. And so there really is scientific validity in reaching out to patients, including them in trial designs. But it seems like the therapeutic research is going well, but it's still kind of hard to know how to measure improvement in these clinical studies and to be inclusive of those of us who are no longer ambulatory, as well as the younger ambulatory patients.
Oh, yeah. Josh, that's a great point. And, when we think about how much is focused on whether you're walking or not, and muscular dystrophy almost affects every muscle group. In some cases, the heart and the breathing muscles are not affected in some LGMDs, but in all cases, you're going to see some deficiency in the mutation in every cell in someone's body that's a muscle group. So you're going to see it, and it may be that it affects the legs first or the hips first or so on and so forth, but in 2I, it hasn't really affected my arms so severely yet, but it eventually does,
Right.
but that's a capturable, you can capture the decline there. You can see that it didn't decline as quickly. It's almost like we're obsessed with the legs or something. It's
When say, but it only affects your legs, right? And I'm like,
yeah, exactly. Whereas there's so many other muscle groups that we could look at and really start to quantify progression. And when we start to look at natural history, that's data that's really gold that we really need to start capturing more closely. I think you've got a great point there. Catherine Noonan, I know you have some thoughts there too. Would you like to share on this question?
Yeah, I mean, having taken part in a trial, I decided that whilst there was a lot of thoughts that I do have, I really wanted to concentrate on one which kind of picks up on what Melissa and Brandy were talking about earlier, which is this idea that once you've taken part in a gene therapy trial, you develop antibodies to the viral vector that's been used, and you're therefore not eligible for any other treatment. And in terms of designing a trial, I think the concept of do no harm is a well understood concept in the medical community. But for me, that includes not leaving people worse off than they were before. And if you design a trial in isolation of that question, if you give someone a gene therapy treatment to a small part of their body or at a dose that's too low to affect any functional change, you've given them no benefit, but you've also denied them now the opportunity to get that treatment in the future. So for me, arguably, morally, you've left them in a worse off position than they were before. If a 2D treatment was announced tomorrow, I'd be thrilled for the community. I would be devastated for my son because he wouldn't be able to have it, having taken part in the trial. So when we think about designing trials for the future and what the takeaways would be, I really kind of feel that this concept has to be a fundamental part of the trial design. We're starting to address it with doing placebos in some trials and guaranteeing a full body systemic dosing of the treatment at the end of the trial. But you've even then got potential issues if the dosage wasn't enough for a functional benefit. So I really think redosing research has to be a fundamental part of a drug's development process, not something that's a nice to have that might come along at some part in the future. If you're going to give this to somebody, you have to know how you will ensure that they are no worse off than they were before. And I think that also includes developers making binding commitments to trial participants, whether that's about the treatment at the end of a placebo or whether that's about, what commitments their company is making to research into the redosing process, what commitments they're making to trial participants about bringing them back. And I really think that would go a long way to helping people like Melissa and Brandy and probably hundreds and hundreds of other people with the same concerns about taking part and helping to advance the science. It would really help allay their fears about taking part if they knew that the trial had been designed to not leave them worse off than they were before. So there's many things to say, but for me, that's really the big one.
Thank you so much. What great insight. Concerning the differences between countries and the drug development process, what barriers have you encountered in finding out about trials that exist? That's a great question. Let's start with Carlos on that.
Yes. Yeah, for me, I think it's evident. The most important barrier for us is the language. Because for that that are not English speakers, the fact to not have the information in our language is really a barrier. To understand the scientific, again, the bureaucratic or legal language is very complicated for those ones that are not familiarized to it. So to understand it in a language that is not your mother language is almost impossible. It's double complicated. So this is one point. Another point, another important barrier that we found is the the coordination. The coordination is very complex for many of our disease, as we are affected in a few people in each country. So we are making great efforts to coordinate and ensemble our voice. The same we are reclaiming for the physicians. Often at the institutional level, the chain of coordination is broken. We feel that it's broken. So we are submitted to repeated equal exams at distance of few days sometimes. And this is not for the matter of privacy of the data. It's for bureaucratic issues. And to overcome these barriers, we ask for more generosity in a scientific level and more coordination. And that are the two main points that I wanted to underline.
Thank you. Good point. Catherine, I know you have some things you wanted to share on that as well.
Yeah, well, Carles, I speak English and I find it hard as well. On top of the language barrier, I think it is just tricky to find out what's going on. You know, there's the clinicaltrials.gov website in the US. I had a look and I found there's now a WHO clinical trial website, which didn't seem to have too much on it. I personally have Google Alerts set up. I'm a member of patient groups, Facebook pages. I attend conferences. And even with all of that, I do find it's hard to know what's going on, what's starting, how you get involved. And even when you do find a trial, I think sometimes the application process isn't clear and easy. You know, I'd really love to see a standard set of information that's required, an end date by which trial applicants will be chosen, so that you don't feel like you're part of an open-ended beauty pageant trying to sell your wares to someone to get them to pick you to be in their trial, which is kind of how it can feel. I think Carles' point is a really good one about international collaboration. You know, as I said, I spent a total of 6 months overseas over the course of 2 years. I had to leave an 8-year-old child behind for that whole time. And we spent a lot of money on travel. We had difficulties getting visas to return to the US on a regular basis. And, you know, if there's one thing that's happened that could be good out of the last 2 years from COVID, it's shown us that when there's enough impetus and when there's enough desire and will, international collaboration, information sharing, getting rid of those communication barriers, it is all possible. COVID has shown us that. So I'd really like to see trial centres open in multiple countries for a trial that's being run out of one. And that would have the knock-on benefit of the patients in those countries finding it easier to get information about that trial, because by definition, an outpost of it is now happening in their country too. So we need the will of the COVID solution applying to our international collaboration.
Mm-hmm. Very good point. I've heard patients say if 2 million people were affected in the United States or over in Europe with limb-girdle, that there would have been a treatment. And it makes sense. It's a rare disease and there's not as much interest in finding the solution. Very good answers. Okay, our last question, and then we will close. This question is about double-blind placebo trials. And so how do you as patients feel about double-blind placebo trial designs? And are you willing to sign up when you see placebo-controlled trials? Let's start first with Melissa Grove.
Yes, I have no problem with that. I have no problem with potentially being in a placebo group, as long as at the end of it, I'm guaranteed that I would be able to be in the study. And from my thinking, it's almost like an extra layer of safety, because if I get in the study and I'm in the placebo group, there will be even more data by the time they go to dose me. And so for me, I don't think it's a huge negative. Now, obviously, we run the risk of getting worse while we're on the placebo, but I'm getting worse anyway. So to me, I don't mind being in the placebo group at all if it comes to that. I have had friends who have done extensive studies. They've been in a few studies, and with using up all their vacation time for the whole year just to be in the study. And at the end of the day, the study didn't prove that it was doing anything. So being in a placebo study in that would be discouraging, or even being in the study, because it is a huge investment of time and money and energy, and it is a diversion from your life. It is a commitment, as Catherine was speaking so well about. I do think that if it is a required thing, and I can understand why, I'm all for it. I'm up for doing anything that moves us along, because for 33 years I've had no treatments. So if potentially I have to wait 6 more months, I'm okay with that.
Good answer. Josh, what's your take on this question?
So I agree with Melissa, and in fact, I've enrolled in a double-blind study once. Turned out I was on the drug. I actually found out midway through by accident, because this trial coordinator told me and another participant who showed up on the same day that the pharmacy said the delay is caused by having to mix 2 doses of drugs on the same day. So, oops. But where I differ a little bit maybe is, I think we're getting to a point, particularly with the limb-girdle muscular dystrophies, where there should be enough natural history data collected that the FDA can rely on historic controls, right? Because we know basically that if you have LGMD, any one of the subtypes, because they're different diseases, but none of them reverse themselves on their own. In none of them, do the biomarkers change radically over a few months period of time. They're generally slowly progressing, and that's tricky. But you can establish that through historical data without having to, ask a group of, say, 10, 20 people to travel to a study where they'll either get the drug or not get the drug, and where if they don't get the drug, their progression, will be predictable in any event. So I understand why they are important. But I think once you've established that the safety, which is going to apply whether or not, safety issues will apply whether or not you have the disease, I just am not convinced that the placebo control is required for the efficacy side of things. Because if there is a significant improvement, and that goes to why I'd like to see more clinical endpoints developed that are more subtle. So yeah, I would like to see a day when we don't need placebo controls anymore, at least for muscular dystrophies.
That's a good answer. And, there's so many good points. I wish we could just spend another 30 minutes just going through all of some of the stuff y'all mentioned was so good. But you made such a great point just now, Josh, about the natural history data is really there for a lot of the subtypes. And something to point out to patients as well, you can enroll in more than one natural history study. You're not limited to just one. A lot of patients think, oh, I'm in one, therefore that's all I need to do, because all that data exists and that's all. We just need one. But patients don't realize that there are industry-sponsored natural history studies. Some natural history studies are taking place in academic institutions and they're not sponsored by industry per se. And so a lot of times patients don't understand those nuances. So I encourage patients to enroll in natural history studies anytime you see them, because when industry is looking to have clinical trials, they also have to identify and locate patients. And natural history studies are a good way to naturally locate patients. And it's not necessarily a guarantee if you're in a natural history study that is a clinical trial readiness natural history study that you will be in a trial or anything. But it is a good thing, I think, to be in natural history studies, because it kind of sets the stage that easily that data is there. They locate that family, that patient, they know their age, where they're at in progression. And it's much easier to locate that patient than it is to go through a big registry. And sometimes the registry doesn't have the date wrong, the age right. They don't know the stage of progression. There's a lot of different data points they don't have. So I always tell patients, do enroll in natural history studies. You are not wasting your time, though a lot of patients think they are, but you're not.
No, I mean, for me, it's a nice psychological benefit to just feel involved in the process. And it is, I mean, it's also can be burdensome. I mean, if you have to travel and you have a 350-pound wheelchair, it is a commitment. It is a commitment, but it's not a waste of time, and it's safe. And I don't know, it's a way to be involved. So.
It is. And it also is a good way to get the best care as well, because you do have typically top researchers and clinicians looking at you. And that is a really nice benefit, because you're not necessarily going and getting localized care. You're getting really usually care in some of the best centers for limb-girdle, usually taking place in GRASP LGMD sites. So that's another benefit of doing natural history studies, I think. Well, guys, you guys all seem so knowledgeable. You are so knowledgeable. And I just know that, for years to come, people are going to go back and listen to this roundtable discussion, because there's so much insight. So thank you so much for sharing your personal stories, for willing to get on a video and share them with the entire world. It was very brave and courageous of each of you. I want to say thank you, and you guys are just wonderful for sharing. And y'all have a great evening. Thank you. Thank you to Roundtable 1 for your excellent insight. We now welcome our expert speakers for our U.S. advocacy session. We want to warmly welcome Paul Mehlmeyer, the Vice President of Public Policy and Advocacy at the Muscular Dystrophy Association. And we also welcome Sharon Hesterly. She is a PhD and the Chief Research Officer at the Muscular Dystrophy Association. They will share their insights with us on ways patients can get involved in the regulatory drug process. After their presentation, we will have a short Q&A as well with Dr. Brad Williams and Sarah Emmons from the Jane Foundation.
So thank you, Catherine, for that introduction. And I am thrilled to be here talking to you now about how a good idea becomes a therapy. And it is a complex process. It's kind of like launching a space shuttle, but we're going to make it as simple as possible. So their goal on the side, you see this little guy over here. It kind of reminds me of the Schoolhouse Rock shows that those of you of a certain age will remember, and how, a bill becomes a law. So we're going to talk about how a good idea becomes a drug. And it usually starts down here at number 1 and 2. We'll see number 1 in a minute, but with a granting agency. So NIH, MDA, one of the many limb-girdle organizations who fund research, will fund what is a good idea from a grant applicant. So these are usually academic investigators, professors, and other researchers at universities who are doing work in their labs, and they come up with a great idea. They submit a grant application, we review it. If it rises to the top, it gets funded, so they get the funds, and then that goes to the university, where work continues. So if the idea is for a therapy, what usually happens at the university level is what we call proof of concept. So the investigator will test the idea, sometimes in a laboratory dish, sometimes in an animal model, to say, you know, this really does do what I thought it was going to do. So this is working. So that's sort of our proof of concept that this could work. This could be a therapy. And if it looks promising enough, what typically happens is the university patents the idea. And then if it's really promising, they're able to out-license that. So what it means is that some company will come along and say, hey, we also think that's a good idea, and we want to help you develop the drug. So academic universities aren't really set up to do hardcore drug development. So that's when things usually transfer outside of the university setting and into a company. And so what that looks like here is the biotech or pharma will pay money to the university to obtain a license to that information, that knowledge that has come out of the university. And then they will continue testing. So then they move into sort of the preclinical and clinical phases of testing, where they say, okay, we've seen your proof of concept. It looks like this is a good idea that could work. We're going to really put it through its paces. We're going to find out what dose is required. We're going to do all of the tough work. And then we're going to test it in humans when we know that it's safe and that we think it's safe. And if that all looks good, they submit this huge dossier to the FDA for a new drug application to say, we'd like to make this a drug. And then if it gets approved, that's where you get your drug. So that's the ultra simple model for what's actually happening. And you can kind of see everything from the idea to the funding, the university to a company. And not every drug takes this exact path, but this is probably a pretty typical one. So just to give you an example, so this is from the DMD space, but it's a good example, I think. We start up here in the corner. This is Dr. Steve Wilton, who in the mid-1990s had this idea that you could actually use certain pieces of DNA to cause, to change the way cells process their mutated genes and actually produce a functional protein. So that's sort of the short idea of, his idea. And he published this idea. He submitted a grant application to MDA saying, I think we can make this a therapy for Duchenne, this kind of wild idea that we had. And our reviewers agreed, and we funded it. And it looked good enough that Sarepta, so this is where the company comes in, a company called Sarepta came in and licensed that idea from the University of Western Australia, where Steve Wilton resided. And from there, they developed it into a drug. So the first drug approved for Duchenne in 2016 was based on this work. So this is sort of what this chain looks like in action. And the funding, came from MTA, but several other organizations, NIH, the Australian government. And so what actually happens here is as these drugs go on the market, is a certain amount of money gets paid back to the university, and then the university in turn redistributes it sort of proportionally to everyone who put funding into this drug. So we get a very small amount of money because this drug was approved, because we helped fund the research. But that's, again, an example of sort of a typical path that a drug could take. And that's just showing that there were additional drugs that came from this same research project that was back in the 90s. But one thing to call your attention to is this started in the mid-90s. You see the dates when these drugs were approved. This is a bit of a long pathway. I think we're doing everything we can to shorten it, and you will hear from my colleague Paul in a few minutes about how you as an advocate can help shorten this pathway to drug approval. So just quickly, a little more on the stages of drug development. Again, I've put years. These aren't hard and fast. They can, be more or less depending on what happens. But in general, just some ideas of what we're doing. So basic research. This is what's happening at the university and the laboratories. And the kind of questions that you're asking are like, I've never seen that before. That's odd. How does that work? How do these pieces all come together? Does it work the same in every species? Does it change over time? And that kind of gives you an idea. Maybe this is something we can target for drug development. So then you move to the next stage and say, well, can we change the way this works? We see what happens when you have a mutation in the gene. We kind of see what's going wrong. Can we make it go right? What could I tweak to make that happen? And so you can screen and look for new drugs that will help actually put things back on track in the cell. When you have something that looks promising, then you do what we call the proof of concept testing that I talked about earlier. So this is where you ask, does this change, this compound, it seemed to work in the laboratory dish. Does it now do the same thing in this animal that might have the same type of genetic mutation? Are these results reproducible? Does it work every time? It wasn't just a fluke, and is it generally safe? If all of that looks good, then you move into the stage called translational research. And what you're really asking here is this going to be feasible to test in humans? So you have a whole bunch of questions you have to answer before you ever put a drug into humans. And this is like a sort of really intensive testing phase. So you want to understand, how high can you dose this compound before you get some kind of adverse event or toxic effect? Anything, if you dose it high enough, even water can be toxic. So the goal here is just to figure out what is that window? Where do we have the safe space that we can work in for a dose? What is the ideal dose? When should you start dosing? In some cases, if you have a disease where the onset is in the teens, say, 15, 16, do you want to start dosing early if you know you have the genetic mutation? Do you want to wait until after you already have symptom onset? How often should you dose? Is it something that you have to take a pill or inject? Do you need to do that weekly? Do you need to do it daily? All of this stuff gets worked out, and you don't know the answer for sure, but the goal is to have a good idea what the answer is before you go into humans. So you feel pretty comfortable. You kind of know what you're going to expect in humans, with the caveat that animals aren't humans and that this doesn't always translate perfectly. And that's why drugs don't always succeed. But then you get to the clinical testing phase. So there are, very key questions here. Who is best to test, and whom should we best test this drug? If you take all comers, you don't want people necessarily participating in a trial who might have other underlying health problems, so that if there is a problem with this drug, it might actually make it worse. So you don't want to make anyone worse. You also want to be kind of consistent in who you test things in, so that you decrease the number of changes. You want to make sure that any change you detect is actually due to your test drug. So that's why you see all those stringent requirements. When you look at clinical trial eligibility requirements, they're always, you have to be between this age and not have any of these other problems. And, all of that, it's frustrating because it limits the number of people who are eligible, but it's all designed to give this drug the best chance it has to work. And if you can show that it works in a sort of narrowly defined population, usually the goal is then to say, let's do a bigger study, or if it's going straight to approval, let's see how it does in the general population. But you want to give it its best shot possible. So that's what this is about. So again, when you get to human testing, I know you keep seeing these different steps for every stage of the way, but like I said, it's like launching a space shuttle to some extent. The amount of work that has to be done to make sure this drug is safe and effective. But in human testing, it starts with some acronyms, and I'm sure Paul will have some acronyms in his slide too. But ones you should know about drug development testing is if you have a compound that's never been put in humans before, you apply to the FDA and say, I really want to test this compound, and you submit all the information that you've gathered, and they will issue, and this is called an investigational new drug application. And what actually happens is you submit that to the FDA, and it's sort of a shot across the bow. It says, we're going to do this, and here is all of the paperwork for it. And if you don't hear from the FDA within 30 days, you're good to go. If you do hear from them, usually they're asking for additional data. And so you can't proceed until, you've satisfied all of their questions. So there's a whole lot of words under here that are pretty complicated, but these are all the pieces that go into that investigational new drug application. And these are kinds of things you're testing in a phase 1 study. So a phase 1 study traditionally is actually a drug or compound you tested in what we call healthy volunteers. So people who don't have the disease or the problem that you're trying to address. And all you want to understand is in that background where you don't have a disease background, is the drug safe? Is it going where it's supposed to go? Does it stay in your bloodstream long enough? If that all looks good, then you might move to the next phase, which is phase 2. There you're primarily testing safety. So now you're testing the drug in the population that it's intended to treat. So this would be where, people with limb-girdle would be in the study. But the primary goal, again, is safety. So you want to make sure it's safe. But, people always want to, ask at the same time, is it working? Usually the study won't answer the question completely if it's working, but it'll give you hints that, yes, it's likely to work. These can be placebo-controlled studies. That means that you'll just have a group that doesn't know, certain people, if they're getting drug or placebo. And that's just a control for those mental things that happen when you get excited about something, or sometimes you come into it thinking it's definitely not going to work. So you just want to kind of control for that. And so if you don't know if you're getting drug or not, then those sort of like mental thoughts can't influence how you perform on tests, for example. So in the final stage is phase 3, where you really want to understand if the drug is working. So you still test safety. You set up safety throughout the whole thing, and you're testing it in larger and larger numbers of people. Phase 3, you might have sort of wider eligibility criteria. So, the group of people participating in this study might not be as similar as the ones in phase 2, but you really want to understand this drug work in a wider group of people. If that all looks promising, then you can submit what we call an NDA, which is a new drug application. This is, stacks of paper that would go from here to the ceiling. These are really, really big dossier of everything you've learned in all the clinical and preclinical testing up to this point. And it contains sections on manufacturing, how the drug is manufactured, all of the safety, all of the clinical results, all of the background. And the FDA has a certain amount of time to review that. And if it happens to be a biologic that you're testing, so this is something typically that would be an injectable, so it's made of DNA or protein, then you will also need a BLA, which is a biologics license application. If all of that gets approved, then the drug can be marketed and can be available, and you can go to your doctor and have it prescribed. But this is the crazy path that you go on. It's very long. And at any step here or even previously, a drug could fall out and we could just say it doesn't work. And so I know an example of a drug that we reviewed years ago really looks good in the animal models. It looked like it could do everything it needed to do. But when they got to this human testing phase, even in phase 1, this drug just, it was a really oily kind of drug. It didn't go into solution very well. And they just weren't able to deliver it. They tried all kinds of things and couldn't get this drug to just hit its target. And ultimately it did go through a phase 2 and a phase 3 test, but eventually it failed. And it was maybe not because the compound was bad, but just it just wasn't right. It was hitting the right target, but it just, didn't have the right properties to be a successful drug. So this is where you learn how all of this works. So just to take a peep at what's happening in limb-girdle. So this is what's all going on behind the scenes. So where are we in the therapeutic landscape for limb-girdle muscular dystrophy? So this is just a quick rundown. I may have missed something. My colleague Chris is fabulous at staying on top of these things, but he and I both say it's so, things move so quickly, it's hard to keep up. But you can see that some of these drugs, for example, this BBP 416 from ML Biosolutions is in a phase 2 study. So it's in a clinical trial, and people with limb-girdle are participating in that study. And also a couple of the Sarepta drugs, and these are gene therapy approaches, are also in a phase 2 study. There's one drug in phase 1, and this is a small molecule from Edgewise, and it just means they're testing it in healthy volunteers. So nobody with limb-girdle has been tested yet. And they haven't even disclosed what form of limb-girdle they might test their drug in. So if we look at, I guess one point here is that this is, most of these projects are preclinical. So we're still sort of early days in our limb-girdle drug development path. And I think there'll be a lot of things that we learn from these first drugs that are in early phases of testing. So if you look at the type of therapeutics that are in development, this is just a different breakdown. The majority are actually gene therapies. So in the dark blue, you see gene therapies. At least one cell therapy, one sort of undescribed therapy. I don't think they've determined yet which direction they're going to go. And then small molecules here in the sort of aqua color. So there are several of those as well. But it is a preponderance of gene therapy, which is interesting. So what are some challenges for drug development in limb-girdle? And I think the primary one is because there's so many different types of limb-girdle, any of the individual types actually have relatively low numbers of people affected. And the challenge there is when you don't have large numbers, you sometimes have low commercial interest. Clearly, it's not too low because we just looked at, a whole long list of therapies that are in development. But those are largely for the more common forms of limb-girdle. So what do we do about making all the forms of limb-girdle look like good candidates for drug development? And that comes into what we call de-risking strategies. And that involves creating the information and the tools to make these different subtypes of limb-girdle look attractive for drug developers. And so, say we know all the natural history and we have endpoints and we can identify all the patients. Well, even though there aren't a lot of people, that's a whole lot of work the drug company doesn't have to do. So just quickly here at the end, a couple of examples. So de-risking natural history in limb-girdle. Some great examples of how we can do this. There's not enough of natural history. So natural history is how a disease will progress if it's untreated. And what is the impact on the people living with that disease? So there are a couple of things. This is not exhaustive. There are more projects than this funded, especially this is sort of a US-centric view. But the GRASP Limb-Girdle Consortium, for example, is an academic consortium that's designed to help identify people who don't have a genetic diagnosis. And also improve natural history. There's a limb-girdle clinical research network that's funded by multiple groups, including MDA, NIH, and others, and it has multiple sites. And so that, again, is designed to help collect that natural history. And registry, so MDA has the MOVER registry where we collect data from clinic visits. TreatedMD has extensive limb-girdle registries as well. The MOVER, for example, we have 106 limb-girdle participants, and that represents 18 different subtypes of limb-girdle so far. So we don't have any limit on who with limb-girdle can enroll, so any subtype. But all of this gathers data, and all of this data helps de-risk drug development, which is really important. And just a quick final example, how do you de-risk clinical trial recruitment? So if you're doing a trial in a subtype of limb-girdle where you don't have a lot of people, finding those people is really important. And there are a lot of ways to do that. And I'm not going to walk through each one of these individually, but just to say, I think, all of the organizations in this space who support limb-girdle research and advocacy and education do a lot of these things, from magazines and newsletters that advertise trials to educational things like this, exactly, sort of symposium to talk about awareness of trials and what's required. And those need to be aimed at both potential participants, but also clinicians. Your doctor needs to know about these trials too. So there's a whole education component to make sure that doctors are up to date. So I won't read through all of these, but these are all things that we do that, organizations that you support, participate in, and that you yourself can do to help de-risk that clinical trial environment to make it easy for drug developers to want to be in that space. So with that, I will stop here and I will turn it over to my colleague, Paul Millmeyer, who heads MDA's advocacy efforts. So thank you all very much.
All right, well, thank you very much, Sharon, and good afternoon, everybody. It's great to be here today, and Sharon, it's an honor to share the virtual stage with you. Today, I'm going to pick up where you left off, talking about how the LGMD community can be involved in advocacy as it pertains to therapeutic development. And really, there are three areas, venues, I guess you could say, in which advocacy as it intersects with therapeutic development is particularly important. The first is at the NIH, where a lot of that basic and translational research is funded and is guided. The second is, of course, at the Food and Drug Administration. FDA regulates products that are under development, and there are many opportunities to get involved there. And then finally, at Congress. Congress, of course, actually passes the laws that govern what NIH and FDA does. So Congress and advocating to Congress on therapeutic development is also incredibly important. So let's start with advocacy at the National Institutes of Health, because there are really several opportunities for the LGMD community to get more involved. Here in front of you, you see an org chart, a very convoluted org chart of the NIH. As you can imagine, there are lots of different institutes working on lots of different things, but really, I wanted to highlight just three, because these are the three that can be most impactful as it pertains to basic and translational research in LGMD. The first is the National Institutes of Neurological Diseases and Stroke, or NINDS, who of course are most in charge of funding research in neurological conditions. The second is the National Institutes of Arthritis and Musculoskeletal and Skin Diseases, or NIAMS. And they, of course, fund research into, within the context of LGMD at the very least, the muscular aspects of LGMD. And then finally, the National Center for Advancing Translational Sciences is focused on, well, that very thing, translational sciences, actually translating these basic research discoveries into potential therapeutic possibilities. They also are where the Office of Rare Diseases Research is housed, and a lot of the rare disease focused efforts at NIH is conducted. So within the NIH, there are really several opportunities for those with LGMD and their organizations to get more involved. Within each institute, for example, there are advisory councils. NIAMS has an advisory council. At NIMBS, there's a National Advisory Neurological Disorders and Stroke Council. And then at NCATS, there's an advisory council as well. And these are made up of some researchers and clinicians, but also patient organizations and patients themselves to advise these institutes on their direction and what they should be prioritizing. Of course, there are program officers in each of these centers that are responsible for the actual funding of the research within LGMD and, of course, other diseases as well. But we've known many in other disease areas who have actually gotten to know their program officers and been able to help guide these program officers on what is most important to fund as it pertains to, in this case, of course, LGMD basic and translational research. And then each institute also has a yearly gathering that they put on in which everyone kind of within that community gets together and talks about what NINDS or what NIAMS or what NCAT should be doing from there. And I also want to focus just for a second on the rare disease aspects of it in NCATS, because at NCATS, there's the Office of Rare Diseases Research, and there are many opportunities to get involved there. Not only, of course, meetings and other public opportunities, but there's the Genetic and Rare Diseases Information Center at NCATS, as well as efforts to accelerate the development of gene therapies. And as Sharon already said, there's actually a lot of gene therapeutic development happening right now within LGMD, and NCATS is involved in trying to accelerate that research, trying to knock down some of the barriers that research may be facing, and so forth. So within NIH, there are several opportunities to actually be influencing the basic and translational research that NIH is funding and encouraging within LGMD. Moving along to the FDA, because of course FDA plays quite a prominent role in regulating products and guiding new products. Hopefully that NIH research, that basic and translational research, has actually led to a new potential therapy. As Sharon shared, there's lots of therapies within preclinical stages. Of course, a lot of those therapies did receive NIH funding. And from there, it's when the FDA steps in at that IND process that Sharon already started with. And it's from there in which FDA is influential in regulating these products. It's important to note, though, just so we are clear on what FDA precisely does, FDA doesn't create new medical products, nor does it set prices or create laws or make decisions on insurance coverage. All of these things are very important, but we oftentimes hear from some advocates just a little bit of a lack of clarity in what FDA precisely does. In this case, FDA does certify that the benefits of a medicine actually outweighs the risks of taking that therapy, and that the new product that could be approved by FDA is safe and effective for the population is indicated for. And in doing so, they consider patient viewpoints and experiences on what is actually meaningful, what benefits are substantial enough to potentially outweigh the risks of the product, and what does effective mean to the patient community. So FDA, as you can see from the right side of your screen, even if it doesn't do the stuff on the left, it's still very important with what it does on the right side. So to start with another very convoluted org chart, and I've actually, again, highlighted the areas that I want to touch on. There are many areas of FDA that are very important to those with LGMD because they, of course, regulate products that those with LGMD could eventually be taking. Now, of course, there are many parts of FDA that don't necessarily intersect with the journey of those with LGMD all that substantially. You can see there's a Center for Tobacco Products, perhaps veterinary medicine. All of you have pets, that's important, of course, but in the context of today's presentation, we're really going to be focusing on the medical product side of the FDA. And I want to start at the very top, at the Office of the Commissioner, because the Office of the Commissioner actually has several offices or departments or entities within the Office of the Commissioner that are very impactful for rare disease drug development and, of course, LGMD drug development. Within the Office of the Commissioner, there's the Office of Patient Affairs, and this entity is kind of the front door for patients and patient organizations who are looking to get more involved with FDA. It's the Office of Patient Affairs that coordinates patient listening sessions, and I know that there have been patient listening sessions within LGMD already. And then this office is also looking for educational collaborations with outside organizations such as MDA. MDA has partnered with this office before to bring educational opportunities to the neuromuscular disease community more broadly, and there could be similar opportunities specific to LGMD that this Office of Patient Affairs also could target. In addition, the Patient Engagement Collaborative is made up of patients, individuals with diseases that can then actually advise FDA on how to be more patient-friendly, advise in particular the Office of Patient Affairs. Also within the Office of the Commissioner, there's the Office of Orphan Product Development, and it's this office that actually dictates and decides what a rare disease therapy is. They evaluate if this treatment is actually intended for a rare disease. And they don't only do that, they also actually offer grants for therapeutic development. They have the Orphan Product Grants Program that funds rare disease treatment development. And then, of course, within the Office of the Commissioner, they focus on overarching FDA initiatives and opportunities because, of course, they kind of sit above it all. So consequently, they're going to be looking for ways to guide the agency across all of their centers, all of the medical centers, in ways to be more patient-friendly and to accelerate therapeutic development. Now, these opportunities on your screen are more focused to organizations, more focused to an MDA or to an LGMD organization for that matter. But there are opportunities for individuals to get involved within what the Office of the Commissioner is doing. Of course, within the patient affairs staff, I already mentioned the Patient Engagement Collaborative, an opportunity for individuals to get more involved. And if you're interested in participating within the Patient Engagement Collaborative, you can email patientaffairs@fda.hhs.gov for more information. Also, there's the patient representative program. And as a patient representative, an individual with LGMD could actually serve on an advisory committee that the FDA holds when they bring in outside experts to advise FDA on what decisions they should be making about a new product. And more information about this program, there's a long URL that I'm sure you cannot click, but if you certainly just went to the good old Google machine and searched patient representative program at the FDA, you could find more information from there. So to go back to our org chart, to move away from the Office of the Commissioner, I want to move down to the left to the Center for Drug Evaluation and Research. Now, this is the entity within FDA that regulates small molecules. So you heard Sharon mention small molecules in her presentation. So generally, the kind of the pills, the capsules, the drugs that come in the pill bottles. Those are traditionally thought of as small molecules. And it's the Center for Drug Evaluation and Research that regulates those. Within CDER, there's a Professional Affairs and Stakeholder Engagement Office that is kind of CDER's front door to patient organizations looking to collaborate. And there's also the Rare Diseases Program, in which rare disease experts, and these are generally clinicians and other medical professionals who are very familiar with the rare disease community, advise the different parts of the Center for Drugs, or CDER, on how to actually focus on those with rare diseases and really regulate and make decisions in a way that is patient friendly. Now, of course, CDER has its own org chart, which is every bit as convoluted as all the other ones that I've shown you. And here you can see that these offices are generally divided out in kind of areas of medicine, I guess you could say. Now, for our purposes today, we don't have to be worrying about the Office of Oncologic Diseases, but we should be worrying about the Office of Neuroscience, and individually within there, the Division of Neurology 1. Also the Office of Rare Diseases, and of course within there, the Division of Rare Diseases and Medical Genetics. These are some of the review divisions within FDA, and specifically within the Center for Drugs, that actually make the decisions on whether to approve a new product or not. So it is here where it's actually really great to get to know these offices, because there will be times in which they might be considering a new product within LGMD, and they might have questions. They might not know precisely how to approach something with an LGMD. And so consequently, if they know the organizations with an LGMD, perhaps even they know some individuals, that they can turn to the actual experts in their disease, which are the patients and the patient organizations themselves. To continue within Center for Drug Evaluation and Research, patient-focused drug development is one of the best ways in which patient organizations can actually offer the patient perspective to the Center for Drugs, as well as actually the Center for Biologics, who we'll get to in a moment, but the Center for Drugs on the viewpoints of patients on what risks they're willing to tolerate within clinical trials, what structures they would prefer within a clinical trial, what benefits to be looking for from a new therapy. And I do believe there actually is a patient-focused drug development meeting in the planning process right now for LGMD. So that really should be able to educate the FDA on precisely what it's like to live with LGMD. And for more information on this, of course, you can always email patientfocus@fda.hhs.gov as well. And then finally, since Center for Drugs, of course, is the entity that is responsible for not just approving the therapy, but overseeing the development of the product itself, there's a lot of opportunities to get involved with the development of clinical outcome assessments or other drug development tools, which can actually accelerate the development of a treatment itself. This is kind of the next generation of how to measure the experiences of a disease in a meaningful way. The Center for Drugs has opportunities for organizations and individuals to get involved there. Similarly, there's a Rare Disease Cures Accelerator program, which is developing a core set of patient-focused measures, and they're partnering with CPATH and NORD on that effort. There's a policy office, of course, within the Center for Drugs, and then hopefully this will never have to be experienced by those in the LGMD community, but there can be shortages of therapies as well, and that's when the drug shortages team comes into play. Okay, so I'm going to zoom back out from the Center for Drugs, and I'm going to move quickly to the Center for Biologics, because it's the Center of Biologics that Biologics Evaluation and Research is full name, that actually reviews in the same way that CDER does, but in this case, they are the ones who review the gene therapies and the cell-based therapies and some of these complex therapies that you're not going to get in the pill box. You're instead going to have those infused, or you're going to have those injected, whatever it happens to be. These are oftentimes the complex therapies, but it can also be quite life-changing as well, especially in the context of gene therapy. Now, CBER also participates within patient-focused drug development, and they're also working on the development of drug development tools. They also have a rare diseases program within the Center for Biologics that can plug into the efforts across the Center for Biologics to ensure that the rare disease voice is heard and that it's understood. Within CBER, similar to CDER, there are review divisions responsible for a variety of different kinds of therapies. In our case, within LGMD, the review division to know is the Office of Tissues and Advanced Therapeutics, or OTAT. This is the office that reviews all gene therapies and also cell-based therapies. And actually, it's a former head of an MDA care center, Dr. Wilson Bryan, who is the head of the Office of Tissues and Advanced Therapeutics. So there's someone familiar with the neuromuscular disease community who heads this effort. Now, of course, to get more involved within CBER, one could email CBER patient engagement at fda.hhs.gov for additional opportunities to get more involved. Finally, the last center I want to mention within FDA before moving along to congressional opportunities is the Center for Devices and Radiological Health. And this is the center that regulates all these medical devices that can be just as impactful on the life of an individual with LGMD as any other medical intervention. We're talking about wheelchairs and vents and trachs and BiPAP machines and other mobility devices and diagnostics. Really, the list goes on. If it's a device, if it's a technology of any kind that helps diagnose or treat a disease, then it's going to go through the Center for Devices and Radiological Health. Here, there's a program called the Patient and Caregiver Connection, which actually MDA is part of, that allows for organizations to advise CDRH on patient-focused matters. And MDA was very privileged to advise FDA on how to approach the recall of certain CPAP, BiPAP, and respirator devices because we are part of this Patient and Caregiver Connection. So through that, organizations that serve those in the LGMD community are able to actually advise FDA on how to approach things pertaining to devices. There's opportunities to conduct patient preference information studies on medical devices, as well as opportunities to simply just get to know patient-focused leaders. Dr. Michelle Tarver is leader of this entity, and I know she's always happy and willing to talk with communities about how the Center for Devices is impactful for those within the LGMD community in particular. So those were all, again, kind of organizational opportunities, but for individuals, the Patient Engagement Advisory Committee is really a great opportunity, because this is where individuals with the disease in question, whatever it happens to be, can actually participate on this advisory committee and advise FDA on how, and in this case, the Center for Devices, on how it should be operating. There have been Patient Engagement Advisory Committee hearings on clinical trials and on remote assessment technology, also artificial intelligence. There's some really interesting ways for individuals to be able to contribute their viewpoints to the Center for Devices on how they should be moving forward in regulating and also trying to spur innovation in these areas. Two more critical opportunities before I close with some opportunities up on Capitol Hill. There are a lot of public meetings that FDA holds that traditionally have been in person, but of course during the pandemic are now virtual, that are really a good opportunity to get the latest from FDA. And those can be found at the URL here again that I'm sure you can't click, but you can do a quick search for that URL and find all the list of public meetings that they're holding. And if they're of interest, I encourage you to attend. And of course, if they were in person, there'd be good networking opportunities, but virtually maybe not quite as much. And then finally, FDA publishes guidances on FDA's thinking on anything to do with drug and device development. And there are opportunities from there for organizations, but also individuals to comment on these guidances and to contribute your thoughts on, is FDA striking the right balance with whatever FDA might be targeting. These are oftentimes very wonky, very esoteric, but can be fascinating for those regulatory experts out there who would like to learn more about what FDA is doing. Okay, I'm going to close with just a minute or two on advocacy in Congress, because we've already mentioned NIH and FDA, but as I said at the outset, it's Congress that actually passes the laws that govern everything that FDA and NIH does. Congress can decide to change everything I just told you about if they wanted to, passing a different law. Thankfully, they don't want to blow things up that much, but they do like to pass legislation, or they at least try to pass legislation that can actually accelerate this process, actually try to pass bills that deliver more and better treatments to the LGMD communities. There's lots of opportunities to get involved. There's online opportunities, like if you were to sign up for our advocacy program, for example, at mda.org/advocacy, but also there are a lot of other advocacy programs out there as well. You can receive updates through these programs on how to be an online advocate for the LGMD community. You can email and tweet at your members of Congress, take action on government agency regulations through this very same system, and just sharing your story, just getting to know your lawmaker, even if it's through Twitter, even if it's through a phone call or maybe a Zoom call that you've requested with your member of Congress, just getting to know them and perhaps supporting legislation that is impactful to those with LGMD is a great way of getting involved. Of course, there's the traditional offline opportunities as well that are coming back at this point, such as doing a visit to members of Congress. You can do it virtually, but you could even do it in person at this point. Maybe not quite as much in DC because they're still figuring out the security situation up there, but you can go to your local member or your local office of your congressman or your senators to discuss with their staff what's important to you. You can always call, as I mentioned, write blogs and other pieces, organize in the community, all great opportunities as a grassroots advocate. And really, I can't put enough emphasis on this point. It's the power of your story that actually makes things move. It's not the rest of us in DC who, are advocating every day for the important bills, the important regulations, that it's not us that really kind of make the wheels turn up in Congress. It's the stories of everybody who is living with LGMD or their caregivers or a family member or, whoever has that story to tell from a personal perspective. That's what really makes things change up in Washington. So, there's opportunities, as I mentioned, through our Hill visits and elsewhere to really make your story known to your legislator to really try to drive them to support things, to support a variety of different bills that are impactful to the community. So what can you do now? As I mentioned, again, this is a plug for MDA, but you can sign up at mda.org/advocacy for our program. We have a number of active opportunities to get involved right now, some of which that aren't quite as focused on treatment development, but I'll just give them a quick shout out right now anyway. Urging Congress to fund and reform home health care is one. And then on empowerment and independence, urging support for accessible air travel, as well as disability employment legislation. So with that, again, thank you very much for the opportunity.
Thank you, Sharon and Paul, for those wonderful and informative presentations. So one question I wanted to ask is, can you share with us some of the lessons learned by other neuromuscular disease communities, for instance, Duchenne muscular dystrophy and spinal muscular atrophy, who've gone further through the drug development process, and what are some of their lessons learned in working with drug developers and with regulators that the LGMD community can benefit from?
Yeah, I can take that one first if you like, Paul. So one example that comes to mind is, remembering that this is an experiment and that it doesn't always work when you're testing drugs. And an example that I can give, in the Duchenne space, there was a drug that was being developed for a certain type of mutation in Duchenne. And, partway through one of the clinical trials, the Data Safety Review Board, the DSMB, it's another good acronym, looked at the data and said, there's just not enough evidence that this drug works. We're just going to stop the study. And it just stopped cold. And, sites were kind of notified. Some people heard directly from their doctor. Some people heard it through social media. It just became this huge thing. And I don't think anyone had thought that the trial would just be shut down very abruptly like that. And there was no safety issues. The safety was fine, but it was stopped because of lack of efficacy. And I think it was just very jarring for the community to experience that. And I think it was a lesson that can happen. That committee says, who's looking at the ethics of it, says we can't keep giving a drug to people if there's no evidence it works. Even if it looks safe, it doesn't look like it works. So we have to stop it. And eventually they were able to get, people back on drug who wanted to be on drug, because of course there were some people who did think they saw some efficacy. And eventually this drug was approved in Europe. It was never approved in the US. But I think that was a lesson learned sort of for the Duchenne community, that this is something that can happen in a clinical trial. They don't always, it's not always smooth sailing. And since then we've had, certainly in gene therapy, some patient deaths that really sort of remind you that this is, it's an experiment, and no matter how safe you try to make something, you can't always predict what's going to happen in humans. So I think there have been a couple of situations like that where we've learned it's, it's hard. Sometimes it's harder than people hope. So I'll pass on to Paul.
Yeah, thank you, Sharon. I think that's a really important point to make. And we hear this a lot from those in Duchenne, just the exhaustion of all of the trials that thankfully some have succeeded, but many have not. But the importance of continuing to participate for those that do succeed, and for those that do actually result in an FDA-approved treatment that then everybody can take from there, at least if it's indicated for them, not just those who participated within the trial. Something I think also that those within the Duchenne and the SMA communities, as well as other communities, have found is just the importance of collaborating early and often with FDA for them to actually understand what it's like to live with the disease, because these are clinicians, these are scientists, these are other folks who may have some cursory understanding of the disease, but certainly not from the perspective of someone living with it, and certainly not from the perspective of a caregiver or a family member either. And so they might not understand what is meaningful. The important word here is meaningful, because FDA is always hoping to understand from the patient community's viewpoint what is actually a meaningful change, what is a meaningful, effective treatment for the community, because FDA might not think it's all that important initially. Maybe if an individual is able to walk up the stairs for a month longer, or is able to reach the light switch for a month longer, or to reach up in the cupboard and get a glass. These are all these little activities that, if preserved and actually can be maintained for longer in these communities, it can actually just be huge. But to an individual at FDA who's a reviewer, they might not fully grasp the importance of being able to do those kinds of activities. And anybody watching can name that activity perhaps that they would say is what is meaningful to them to be able to do for a little while longer, maybe even longer than that for that matter. So really putting in perspective to FDA how some of the improvements that these potential therapies could actually be meaningful to them, even in ways FDA before probably didn't fully understand. Consequently, that helps FDA when they're evaluating the data to actually put the kind of patient experience later on top of that data. Sure, they saw the endpoint. There was a measured improvement with the R squared number of so-and-so. I would get out of my depth when I start talking statistics, but they're able to not only see that, they're able to also see, okay, well, this means that individual can, walk down the steps for another month or 2 months or 3 months longer than they would have otherwise, which is a whole different context than they can get just from looking at the data in front of them. So I would say one of the things that those in the DMD and the SMA communities, other communities are learning is how important it is delivering that message to FDA so that the FDA reviewers there can understand that context, not just the data that's going to be placed in front of them.
Thank you, Paul, and Sharon for sharing such a wealth of information and experience with the community. Another question we have for the community is, what are some of your suggestions for the LGMD patient community for how to balance their hope and excitement with realistic expectations about timelines and transparency limitations, and just remaining strong in mind and body during the drug development process?
Yeah, I think that's always tough. You want to be hopeful. And yet, the example I just gave of the drug that just, trial just stopped, those things happen. But I guess those are skirmishes and you're trying to win the war. So we have to keep in mind that it is this series of, there will be this series of, issues and problems and challenges that come up. But inevitably we seem to overcome them, and it doesn't happen as fast as anyone wants, but we get there. So I think it's more of a slog than a sprint. I do think you need to balance hope. These are experiments. We don't know what's going to happen, but, we do get drugs approved, and we've gotten drugs approved in other areas. And I think with enough attention and focus and effort in limb-girdle, we're going to see the same thing happen. It's just going to take a little while, but don't be disheartened by some of these temporary setbacks, because they're going to happen. They absolutely will happen. And sometimes even some really bad things happen, but eventually we overcome them.
Yeah, and I would add to that, Sharon. I think that's very well said. I would add that there's almost always something that you can be doing. I mean, clinical trial spots can be limited at times. Of course, there could be exclusion criteria that only allow a narrow subset of the community to participate, or there's only so many spots available. And then from there might not be expanded access, which is also opportunities to get that investigational therapy outside of the clinical trial. And this is generally within kind of the phase 3. If you remember back to Sharon's phases, that's generally more common within phase 3 of drug development. And as we saw within LGMD, there isn't any therapy that's actually at that phase 3 stage quite yet. So consequently, just actually participating within the clinical research is obviously one of the best ways of getting involved and to really try to accelerate that development. The quicker the trial can enroll, the quicker it can conclude, the quicker we have that data. Hopefully it's positive, the quicker that drug can get on the market. But, in absence of being able to participate within a trial, and there's frustration with how slow things are going, there's all these opportunities I already kind of went over in some ways of trying to accelerate the process from there, whether it be educating FDA's review divisions, whether it be participating in these advisory councils within FDA, whether it just be signing up to be an advocate with MDA or with any of the other organizations here. There's always ways, even if it's not directly actually participating within the trial, that you can be contributing to the overall effort to accelerate this process, to try to get it to the community that much more quickly.
Yeah, that's a great point, Paul, that, you can participate in surveys, even participating in registries, all of those sorts of activities also help de-risk, that de-risking process I talked about, by providing information. So lots of things that you can do.
Thank you for that answer, Sharon and Paul. I guess one other thing, how does the sort of general category of raising the public awareness, public awareness and awareness on the part of regulators and drug developers of these diseases fit into that?
Yeah, I think that's huge. I think that's a big part of what we do. It's important, both for regulators, because regulators, just like Paul explained, they have to really understand what's meaningful. It's not just in addition to all of the biology of the disease. But I think raising the profile of these diseases for drug developers is important. So they often have a technology that's sort of looking for a home. Sometimes you'll have a company that says, we're going to, focus on muscle disease. But a lot of times you'll have a company that has a technology that might be a good fit. So what can we do to make them aware of a good indication? And if it's a limb-girdle indication, to say, have you considered this, subtype of limb-girdle or this subtype? And, we can find all of the people, and, there's a disease organization that's focused on this particular limb-girdle. So the more that we can do, I think, to pull all of those pieces together and package it and make it easy, the better we are. Because, it's not as common as cancer. We don't have companies jumping into this space. I think there are some here, which is fantastic. And the more we can do to make that easier and make information available to companies, it's really helpful. So that's part of the de-risking, again, that I think is useful.
Yeah, I can take a more of a congressional angle, I guess, on this question, because we have seen several communities who have really kind of taken Capitol Hill by storm. And by that, I mean, they have gotten to know their lawmakers. They've gotten to know their lawmakers' staff. They're reaching out to them always, even just to have a quick catch up. A conversation doesn't even have to be, I want you to pass this bill. It could just be, this is my story and you're representing me. That it could be that simple. But what can happen if there's a community that does this kind of concertedly enough, I guess you could say, and we can look at the ALS community as one such community, then all of a sudden you have all of these champions across Capitol Hill, some of whom do have personal connections to ALS, but many who don't. They were just impassioned by the advocates who talked to them about ALS. And all of a sudden there's an ALS caucus and there's efforts to pass bills with ALS in the name. And there's new funding for research being passed by Congress that's specific to ALS. Now, this has obviously been a long time coming for the ALS community. I'm not saying that happens overnight by any means, but, thinking about ways in which raising awareness in LGMD can be impactful. If these relationships can be developed, these friendships can be made up on Capitol Hill, then all of a sudden we might start seeing some targeted funding for LGMD research, or some efforts at FDA that Congress encouraged FDA to do because these lawmakers really wanted to be doing something for the LGMD community. And that's all just about raising awareness and building these relationships on Capitol Hill in particular. So it's another way in which what might seem as a bit nebulous, like raising awareness for the disease, can have some really important real world impacts.
Well, a huge thank you, Sharon and Paul of the MDA, my colleague Brad from the Jane Foundation, and everybody at the Speak Foundation for leading this important summit. I'm sure there's so much that the patients of the LGMD communities will take away from it. So a huge thank you.
Thank you.
Thank you for having us.
Thank you.
At this time, we will present the U.S. updates. First, Dr. Nick Johnson, the founder of GRASP LGMD, will share with our audience clinical trials that are coming up that GRASP LGMD researchers and clinicians are involved with. Then we will hear from Jane Foundation as they share updates with the 2B dysferlin community. We will hear from Doug Albright and Laura Rufibach, who are co-presidents of the Jane Foundation.
Thanks, Catherine, for the introduction. I'm excited to see all of you here, and I hope this is going to be a great event. Really thanks to the Speak for putting this on. I'm here to talk about our GRASP LGMD consortium updates. I'm the PI of that consortium here in Richmond, Virginia. What are the goals of our consortium? Number one is to improve diagnostic testing for individuals with limb-girdle muscular dystrophy, to validate survey and functional measures for limb-girdle muscular dystrophy, support biomarker development. Most importantly, the reason why you're here today too, speeding therapeutic development across the limb girdles. These are consortium sites you can see spread across the United States and Europe. And these are some of our active studies. So we are actively enrolling individuals with calpain-3, ANO5, dysferlin, DNA JB6 in a 12-month study looking at trial endpoints in those conditions, as well as similarly individuals with sarcoglycans sponsored by Sarepta, again for a 12-month study. We have completed enrollment in a biomarker development LGMD2I or FKRP, and that study should be ending here this fall. And then we're just ramping up study for Becker muscular dystrophy in enrolling 150 individuals over a 12-month duration. So mix of studies for any individual. We are actively looking for individuals with those particular mutations. And the reason why we're looking for those individuals is to see whether or not we can validate clinical endpoints like the North Star. Here you can see some scores at the bottom left indicating that changes with ambulation, and that's the kind of information that's important for trial development. If you're interested in participating in a study, save this slide right here. You can email our network manager, Jessica St. Romain, or myself. We will make sure we get you to the right site. And beyond our study enrollment, we're also very interested in improving genetic diagnosis for limb-girdle muscular dystrophy. This is really led by Chris Weil at Washington University in St. Louis in establishing gene-specific guidelines to improve the genetic diagnosis. You can help with this. Save this slide too. As an individual with limb-girdle muscular dystrophy, or you know somebody who is, you can volunteer to support improving the diagnosis. You can email Chris Weil. His email is down there at the bottom. No experience is necessary. Patients, caregivers, advocates, anybody can participate in this. And we definitely need your help in improving the genetic diagnosis. As many of you in the audience may know, sometimes it's uncertain in terms of the particular mutations and whether or not they're actively causing disease. So these are other consortium activities of variant resolution progress, collaborating with Imaging DMD for quantitative MRI, protocol reviews, and of course our ongoing partnership, the reason why I'm here today, with the Speak Foundation. But most importantly, we need your help, we need your support. Please enroll in a clinical study if you are eligible to, and if you're not, or even if you enroll in one of our clinical studies, please consider working with Dr. Weil in terms of variant resolution. And thank you very much for your time today.
Hello, my name is Laura Rufebach, and along with my colleague Doug Albrecht, we are going to give you a research update for dysferlinopathy. Both Doug and I are co-presidents of the Jane Foundation, whose staff are shown here. The Jane Foundation is a nonprofit disease foundation and advocacy organization that focuses solely on dysferlinopathy. The Jane Foundation's main goals are to identify, develop, and evaluate potential therapies for dysferlinopathy and cultivate a robust, clinical trial-ready community. To begin with, I'd first like to address what I mean by the term dysferlinopathy, especially since this meeting is supposed to be focusing on the LGMDs. Dysferlinopathy is a term used to refer to muscular dystrophy that is caused by pathogenic mutations in dysferlin, or the DYSF gene. This encompasses multiple clinical presentations, including LGMD2B, or as it's referred to with the new nomenclature for LGMD, LGMD-R2. There's also Myosin myopathy type 1, as well as others. Analysis of all these different clinical presentations show that they are the same disease. Therefore, we use the term dysferlinopathy to indicate that the information we're presenting is relevant to all individuals with pathogenic mutations in dysferlin, no matter what name they have been given for their clinical presentation. As shown here, the development of any treatment requires the identification of three main things: what to test, how to test, and who to test. All of these elements are required for the successful development of therapeutics. The remainder of this presentation will focus on how the Jane Foundation addresses each of these areas and provide you with updates on where we stand in each area. Over to you, Doug.
Thank you, Laura. Our approach to developing a treatment for a disease as rare as dysferlinopathy starts with mapping what is known about the cause of the disease and then identifying possible intervention points. We use the diagram shown here internally at the JF as a visual tool that helps us identify knowledge gaps that we can then address through research projects. Over time, we have managed to fill in the diagram as we learn more about dysferlinopathy and why dysferlin's absence leads to muscle problems. We then use the diagram to identify points in the disease process where there are opportunities to intervene, which are shown here in gold. At a high level, interventions can be divided into two distinct categories: those that seek to fix the problem of dysferlin deficiency at the genetic level and restore expression of the dysferlin protein, and those that might slow disease progression by affecting one or more of the consequences of dysferlin's absence. As you can see here, the scope of our research programs is very broad. We're typically running around 20 active projects at any one time that are addressing the most promising routes to therapy and which reflect the latest results and advancements in the field. The complete list of the active projects we have going, as well as past projects and a summary of their findings, can be found on our website. We can't possibly cover all the projects and their individual progress in the short time we have today, so instead we'll give one example of a genetic therapy and one example of a disease-modifying therapy that we are excited about. For the genetic therapy, while the concept of restoring the expression of dysferlin is simple, the practical application of gene therapy has many challenges. The most advanced and successful gene therapies today are using the very small AAV virus to deliver the required genetic construct to the target tissue. The dysferlin gene is too large to fit in an AAV virus, which has left us with a problem to solve. The Jane Foundation worked for many years with multiple research labs to develop the dual vector AAV system that is shown here on the left. It is able to deliver both halves of the dysferlin gene to the nucleus of muscle cells, where those two pieces of DNA find each other and create a single full-length copy of the dysferlin gene that the muscle cell can use to make dysferlin protein. This dual AAV gene therapy has successfully met all the safety and efficacy criteria required by the FDA to move into human testing and is licensed by Sarepta Therapeutics, who are continuing to develop it into a treatment. Sarepta recently announced that the first intravenous dosing of a small number of dysferlinopathy patients in a proof of concept trial is anticipated to begin late in 2022. While we have high hopes for the Sarepta program, the JF is continuing to explore new technologies that may make dual vector gene therapy more efficient and thus safer and more effective. One example, shown here on the right, uses special protein sequences called inteins. Inteins can combine two pieces of a protein into a single full-length protein. It's not clear yet if an intein system, which works at the protein level, will be more efficient than a system that works at the DNA level, but we feel it's important to continue exploring new technologies so that we can bring the best therapies to dysferlin patients. An example of a disease-modifying program that we're excited about is treatment with a protein called galectin-1. Galectin-1 is normally made by muscle and works on both the inside and the outside of muscle cells to achieve a variety of tasks. Galectin-1 can improve membrane repair, promote muscle growth and regeneration, and suppress some of the detrimental effects of inflammation, all of which may help slow the disease process that happens when dysferlin is absent. Recent publications from the Van Rij lab that were supported by the Jane Foundation show that injecting dysferlin-deficient mice with galectin-1 can produce some encouraging short-term benefits. The next step is to treat mice with galectin-1 for longer periods of time to better assess and demonstrate the benefits of galectin-1 treatment. If those experiments continue to go well, we hope to find an industry partner to take on the program and do the formal toxicity and dosing studies that are necessary to gain permission from the FDA to conduct tests in people. We expect that work to take several years, at which point we hope to be in a position to begin clinical trials in patients. I hope you're excited about the progress we are making in these research projects, and we look forward to telling you more about them in the future. Now Laura will continue with the important steps the Jane Foundation is taking to prepare for testing potential therapies in clinical trials.
Thanks, Doug. The next area to cover is how to test. In all clinical trials, there is a test or assessment that is referred to as the primary outcome measure, which the FDA uses to determine if a clinical trial was successful. Therefore, the choice of the right primary outcome measure is critical to the success of any clinical trial. To determine the best outcome measure, you need to understand the disease process and evaluate multiple potential outcomes to find those most sensitive to change, preferably in 1 year or less, as that is the typical length of most clinical trials. To identify the best outcome measures for dysferlinopathy, the Jane Foundation embarked in 2012 on a large natural history study with over 200 participants, which we call the Clinical Outcome Study for Dysferlinopathy, or COST for short. As you can see from the diagram, we are learning a lot from COST. We have identified assessments that we believe could be used as primary outcome measures, including one designed specifically for dysferlinopathy called NSAD. We are also gaining a deeper understanding of disease progression, identifying information that will help your clinician better care for you, and enhancing our overall clinical trial readiness. The last area to cover is who to test. One of the greatest bottlenecks that slows down clinical trials is the difficulty in finding individuals to participate in the trial. This is especially true for rare diseases like dysferlinopathy that have so few individuals. Our goal is to remove the bottleneck by identifying a large group of dysferlinopathy individuals that can be used as a recruiting resource for companies. We do this through a patient registry that we call the Dysferlin Registry. To date, the Dysferlin Registry has almost 1,000 members who are genetically confirmed to have dysferlinopathy from over 50 countries worldwide. In addition to being a recruiting resource, the Dysferlin Registry is a tool that helps with diagnosis, provides helpful information about dysferlinopathy, and is a place to interact with the Jane Foundation and others with the same disease to find friendship and support. Doug and I hope that this presentation has given you a good idea of what it takes to develop therapies for dysferlinopathy and where we currently stand in the process. If you would like to find out more information about dysferlinopathy, the Jane Foundation, how to join the Dysferlin Registry, or if you have other questions, please visit the Jane Foundation website or contact us using the information provided here. In addition, in the summer 2022 LGMD News magazine published by the Speak Foundation, we will have an article that will go into more detail on these topics. Thank you and have a great day.
Welcome to Roundtable 2 at the Global Advocacy Summit. We have assembled a group of individuals or caregivers who have experiences with living with limb-girdle muscular dystrophy. So we're going to take a moment and just go around to our panelists and let them introduce themselves and share where they live and what subtype of limb-girdle they're living with. But first, I want to introduce our co-moderator, Sarah Foy. Sarah, will you introduce yourself?
Sure, thank you. I'm Sarah, and I'm from New Jersey, USA, and I'm the founder of Team Titan that helps support families and individuals affected by limb-girdle muscular dystrophy type 2J or R10.
Okay, thanks, Sarah. And Sarah and I will be moderating today's panel. And so we're going to go around and we're going to start with Kelly Brazo. Introduce yourself and tell us a little bit more about you.
Hi, my name is Kelly Brazo. I'm from Lancaster, Pennsylvania, USA. I have a daughter who's 14 years old who's diagnosed with LGMD type 2I R9. And I'm also the founder of the Cure LGMD 2I Foundation.
Thank you, Kelly. We're going to move down to John Favor. John, introduce yourself.
Hi there. My name's John Favor. I live in Cincinnati, Ohio, USA. And I live with limb-girdle muscular dystrophy type 2J.
Thanks, John. And then we're going to go to Rachel DeConti. And Rachel, share your story with us.
Hi, my name is Rachel DeConti. I am from Newington, Connecticut. I have a son who is 5 years old. He was diagnosed last fall with limb-girdle muscular dystrophy type 2D R3.
Thank you, Rachel. And we're going to just go around and do a round robin for this first question. And the first question we're going to ask is, what do you feel are the biggest challenges as a patient or caregiver in understanding the rare disease drug development process? And we're going to start first with John.
Thank you. For me, the biggest challenge is understanding where a drug or treatment for a particular LGMD subtype is in a particular stage in development. You know, what those milestones are and what it means for other potential drug development successes that may be relevant to other subtypes when the milestones for one drug therapy are met. So in other words, it's hard to know when a success for one subtype is a success or even partial success for others. Since science can build upon itself so much in this field, I can only assume that a single success for one particular type of LGMD will at least partially help move towards successes for other subtypes. But getting a grasp on exactly what successes are being made and the potential impact they may have across the entire spectrum of people with all forms of LGMD is a challenge, but it's very critical.
Oh, John, that's a very good answer. And it's hard to know that, isn't it? Because you'll see, you know, clinical trials for one subtype, and then you wonder, okay, does that mean mine's next? Or when will mine come up? And if this one's successful, will it help further mine? And that's a very, very, very good answer. Kelly, why don't you take a stab at that question as well?
Sure. I think some of the things that most people are concerned about are just understanding the complicated process and this seemingly lengthy timeline that clinical trials seem to take. Where do we look to find appropriate resources to learn about the different treatments that may be in development for your particular diagnosis? And also finding out the recruiting process. How do we get enrolled in studies? What are the parameters in order to be able to qualify for a given trial, whether it's your age or your physical abilities, and just knowing all the differences out there and where to look for that.
A good answer. Rachel, why don't you take a chance on this question too, and we'll hear your responses.
Yeah. I have to echo what both John and Kelly said. I think that it's understanding, especially, you know, being a parent with a small child with LGMD, what's available to us, what the process looks like. If we are at a preclinical stage for LGMD 2D, what does that mean? How long will we be in preclinical for? And when can we expect a trial to be available? I think in these types of instances, the more communication, the better for the community and understanding what's available to us and when.
Yeah, Rachel, good response there. And Kelly and John as well. Rachel, don't you feel too that it's difficult for new families to understand where to go to find that information? I find that that's the most challenging for new people in the limb-girdle community. What do you think?
I absolutely agree. I mean, again, we just found out last fall, and the feeling that you have when learning this diagnosis is so overwhelming. You're trying to wrap your head around the fact that this is now your life and understanding what's available to you. And of course, as a parent, your first thought is, okay, so how are we going to cure this? What are we going to do? What's available to help my son, my baby, right? So yeah, understanding where to go, especially for your specific subtype or where that is in certain companies' pipelines, I think is so, so important. And there isn't really one spot to find that. I think a lot of companies do a good job, but really diving through the details is critical and helping parents and patients ease mind on knowing that their future is, being considered by all of these companies.
Yeah, good response there.
Well, you guys, thank you for all your perspectives. And our next question is going to be, what do you believe are the ways advocacy and patient participation play a role in seeing treatments develop for rare diseases like LGMDs? And I'll turn this one over to Kelly.
Well, as a parent of a child with LGMD2I and also the director of a patient advocacy organization, I try to personally help bridge the gap between industry and the patient community. There are so many patients that are really confused by the different treatments that are in development. And I think it's important for the advocacy leaders to help patient communities keep them informed by disseminating information that they might have, have access to from the biotech industry. And at the same time, it's super important for patients to share their concerns, their fears, their hopes with the advocacy leader so they can then share that information back to the industry partners. As many more biotechs seem to be interested in this rare disease space, which obviously we're super excited about, they really do want to know what is on the top of priority lists for our patient community, because obviously they want to develop clinical trials that are successful, that are efficient, and they really want to make sure that they're serving the needs of our patients in order to bring these treatments to market, which we're all super hopeful for.
Yes. So I'll turn it back over to Catherine.
Thanks, Sarah and Kelly. Our next question is, can you describe some examples of post-treatment outcomes or effects that would justify participating in an interventional clinical trial, specifically things you might deal with every day, and nothing is too small? And we're going to go first to John Faber.
Thanks, Catherine. The post-treatment outcomes that would justify my participation are basically endless. Participation would be justifiable if I could expect to start returning to walking my dogs or showering without help and without having to sit down. Simple things like steps that are impossible barriers would become, easy obstacles. And I would no longer be expecting to be heavily relying on a wheelchair within the next couple of years of my life personally. Being able to dance with my wife, running through a field, riding a dirt bike again. Everybody here who has, one of these limb-girdle diseases has lost the ability to do something that they love and feel very deeply connected to, whether it be a want or just a small need in our daily life. And so if any of us could get even one of those things back, I think that would definitely justify our participation.
Rachel, what are your thoughts on this question? Let me ask you.
I think that there are many examples for my son that would help us, make this decision, and that post-treatment outcomes would be greatly helpful. We found out, as I mentioned a couple of times already, that my son has limb-girdle through a case of rhabdomyolysis that he suffered last summer. And through, tests, of course, ultimately we determined that it was the limb-girdle that he was suffering from. So this was a complete shock to us. And since that one case, my son still has myoglobin in his urine and has to drink at least 48 ounces of water today, every day, for him to, keep those myoglobin levels down. So this is constantly on our mind. He has a huge water bottle that he has to bring to school. He's an active 5-year-old boy. So, making sure that he's drinking water at all times is so critical to us. We're constantly asking him, what his urine color is. And as a 5-year-old little boy, he doesn't really know why we're asking these questions. So having him be able to not worry as much, or having us not worry as much about how much water intake would be a critical outcome for us. He's also restricted from playing team sports right now. He can't play sports because of, the physical endurance that it would have on his body. So, hearing and seeing other kids being able to play sports and him not knowing why he can't, fully understanding why he can't, is heartbreaking as a mom. And he should be able to play sports. He has the right, and this is such an important time in his life, and we're trying to find other ways to keep him active and make friends. And it's something that we think about every single day. And we want to be able to see him hopefully someday be able to participate in these sports that he would love to play in. And then also, for a parent, him going to school. A lot of times you always worry about your kids going to school, but this is a different worry for me because I have to worry about what's going on at recess, what's going on at gym. He comes home sometimes and says he has to take a break because his legs hurt. And, it's heartbreaking. It's heartbreaking to hear, and you don't want to have to have those constant worries. And if he does get hurt, God forbid, in one of these types of situations, it could be a lot different for him than it would be for other students or other children. So these are the things that would make, exploring a clinical trial so beneficial, and ultimately having an active and healthy life as a child and grow to be a healthy, active adult would be one of the best outcomes we could ever ask for.
Thank you, Rachel.
So thank you for sharing about what outcomes would be important to you. We're going to continue to expand on that to think about what factors might cause you to want to participate in a clinical study or clinical trial. And Rachel, why don't you go ahead and answer that one?
Yeah, thank you. So like many within our LGMD 2D community and broader, of course, we want a trial to participate in today. And as a parent, of course, you'll do anything for your child. So some of the factors that I just pointed out would be one of the main reasons why we'd want to participate in a clinical trial. But also my son at 5 years old has very mild symptoms right now for the most part. So getting into a promising trial and treatment would be drastically, could change his life drastically. And even cure him, giving him his body the protein that it's missing. So a treatment would really change our life. It could change his life, and it's time being of the essence. It's critical to get into treatments today.
Thank you.
Thank you so much. I've got a question. It's very similar to that one, but what factors might cause you not to want to participate in a clinical trial or even a natural history study? Let's go to John Favor on this question, and we'll also ask Kelly Brazo this question as well. So John, we're going to go to you first.
Okay, I want to preface this by saying I would join a 2J trial immediately if there was one open today, without any reservations. But personally, I do have some worries or possible scenarios that I am concerned about. One being, if I were to join one of these studies and be put in a placebo group, and then later, even after the study, not receive the treatment as part of the study, that would be very disheartening, but I'd still be happy to contribute to the science. Also, if I were to receive a treatment that would be sort of like a partial or half measure that would somehow disqualify me for future treatments, that would be very discouraging and something I would want to get clarified before I joined. But, largely I would, immediately jump at any opportunity for a trial. Yeah.
Thank you. I just wanted to touch base on a little different question, which is, do you think companies have an ethical responsibility to communicate timeframes to patients? And I'm going to ask Kelly that question.
Yes, absolutely. I really do feel like patients should be informed so that they can make an educated decision for themselves and or their family member that they might be caring for. Patients are constantly trying to educate themselves to make difficult decisions regarding their ability and willingness to participate in a trial. The past few years have obviously created additional challenges for the biotech industries due to the pandemic. The timelines have constantly been changing. We have heard things were to have started a year and a half ago with enrolling for trials. And just as the patient community is frustrated with the delays that have happened, I know the biotech industry is also equally frustrated. That we've all had to evolve over the past 2 years of making changes. But at the same time, we are all fighting for a similar progressive illness. And it seems like at times that our situation is progressing more rapidly than the drug development process is. And it can be really discouraging and overwhelming and exhausting, just waiting for the news to say, it's your turn, your time has come, come into this trial, we're finally ready for you. And so patients are just craving updates for this information, even if it's saying we have to wait 3 more months or we have to wait longer, they just want to know. And I think a lot of times if you go onto an industry website, it says it's in the preclinical stage. What does that mean? How much longer for preclinical? How much longer for that you can just put your name in the hat? Please pick me. And I think that's just the communication. I think John, I don't know if it was John or Rachel, either one of you had said that there can never be too many updates. Just inundate us with information. We will never be upset by that. And whether it's advocacy leaders, we're happy to share that information. But to share that timeline is just answering questions that we're all waiting for. So
the need for good, timely, and repetitive or repeated communication sounds like a key point from a lot of the speakers on a lot of the questions today. I'm going to turn the same question about communicating timeframes to patients over to John.
Thanks. I believe a company's transparency is directly linked to its ethics in this case. Transparency and communication are of the utmost importance in drug development industry from a patient perspective. And I think we all understand that there are risks to full transparency for a private company making proprietary therapies. But as a community, I think that's largely understood. However, making your successes and failures as a company known keeps our community educated. It keeps us hopeful for future successes. It makes us eager to participate and provide invaluable feedback directly to the industry that is trying to serve us. And it also allows for those suffering from these terrible conditions to generate a hypothetical timeline or potential roadmap in their mind that they can follow as treatments get closer and closer to full development. I just want more communication, like everybody else is saying.
Thank you.
Thank you, guys. And then our last question is, if you could share just one thing with the pharmaceutical industry or even the FDA, what would that one thing be? And let's start here with Rachel DeConti.
Thank you, Catherine. I think it's a great question and one I feel very fortunate to be asked. I have one thing. I'm going to kind of make it into 2 parts, but it is one statement, which is LGMD is just as important and should be focused on just as much as other muscular dystrophies and other rare diseases. And with that, time is of the essence. For us as caregivers, as patients, every minute, every day, every week counts. That really can make the biggest difference in somebody's life and being in a trial. A patient who needs the patient who needs the treatment today, like my son. So just keep in mind that we're here. We thank you for focusing on this. For those who are in the summit today, for the companies who will participate in the future, we thank you. And we're here to help quickly get these treatments for our children and for patients in need.
Thank you. Thank you, Rachel. Kelly, why don't you try and answer this question as well? I know you have a lot of passion about this issue too.
Well, Rachel, I would say everything you just said if I could, but I'm going to say something different. But I firmly feel that what everything Rachel said is just so pertinent to our situation right now. But I also want to ask the biotech industry, as they are considering, whether it's natural history studies or clinical trial development, please consider that, most of our patients in our rare disease community are more than willing and beyond excited to participate in a clinical trial. I think we've all expressed that today. But on that same note, patients are, really giving up and sacrificing a lot to be able to be in these trials, not knowing if it's going to be successful, not knowing if they're actually getting the treatment versus the placebo, like John said. And with that, they are missing days of work. Children are missing many days of schools and other activities that they would otherwise be trying to participate in at home. Many people need caregivers to come along with them, which is an additional expense and challenge for coordinating all of that. And certainly the pain and discomfort of being in the clinic all day, the exhaustion that comes with, numerous tests, blood draws, muscle biopsies, whatever the case may be. And, especially children, it's scary. I in particular had a daughter who was had severe anxiety with any sort of needle related treatment. And she's learned to deal with it over the years. But just knowing if there's any way that we can, make patients more comfortable, maybe limiting the number of trips to a clinic that is, hotel rooms are certainly less than accessible, travel challenges, whether it's by air or train or car, that if we can make the patients who are sacrificing their time to be in these trials, to make it more accessible and less, taxing for them, we would appreciate that. But we're very excited about all of the, momentum that's happening. And as I said at the beginning, everybody that I talk to wants this to happen and they're willing to do what it takes. But I think it is just a working relationship if we could, know that these patients are not just a number. They are humans with lives and pain thresholds that want to get home, as soon as they can.
Thank you, Kelly. And I just wanted to say thank you to all of our participants today and our co-moderator, Sarah Foy. The work you guys are doing by sharing your thoughts today really helps give a voice to our limb-girdle community. And it also helps our industry partners just hear directly from you and what you really are feeling. So thank you all for being a part of the Global Advocacy Summit.
I want to share with you a little more about the Believe There Is Hope for a Cure campaign. In 2022 and in 2023, we will focus at the Speak Foundation on raising funds to help further advocacy and research for all forms of limb-girdle muscular dystrophy. Currently, there is a large disparity in research where potential treatments are just not applicable to all forms. With over 30 genetic forms of limb-girdle muscular dystrophy, we want to look at research projects that look at innovative ways to deliver gene therapy to all. We also want to fund research projects where new types of medication could be useful across the board. You can get involved with this fundraising campaign by signing up at believethereishopeforacure.com. You can raise money your own way and help contribute to these research projects that will benefit all forms. Thank you so much.
Sarepta Therapeutics is on an urgent mission to engineer precision genetic medicines for rare diseases that devastate lives and cut futures short. As a part of this mission, Sarepta's current gene therapy pipeline includes investigational compounds to address 6 LGMD subtypes. In the following presentation, Sarepta's Head of Research and Development and Chief Scientific Officer, Dr. Louise Rodino-Claypack, will provide an LGMD community update. Thank you, Sarepta, for the work that you're doing and for participating in this special event.
Thank you so much, and thank you to the organizers for inviting me to speak to you today. I've been working on limb-girdle muscular dystrophy as a researcher for almost 2 decades, and this is near and dear to my heart, and I'm very pleased to be sharing this information with you today. Just want to highlight that during the presentation, I'll be making a number of forward-looking statements about things that may happen in the future. There's some risks and uncertainties to this, many of which can be beyond our control. You can feel free to view this complete list on our website at your leisure. At Sarepta, we have an urgent mission to bring advanced science in genetic medicine. And so this is to help individuals with rare diseases like limb-girdle muscular dystrophy. And we understand the urgency around this. We know that every 24 hours is important. And so we're constantly trying to drag tomorrow into today, which means that we're trying to move as fast as we can to bring these therapies forward. For the presentation today, just want to go over a brief agenda. First, I'll be starting out with a call to action. You in the limb-girdle community have asked us how you can specifically help, and so I'll be sharing some information on that. We'll talk about our LGMD pipeline and some specific updates around a natural history study and 2 of our programs. Also be highlighting a little bit about manufacturing, which is important to gene therapy and the process of moving forward, and then summarizing at the end. So as a community, how can you help? And the answer is in a number of different ways. We are an urgent mission, but we need to do this in partnership with all of you. And so there are several different ways, and we'll talk through those in the next few slides. So one way is to connect with a neurologist. This is important to identify a treating physician that specializes in LGMD research and clinical care. And along the same lines, getting genetic testing to learn your LGMD subtype. For many years, individuals were just classified with LGMD, but that's importantly has been changing over the last 10 to 20 years. It's important to know your subtype so that you can get the appropriate information care. And this information may be required for future clinical trials like the gene therapy trials that I'll be speaking to you about today. You can also participate in natural history studies. I'll be talking about one today, which will provide us and the community and yourselves with critical information about how to speed research and development for these diseases. You can engage with patient advocacy organizations. This is important to access community news, education, regulatory engagement opportunities, or just be aligned with individuals yourself, have a community so that you can both share information, learn information, and help others as well. Finally, you can join a patient registry. These are essential with our ability to develop a foundational understanding of the number of people around the globe that are living with limb-girdle, and then helps us provide information in terms of clinical trials or other potential organizations. You can always contact your physician or your patient advocacy organizations. We have some information on this, and at the end of the presentation, I'll also share where you can find more information from us. So one other way that we've been able to allow you to potentially help, and we're announcing a limb-girdle muscular dystrophy grant award program. And so the goal of this program is to shorten the limb-girdle diagnostic journey. And for this, we're asking for your help and your insight. We're looking for applications that support original, meaningful efforts through one of the following options or multiple of these options. So one of the applications could be promoting recognition of early signs and symptoms of limb-girdle muscular dystrophy. Second, enhancing participation in genetic testing and genetic counseling programs. And finally, empowering families to take an active role in pursuing a confirmed diagnosis. And so how can you apply? We're seeking these project applications, as I mentioned, to support the diagnostic journey. Patient advocacy organizations can apply, non-governmental organizations. Individuals can also apply, but we do recommend that you partner with a patient advocacy organization or another non-government organization to do that. In total, we will award up to $100,000 to the selected projects. This could be one project, or this could be multiple projects that equal the dollar amount. The application, you can apply by September 5th, 2022, and you can learn more on our website, which is sarepta.com/LGMD Grant Award Program. And I'll be providing our websites at the end of the presentation as well. And so that's one way in which you can help in your community, but there are others as well. What I wanted to next highlight is our gene therapy pipeline for limb-girdle muscular dystrophy. We have a large pipeline of limb-girdle programs. It's the largest in the industry. To support this, we have to have manufacturing expertise and capacity. I'm going to talk a lot more about that, but with genetic medicines, especially gene therapy, this is one of the biggest hurdles that we have to overcome, but it's an important hurdle to bring these therapies forward. And finally, our third pillar is expertise and tools to advance the science. So this is how do we optimize our gene therapy programs, and then how do we make gene therapy deliver most effective. In our LGMD pipeline, we have 6 programs that are in preclinical or clinical stages. Just to highlight the various stages of development, First, we start with research and proof of mechanism. This just simply means, can we confirm that the drug itself is working? Is the gene therapy that we're delivering able to produce the effect? So is it able to produce the protein that's missing? Preclinical studies refer to animal studies, and these can be small and large animals. And here we test drug safety as well as drug efficacy or drug effect. Now, the next two stages are clinical stages, meaning in patients. So we have early stage clinical trials. These could be proof of concept trials. We're going to talk more about that, where you can characterize safety and preliminary efficacy. And these are usually in small populations of patients. Later stage trials tend to be larger populations of patients, where you take the information like dose and safety from the early stage trial and prove it in a larger setting. And then commercial is once the product is approved and on the market. Often what may be unknown to people is that there's also studies that continue after a drug is commercialized to continue to study it over a longer period of time. We have 3 programs that the sarcoglycanopathies that are in clinical stages. The dysferlin, LGMD2B, is also in clinical stage, and LGMD2L or Noctamin 5 deficiency and LGMD2A or calpain 3 deficiency are in preclinical stages. In addition to this pipeline, we also have a large gene therapy pipeline. We also learn from our other programs like Duchenne muscular dystrophy, and we are also investigating many ways to look at treating patients in the entire population. So there are also patients with pre-existing antibodies to the AAV vector that we use, or patients that have been previously treated with gene therapy. And so we're investigating ways to make sure that we can fully be able to treat the population, including those with antibodies. And that's a huge effort that's underway at Sarepta. So I've alluded to this already, but I just wanted to highlight the process of gene therapy. So what exactly does that mean? Each LGMD is caused by a lack of an important protein. So one particular protein is missing in each limb-girdle subtype. It's an umbrella term for genetic disorders, but then each of those has a subtype. So this change or particular mutation makes the cell not able to produce that protein. It produces either a lack of that protein or a protein that doesn't work as well. And so what we're doing is delivering a new version of that gene to enable to make that protein. And in all the cases of limb-girdle muscular dystrophy, we are delivering the full-length protein. So this is not truncated in any way. This is the full-length version of the protein back to the body. And as you can see here on the right, each of our limb-girdles that we're working on, the subtypes are listed, are caused by a mutation in a different gene: beta-sarcoglycan, alpha-sarcoglycan, gamma-sarcoglycan, dysferlin, Noctamin 5, and calpain 3. Our approach to limb-girdle muscular dystrophy is a gene transfer therapy approach. So this is just another way of saying it, but I think it's important to reemphasize that we're delivering a new version of the gene. We're not doing anything to modify the gene that's within your body. We're delivering a new version using AAV to your body, and that includes the vector, in our case RH74, and then the transgene, which is the gene that encodes for the missing protein. In our pipeline, we have 6 programs. I wanted to highlight that there are 3 components to each of our limb-girdle programs. The first is the vector, which is AAV RH74. This is just a version or a serotype of AAV. We've looked at many over the years, and RH74 in particular is good at getting to muscle, skeletal muscle, cardiac muscle, the heart muscle, and then also the diaphragm, which helps with breathing. So it was important to us to find one that went to all of these places efficiently so that we can deliver the appropriate gene. We use 2 what are called muscle-specific promoters. One is called MHCK7, which expresses in both the heart and the diaphragm. And then also TMCK, which also expresses in the skeletal muscle and the heart, but to a lesser extent in the heart. And so for certain versions of limb-girdle that don't have significant cardiac disease, this is where we use that promoter. We have 6 unique transgenes. So for each program, we have a specific gene, depending on the mutation for that particular LGMD subtype. And as I mentioned, it's the full-length gene that we're delivering. Just wanted to highlight the current status of our programs. We updated you in the fall, but just wanted to highlight you on some new areas for the programs. First, I'll start out with the sarcoglycans. That's LGMD 2E, 2D, and 2C. Importantly, we are advancing our natural history study called Journey. And I'll be talking a little bit more about this in a few slides, but we've introduced new global sites and really excited about bringing this program forward. As I mentioned, natural history studies are extremely important for understanding the natural history of the disease, and then that will help us determine efficacy. In the other programs, LGMD 2L and 2A, we are advancing our preclinical safety and toxicology studies. These are those preclinical studies I talked about, which are necessary for evaluating safety. In all of these programs, we are advancing our manufacturing, which is a critical component and has to be done to advance our studies, and it's important for the rigor with which we need to conduct our studies. In LGMD 2B, we are planning to initiate a proof of concept study, one of those small early phase studies in late 2022, and I'll talk about that in a few slides. And then mentioned for 2L and 2A, we are working on preclinical studies. So I'd like to talk a little bit more about Journey, which is our natural history study. This is in LGMD 2E, 2D, and 2C. There's no drug administered in this study. This is just a study to better understand the disease, and then it informs our clinical trials and also helps the regulators make their decisions. So why participate in this? You're supporting research and development overall, but you will be followed by a clinical team focused on LGMD, which will also support you in your journey with limb-girdle. This will also enable you to be assessed for eligibility for potential future gene therapy trials. This isn't a guarantee to be included in an interventional trial, but it does help in that process. Individuals eligible to participate are males or females, 4 or older with a confirmed genetic diagnosis of 2E, 2D, or 2C, and you have to be willing to travel to an active clinical site. There are some associated expenses can be reimbursed as part of this study. So to learn more, you can go to clinicaltrials.gov and look up the Journey study, or also this clinicaltrials.gov number, or you can also email us at sareptaally@sarepta.com. I just wanted to highlight that we've expanded the number of sites. As of April 14th, this is the current list, but we continue to add sites. We have sites in Canada, Germany, Italy, Turkey, United Kingdom, as well as several sites, 7 now in the United States, and located at all regions of the United States. So please reach out and look at our website as well as clinicaltrials.gov, and you can reach out to these sites directly for any interest. Next, I'd like to highlight our clinical programs in our LGMD pipeline. First, I'll start with our SRP 9003-101 study in LGMD 2E and talk about our newest data. To remind you, SRP 9003 is an open label study in 6 participants. We had 2 cohorts. Cohort 1 received a dose of 1.85 times 10 to the 13th vector copies per kilogram, and cohort 2 received 4 times that amount, and this is our high dose cohort. For this study, patients received a baseline biopsy, a biopsy at day 60, and a biopsy at year 2, and then were followed for functional outcome measures throughout the study. So I'd just like to highlight some of the questions that we consider when evaluating our gene therapies in our trials. So first and foremost is always safety. What's the safety and tolerability of the product? And then the next 3 apply to our biopsies. Is there enough transgene inside the muscle cells, how much is there? And so that's measured by vector genome copies. Next, we asked, is the protein being made and how much is there? And so this is measured by Western blots, tell us the total amount of protein. And then next is the protein at the cell membrane. So the cases of the sarcoglycanopathies, are these proteins at the muscle cell membrane and how much is there? And that's detected by immunofluorescence. And then finally, is the muscle function improved? And this is by various functional outcome measures. So our most recent data update for this study was in March 2022 at the MDA conference, where it presented data. Our newest data is now 3-year data for cohort 1 and 2-year data for cohort 2, or the high dose. And so we presented biopsy data for cohort 2 at 2 years, which was new, as well as the 3-year data for cohort 1. So just to highlight in terms of safety, we had no new safety findings, no unexpected immune responses, and we now have 3 years of follow-up data for cohort 1 and 2 for cohort 2. Just to highlight a few of the new findings in terms of gene expression in the muscle cell biopsies, we now have data from biopsies at both day 60 and at 2 years for both cohort 1 and cohort 2, and importantly show that we have sustained expression, sustained amount of protein there at both day 60 as well as year 2. So that's an important finding in terms of the durability of the response. Now in terms of function, we have similar findings. So now we have been monitoring function over time. And what we found at year 3 in cohort 1, as well as year 2 in cohort 2, is that we have sustained functional improvement or stabilization over the course of the study. So to summarize the most recent findings, the interim analysis, the data that I just described, reinforces our acceptable safety profile with SRP-9003. We showed efficient transduction. That means vector genome copies, and this drove dose-dependent expression at day 60 as well as at year 2. We demonstrated that SRP-9003 demonstrated persistent stabilization in function measured by the NSAD for up to 3 years in cohort 1 and 2 years in cohort 2. We saw similar results for the timed function tests. We also did an exploratory post-hoc analysis looking at comparison of our treated patients to natural history cohort and saw a clinically important improvement based on in comparison to natural history. So far, this observed durable treatment effect provides proof of concept and supports further clinical study of SRP-9003. So now turning to SRP-6004, this is limb-girdle muscular dystrophy type 2B or dysferlinopathy. We've developed an investigational dual vector system called SRP-6004, which is intended to produce a full-length dysferlin. Dysferlin is unique because it doesn't package into the AAV vector on its own because it's too large, but we still wanted to provide the full-length gene, and so we created a system where we delivered the gene in 2 different pieces, and then once delivered to the body using this approach, those 2 pieces recombine and generate the full-length protein. And so this is what we've called a dual vector approach, but it does reconstitute or generate a full-length protein in the end. So as far as our upcoming study for SRP-6004, this will be a proof of concept study to determine if the dual vector approach is efficient by intravenous infusion, meaning can we deliver the gene to all muscles of the body through one single infusion. We've previously completed a first-in-human trial with 6004 and 2 participants by intramuscular injection, and so now we will be transitioning to intravenous infusion. The goal of this study will be to treat a small number of individuals, assess safety, assess the biological activity of the dual vector administration. Many have been asking for details about the 6004 trial. We're currently working with several LGMD stakeholders to design this trial and finalize it, and we'll look forward to updating the community as soon as we have those timelines and plans are further finalized. So next I'd like to talk about manufacturing. As I mentioned previously, manufacturing is a critical component of gene therapy and bringing these therapies forward. Dr. Peter Marks from the CBER division of FDA has mentioned that every aspect of gene therapy is critical, but product manufacturing is the most crucial step to be addressed. And this is an important hurdle that we need to overcome for gene therapy to realize the potential benefit. Just like to highlight how much supply is actually needed of gene therapy vectors for limb-girdle muscular dystrophy. We often talk about doses and amounts, but I think this puts it in nice perspective. So just to think about one of the approved drugs, Luxturna, on the market is for the eye. And so when we think about dose for an individual patient, you can see there 150 billion vector genomes per eye. When we think about in comparison to systemic dosing, like spinal muscular atrophy or limb-girdle muscular dystrophy, we either have 9,000 times this amount or 19,000 times this amount for limb-girdle muscular dystrophy. So these are trillions of particles. You can see 12 zeros for LGMD. And then when you think about that the patients are larger than they are for SMA, for example, we need quite a bit of material to deliver to each patient. So that's been one of the hurdles, but we're making great headway. I'd also just like to highlight that in terms of manufacturing, making the material is just one step in the process. There are also many components that go into the fact of getting the material to you as participants in trials. So this includes analytical development, quality assurance of the product, formulation of the final product, and then getting the product to the pharmacy at the institutions, and then ensuring stability of that product over time. And so these are all important components of this process and something that we take very seriously, and the regulatory bodies do as well. And so this all takes time to develop, but we're doing it as quickly as possible and learning from each program to the next. We're also investing in next generation technologies as well. So just to summarize our pipeline, as I mentioned, there's learnings from each of these programs applied across our pipeline. We have 6 limb-girdle programs that are all using the same AAV RH74 subtype. We also have other programs in our pipeline that utilize RH74, all adding to the safety and efficacy data that we have on hand to inform the development of each of these programs. And this helps us to enhance speed and efficiency for all of these programs. As I mentioned, there are a number of ways to get in touch with us. You can email us at advocacy.sarepta.com. You can also visit us at our website of limb-girdle.com, and this gives us community resources and news on research. We have a GT FAQ series, and this answers your questions specifically about the science of gene therapy, and I think you'll find those useful for addressing general questions about what gene therapy is and how it works. And then finally, you can follow us on social media. We have a number of live events, and as well as community updates on our various social media channels. And so with that, I'd like to end and thank you for listening today. Thank you to our patient advocacy partners, our participants in clinical trials, and the conference organizers for allowing us to speak to you today. And we look forward to hearing from you in the future. Thank you very much.
I'd like to now introduce our next speaker, Dr. Sophie Olivier, the Chief Medical Officer of Atamayo Therapeutics. Atamayo was created in 2020 as a spinoff of Genethon, a French nonprofit pioneer in gene therapy. The current pipeline of Atamayo includes several gene therapy programs for the treatment of the most prevalent LGMDs that should enter in clinical stages over the next 2 to 3 years.
Thanks, Catherine, for the introduction, and thanks for inviting me, giving me the opportunity to present the clinical development program of our first gene therapy candidate for the treatment of fucking-related protein limb-girdle muscular dystrophy, also known as LGMD-R9 or LGMD2I. So here is my disclosure and a few words about Atamayo Therapeutics, which was created in 2020 as a spin-off of French Genethon, a pioneer in gene therapy. Atamayo's mission is to bring to the market new generation gene therapies to patients suffering from neuromuscular diseases, starting with the most prevalent LGMDs. And here on the right, you have our first 5 programs, all with proof of concept, positive proof of concept results, and they should enter into the clinic within the next 12 to 18 months. So I guess you are all familiar with LGMDs, only to remind that there are more than 30 types of LGMDs that have been described in the literature, and most of them have an autosomal recessive pattern of inheritance. They all share a set of clinical symptoms, including progressive atrophy and weakness of voluntary muscles, predominantly at the hips and the shoulder, and the onset of symptoms vary from childhood to late adolescence. And this weakness of voluntary muscle will lead to a loss of ambulation in almost all patients. There are also associated symptoms, and the more important are respiratory and cardiac impairment that can, in the most severe forms of LGMDs, impact the life expectancy of the patients. LGMD-R9 presents some specific features, including the fact that more than 80% of patients are homozygote for the common mutation and present therefore the classical phenotype with an onset of symptoms around the age of 15 and a loss of ambulation 12 to 15 years later. Importantly, LGMD-R9 is associated with respiratory involvement in the majority of patients, more than 70% of them, and sometimes respiratory impairment can appear even before the loss of ambulation. Cardiac impairment is less frequent, still reported in 20 to 30% of patients. So here is a schematic representation of our construct, our gene therapy. We selected AAV9 for its good tropism to muscle cells, and we inserted a codon-optimized sequence to benefit from a higher FKRP expression in the muscle, as evidenced in this graph, where you can see that at both doses tested, we have seen a 5 times higher FKRP expression compared to the wild type sequence. And that was the same result for the 2 different doses tested. We also inserted a heart target sequence to modulate FKRP expression into the heart, because overexpression of FKRP in the heart can be associated with a cardiac toxicity, and I will get back on this a little later. And with all these features, we were able to restore the histological and functional level, the feature of the muscle, and this at particularly low dose in the mid 10 to the 12 Vg per kilogram. We performed our preclinical proof of concept studies in a newly developed muscle-specific knockout mice model, displaying severe dystrophy in skeletal muscles, very representative of the disease phenotype. The mice were treated at 1 month of age, and they were followed for 3 months before sacrifice of the mice. And this slide summarizes the most important result of the proof of concept study, starting with the histological feature. Here you have a normal muscle, histology muscle, with well-organized fibers, perinucleated, and fibers of similar size. And if you compare with the dystrophic mice model, you have now a lot of centronucleation, fibers of different size, inclusion of adipocytes, and fibrosis in between the fibers. Following injection of a single dose of our construct at 3 different doses, you have a progressive restoration of normal histology that was really like the normal muscle at mid and high dose. The centronucleation that we see as disappeared at mid and high dose was quantified, and you can see here the result in this diagram, where the dystrophic mice model is associated with a large increase in the centronucleation index compared to the wild-type mice. And after injection of our construct, there was a restoration to the wild-type level at mid and high dose. Similar result for the connective tissue in the diaphragm that shows the fibrosis in between muscle fibers. And here again, at mid and high dose, we had a restoration to the wild type level for the percentage of fibrosis tissue. At the functional level, we looked at individual muscle force, here the tibialis anterior, but also the global force of the mice. And you can see a restoration at the wild type level of the individual tibialis anterior force, even increase compared to the wild type level at the high dose. And for the global force, again, restoration at the wild type level. So I mentioned earlier the insertion of a target sequence to modulate FKRP expression into the heart. We did this because with an earlier construct in blue in this 3 graph, we had a very good result in terms of FKRP expression in the muscles, tibialis anterior diaphragm, but a very high expression of FKRP into the heart, and this was associated in animals model with a cardiac toxicity. By inserting this sequence, we decreased drastically the level of FKRP expression into the heart without modifying FKRP expression in the muscles. Same result for centronucleation and for global force. No difference between the two constructs. The only result was a drastic reduction of FKRP expression in the heart. And in our top study conducted in rat, it was associated with absolutely no cardiac toxicity up to 30 times the starting clinical dose. And for all those tested, we did not show any macroscopic or histologic changes in any organ. So now moving to our clinical development program, it consists of 2 studies, a natural history study that was initiated in 2020 and our gene therapy trial. For our natural history study, the study was fully enrolled last December with 52 patients enrolled, and as of today we have already half of the population that have reached 12-month follow-up. And this data, this baseline data, help us defining the target population for the gene therapy trial, where we wanted to select a population with already a moderate phenotype of LGMD R9, and we selected this population with a forced vital capacity between 40 and 80%. And as you can see, compared to the whole population in the natural history study, the population is slightly older with a lower FVC at baseline, 65%, showing already a moderate respiratory impairment and a muscular impairment more severe compared to the whole population, with a longer time to perform the 10-meter walk test, 30 seconds compared to 10 seconds in the whole population. You have to know that the normal time to perform 10 meter in a normal population, this is 5 to 6 seconds. And the NSAD, also the NSAD score was far lower in our target population compared to the whole population and a normal population with a normal score at 54. So here is the design of our gene therapy trial. This is a 2-stage clinical trial. We will start with a dose escalation phase, starting with the first cohort of 3 patients who will be treated with our clinical starting dose corresponding to the mid dose in our preclinical studies. And we will move to a second dose 3 times higher. And we will select the dose to carry over in stage 2 of the study based on the overall safety of these 6 first patients, but also based on the result of the muscle biopsy performed at 3 months after injection in the 2 cohorts. And we will select the dose based on the percentage of glycosylation, but also the percentage of positive fibers that have been transduced with our construct. For the second phase of the study, this is a placebo-controlled randomized trial with 33 patients enrolled, 22 in the treated group, 11 in the placebo group, but importantly, after 1 year, the patient will be switched to the opposite treatment. So all patients in the study will receive eventually the gene therapy at the selected dose. As said before, we will select a population with already a moderate phenotype in terms of respiratory impairment and muscular impairment, still ambulant population, but with an impact on the muscular function. And the primary endpoint that we selected and we filed to the agencies is the percentage in the forced vital capacity at 1 year as compared to the placebo group. We have a lot of secondary endpoints, muscular, NSAD, 10-meter walk test, but also other muscular endpoints. MRI, muscle MRI, to look at the percentage of fat repartition, and of course, several biomarkers. So, where we are with this program, we have received approval from UK, Denmark, and the French agencies. We have also the ethical committee's approval. We are finalizing the contract, and we expect the first patient to be treated in the coming weeks after we have activated the clinical sites. But we have also 4 additional programs with in vivo proof of concept, and I will say a few words on the next 2 programs for which we expect to enter into clinics within the next 12 to 18 months. So for gamma-sarcoglycanopathy, LGMD-R9, we have a different capsid selected, AAV8, and this time we inserted the wild type sequence of the gene, the SGC gene, because after some attempt with codon optimized sequence, we showed a lower efficacy with codon optimized sequences, so we went back to the wild type sequence. We also selected a different promoter. Here, this is the TMCK. That was a promoter tested that achieved the lower expression into the liver while maintaining the endogenous expression ratio for skeletal muscle and heart. So this program, we should file the IND, the clinical trial application, second half of this year for first patient treated early next year. And our second program that should enter soon into the clinic, this is for calpainopathy, where we will use a proprietary capsid developed by the Généton. And this proprietary capsid was chosen because of its very high skeletal muscle tropism and very low liver transduction. We also use a new promoter, a proprietary promoter from Généton, and to avoid overexpression into the heart. And here again, for this construct, we were able to show full restoration at the histological and functional level with dose in the mid 10 to the 12 Vg per kilogram. And I will finish with this slide showing the timelines of CTA IND submission for our first 5 programs. I describe a little more detail for these 3, but we have also proof of concept. We start to receive our proof of concept result for dysferlinopathy and alpha-sarcoglycanopathy, but we don't expect this program to enter into the clinic before 2024. Because we still have a lot of work, preclinical work, to perform on these 2 programs. And I think I reached my last slide. So I thank you very much for your attention.
My name is Jennifer Levy. I'm the Scientific Director at Coalition to Cure Calpain 3. It's my pleasure to introduce Vita Therapeutics. Vita is a biopharmaceutical company generating state-of-the-art cellular therapeutics for the treatment of debilitating neuromuscular diseases and cancers. Vita Therapeutics uses induced pluripotent stem cell technology to engineer specific cell types designed to replace those that are defective in patients. The company is progressing its lead therapeutic, VTA100, for the treatment of limb-girdle muscular dystrophy, with the goal of filing an investigational drug application with the U.S. Food and Drug Administration in 2023.
Good afternoon, everyone. And thank you, Jen, and thanks to the Speak Foundation for allowing Vita Therapeutics to be here today, presenting at the LGMD Global Advocacy Summit. To formally introduce myself and Vita Therapeutics, I am the co-founder and CEO of Vita Therapeutics, which is a cell engineering company focused on combining gene editing technology with traditional cell therapy, all with the hopes of potentially replacing defective cells in patients with functional cells. We got started about 2.5 years ago out of Johns Hopkins University. And to date, we've raised about $40 million collectively to fund both our neuromuscular platform as well as our immunotherapy platform. And today I'm joined by our SVP of Clinical Affairs, Dr. Steve Brooks, to talk a little bit more about VTA100, which is the lead program within our neuromuscular platform that we're developing and progressing into the clinic next year for the treatment of patients with limb-girdle muscular dystrophy 2A. So to give a very brief introduction, we use a very specific technology known as induced pluripotent stem cells to derive a very specific cell type known as satellite cells. Satellite cells, as we'll go through momentarily, are really the very foundation of muscle biology. And what we've been able to show preclinically is that we can actually use these satellite cells to actually regenerate human muscle. So long-term, our goal is to actually use these cells to treat a number of different muscular dystrophies. But to start, we really felt that LGMD 2A was really the best disease for us to move this into the clinic. And we've put together a program called VTA100, which right now has really every single partnership that's needed to get it into the clinic already fully established. And we're anticipating finding that first IND at some point in the middle to second half of next year. We really focus on a dual strategy to actually bring this into the clinic. The first focus is what we refer to as autologous therapy. So this means that we're actually building a very specific cell therapy to each and every specific patient. But the long-term goal is to actually use our proprietary hypoimmunogenic platform to actually derive one universal cell type or one universal cell line that we could use to treat every single patient regardless of their DNA. So hopefully that gives you a very quick introduction in terms of what we're working on here at Vita. And now I'll go a little bit backwards and kind of start with some of the basic biology of our technology before I dive into some of the latest data and in terms of where we're at preclinically. So as Jennifer mentioned earlier, we use a very specific technology called induced pluripotent stem cells. So for those who aren't familiar with this is technology that was invented by a Japanese scientist back in 2006 named Dr. Shinya Yamanaka. At the time, many folks were working on trying to develop a final cell type, like a cardiomyocyte, into another final cell type, like a neuron. Dr. Yamanaka, he thought a little bit differently. He said, before we can differentiate into other different cell types, maybe first we should go backwards into the very origin stem cell state. And that's what he did. He actually invented a specific technology where you can take a blood cell from an individual and actually reverse engineer it down to its stem cell state. So now with that invention, in theory, you could then use that technology to then reprogram those stem cells into any potential cell type in the body, and then deliver those cell types to patients who are, currently have defective cells. He would win the Nobel Prize for that technology back in 2012. And then fast forward to today, there's now a number of different companies who are developing this type of technology and advancing it into the clinic. So that hopefully gives a really good understanding of iPSCs in general. And then the next core piece of our technology was actually invented at Johns Hopkins University in the lab of Dr. Catherine Wagner and Dr. Gab Sangley. And really what they had done was use iPSC technology to really, for the first time ever, develop a human and now patented protocol to actually generate a human satellite cell. So why are satellite cells so important as it relates for neuromuscular diseases? Well, if you look at the graph on your right, you'll actually see what's called a muscle fast pull. And what this is really what your muscle cells look like in your body. It's a bunch of muscle fibers, muscle cells that bundle together to form these long fascicle groups. But what you'll notice is that there's these tiny little green cells that sit quiescently surrounding the muscle fascicle, and we call these satellite cells. So I'm sure many people here have heard the expression, when you're lifting weights, your muscles, they tear apart. Well, really what that means is your muscles are getting injured. And unfortunately, muscle cells get injured normally throughout our entire lives. It's this normal process called muscle homeostasis. So when our muscle cells get injured, a signal gets sent from the fascicle to the satellite cell itself to undergo a really specific mechanism known as asymmetric cell division. So when this occurs, two things happen. One, the satellite cell creates another copy of itself, another satellite cell that lies dormant, waiting to be called upon to do its job. And two, it actually creates a myoblast. Those myoblasts, they either go on to repair that damaged muscle or they replace it. All right, so that's why I said earlier, satellite cells are really the very foundation of muscle biology, because once our muscles get injured, they start the series of events that actually go on to repair and replace the muscle, right? So that's why we chose to really focus on satellite cells here, and why we thought that this could be a really great treatment for patients suffering from various different muscular dystrophies. So once Dr. Wagner and Dr. Lee had the protocol in hand to actually derive a human satellite cell, we had to then set up a number of experiments to actually prove that these were real human functioning satellite cells. So one of the first experiments that we did is where we take a mouse and we first induce it to a very specific process called irradiation. Now, for this specific experiment that we're showing you right here, we're actually just doing this in the tibialis anterior muscle of that mouse. Nonetheless, the irradiation comes through and it actually inhibits all of the satellite cells within that muscle. We then inject a cardiotoxin in to come through and actually kill off all the functioning muscle in that TA muscle of that mouse, right? So now there's no functioning satellite cells and there's no functioning muscle cells. We then inject our human satellite cells into the mouse, and after just 4 weeks, something pretty amazing happens. So this big picture on the right that you're looking at right here, that's actually a picture of functioning human muscle that was formed within that mouse as a result of our satellite cells. But what's equally important to this picture is the picture at the top right, which is zoomed in. That's actually showing quiescent satellite cells surrounding the muscle fascicles. So at the end of this experiment, we were pretty enthused about the potential for this treatment, but we still had one major question that we really needed to better understand, which of course involved doing another experiment. So here, we actually take that same set of mice and we actually re-inject a cardiotoxin in to come through and kill off all of the functioning human muscle that we had just created, right? So you can actually see that on the first picture on the right. This is 5 days after that re-injury. You're looking at a picture that has no functioning muscle whatsoever. But when you wait just 4 more weeks, you actually start to see the rise of functioning human muscle. Now, this is what gets us so excited, because what this experiment proves is that our satellite cells, they were truly quiescent, and that once the muscle was damaged, they started effectively undergoing that asymmetric cell division process and repairing and replacing the human muscle. So this experiment, along with a number of others, is what gives us the confidence that this could be a one-time only treatment that will continuously and sustainably go into repair and replace human muscle. So up to this point, all you've done is visibly see the muscle. So I think it's important to show some of the functional results that we had as well. So this was a separate study that we had done in a very specific mouse model known as the MDX mice model. This is the model that is well established and mimics the Duchenne's muscular dystrophy disease, which, as many of you know, is unfortunately the most severe form of muscular dystrophy. And what we're doing here is we're injecting our satellite cells into 3 muscles of that mouse. We're injecting them into the quad, the tibialis anterior, and the gastroc muscle. And it's there's 2 different cohorts: the mice that were treated with our satellite cells, and then we have our mice that were treated with a just static placebo. So really the key test here is actually that first graph on the left. And this is what we call the treadmill test. It's where you put the mice on a treadmill and you see how long they can run. So in this case, we actually tested about 1 month after our cells were injected. And as you can see, in the mice that were actually treated with our human satellite cells, they're running about 108% longer than the mice that were not treated, right? So this is how we not only are we visibly seeing human muscle as we just went through, but we're also seeing a very significant functional improvement in these mice. So as I said earlier, we've put together a protocol, or excuse me, a program that we call VTA100 to move these cells into the clinic specifically for limb-girdle muscular dystrophy 2A patients. So we break the manufacturing process down really into 4 different steps. So it all starts with the patient themselves. At this point, we do a blood extraction, we sort out for their PPMCs, and then we re-engineer them down to their iPSC state using that induced pluripotent stem cell technology. Once we've done that, because this is an autologous treatment, we have to actually correct the genetic mutation involved in the various different muscular dystrophy patients. So as it relates to LGMD2A, we're actually introducing a very healthy copy of the calpain-3 gene. We have a really efficient way of doing that. And once we've done that, we take that calpain-3 corrected iPSC, and we then differentiate it into our human satellite cells, and then finally expand those cells into clinically relevant numbers. So the reason why this manufacturing process is so critical is that really is what kicks off the entire preclinical step. Once we had taken that research protocol out of Hopkins, we had to work towards optimizing this protocol so that we could then actually use it and move it into the clinic. So to do that, about 1.5 years ago, we actually partnered with the CDMO who could help us take that research protocol and actually optimize it to what's referred to as a GMP protocol that we could actually use to treat patients. So fast forward to today, we've now done that. We have a GMP protocol that's now fully validated and fully optimized. And GMP production is actually just about to kick off and just get initiated. The next step in moving this into the clinic is actually kicking off what's referred to as our IND-enabling safety and efficacy studies. Very recently, we had a really fantastic and positive meeting with the FDA where we presented our program. We also presented the models that we had used historically. And the FDA actually accepted a lot of the models that we had used previously. And now all we have to do is replicate the exact same studies that we just did, but using a higher quality cell that was manufactured in that GMP facility. So we have 2 different CROs that we've already signed contracts for, and we're expecting to initiate both the safety and efficacy studies later this year. And then finally, really the last step has to do with the clinical sites that we're going to be working with and the patients themselves. So right now, we've had some really great dialogue with various different sites throughout the US as it relates to muscular dystrophy in general. And we've narrowed down a few that we're going to be looking to partner with more formally to actually kick off our clinical study late next year. And now the final and last step is really going out and talking to some of the patients that we're looking to identify that may be interested in volunteering for this phase 1/2 clinical trial. So hopefully that gives everyone a very good understanding as to who Vita Therapeutics is, what we're working on, and what VTA100 is. And now I'll turn it over to our SVP of Clinical Affairs, Dr. Steve Brooks, to talk a little bit more about the clinical trial. Thank you.
Thank you, Doug, for that presentation on the background on VTA100. My name is Steve Brooks. I'm the Senior Vice President of Clinical Affairs at Vita Therapeutics, and I'm going to talk about our first-in-human clinical protocol. This is a trial entitled a Phase 1/2 trial of intramuscular administration of human-induced pluripotent stem cell-derived satellite cells for limb-girdle 2AR1. It is a first-in-human study. It'll be single dose escalation, and there'll be randomization of the injected upper extremity for the first group. The target population is adults with genetically confirmed limb-girdle 2AR1. It is a phase 1 and 2 trial. So phase 1, primarily safety. Phase 2 will be looking for evidence of efficacy as well and doing some dose ranging. The number of subjects will be 15 divided into 4 cohorts, and the duration of the study will be 24 months with interim safety and effectiveness analysis. The primary objective will be to determine the safety and tolerability of intramuscular injection of the satellite cells for treatment of limb-girdle 2AR1. We have several secondary objectives. The first is to determine the optimal dose of intramuscular administration of the satellite cells. The second is to evaluate the humoral and cellular immune response to the satellite cells. And this is to make sure that the patients are not rejecting the cells that have been genetically altered and the protein, which will be novel to their body. We'll also assess calpain-3 expression in the muscle biopsies. We'll evaluate change in muscle volume and intramuscular fat fraction. And lastly, we'll evaluate measures of efficacy for the satellite cells on motor function, including the North Star test, the pool test, handheld myometry, manual muscle testing, forced stair climb, forced stair descent, timed up and go, and 100-meter walk. The study is divided into 4 study periods. There's a screening period where patients will be recruited, they'll be consented, we'll verify their inclusion and exclusion criteria, and perform baseline functional studies to ensure that they're eligible. If they meet all criteria, they'll then be enrolled. There'll then be a lead-in period. The manufacture of VTA-100 takes approximately 6 months from the time of the blood draw until we have enough cells for injection. During that time, this will serve as a natural history period where we'll take baseline studies and follow up with baseline studies again at 6 months during this observation period. Next will be the study treatment period. This will be when intramuscular injection of the VTA-100 will be done. And then we'll have a follow-up period where these patients will be monitored for safety and effectiveness over the next 6 months. The trial is divided into 2 phases, phase 1 and phase 2. The first phase, patients will be randomized to the treatment arm. We'll be treating one arm and the other will receive an equivalent volume and number of injections of an excipient vehicle solution without the cells. The number of injections and the locations will be identical. All patients in all groups will be treated. There will be no control patients. However, the patients will serve as their own controls. In phase 2, after these patients have reached safety and have been validated by the CEC that we may proceed, we will then treat the contralateral arm. So the arm that was not treated originally will be treated at the optimal dose. So we're guessing that may be the medium dose or the high dose. So patients that receive dosing below that optimal dose will have the treated muscles topped up, and then they'll receive treatment in the upper extremity muscles that were not yet treated. If they're eligible, they'll also be treated in the lower extremities bilaterally. This is a schematic, but it's a little bit easier to understand. Starting on the left, patients will be screened. If they meet selection criteria, including a history, the laboratory studies and whole body MRI, as well as functional testing, they'll be enrolled. Then they'll be enrolled into one of 4 cohorts. The first cohort in phase 1 will undergo a low volume injection, so 1 million cells. They'll be treated into the biceps only, and that's 1 million cells per injection. They'll be treated unilaterally in the biceps, and the other arm, they'll be treated with contralateral control solution. Cohort 2 is the medium dosing of cells, so 5 million cells per injection, and cohort 3 is the high dose group. They'll be treated unilaterally in the arm that they're randomized to, but instead of just the biceps, they'll be treated in the biceps, triceps, and deltoid muscles. On the contralateral arm, they'll have a control solution injected. At 6 months safety, they will be validated by the CEC. If it is deemed that we are safe to proceed, then the patients will be treated again. So patients from cohort 1 that were treated with low dose cells, so 1 million cells per injection into just the biceps, will be topped up to the optimal dose. So if it is the medium dose of 5 million cells, they'll then receive 4 million cells in the biceps that was already treated. If it's the high dose, they'll receive 9 million cells. And then they'll receive full dosing at the optimal dose to the triceps and deltoid on the randomized arm. In the contralateral arm, they'll receive optimal dosing to biceps, triceps, and deltoid. Assuming that they are eligible for treatment in the lower extremities, they'll then receive treatment in the vastus lateralis, vastus medialis, and rectus femoris of both legs. Cohort 2 and cohort 3, they will be topped up if the dose is the high dose, and they'll be treated in the contralateral arm and bilaterally in the lower extremities. At this same time, we will enroll cohort 4. These patients will not be randomized. They will receive treatment at the optimal dose bilaterally to the biceps, triceps, and deltoid in the upper extremity and to the both lower extremity muscles, assuming that they're eligible. The inclusion criteria for patients, they must meet all the following criteria. They must willingly sign the informed consent and agree to the protocol. They must have a clinical and genetic diagnosis of limb-girdle 2A with confirmed bioallelic pathogenic variants of calpain-3. We'll be enrolling both male and female patients between the ages of 18 and 65 years of age, and patients must be able to understand and comply with all study procedures. The exclusion criteria, if they have any ongoing medical condition outside of the limb-girdle which makes them high risk for the study, this may be other cardiac findings, metabolic findings, or complications that would make them high risk, such as pulmonary or cardiac involvement. If they have abnormal liver or kidney testing, they would also be ineligible. Another exclusion criteria is contraindication to MRI. We'll be doing full body MRI if they have implanted spinal rods or other foreign bodies that may interfere with the pacemaker, or that may interfere with the imaging or make MRI unable to be done. These patients would be excluded. Again, if they have abnormal liver or kidney function, they would be excluded. If they are involved in any other investigational trials or have taken an investigational drug within 3 months prior to starting the treatment, we would also exclude them. Female patients who are pregnant or breastfeeding would be excluded. We will be doing some functional studies. So we want to target patients with upper extremity involvement. We know that fatty infiltration, which is evident on MRI, can be seen in the earliest stages of muscle involvement. So we want to have some, but not too much. If the muscle is already so infiltrated with fat, there is less of a chance that our treatment would be effective. Therefore, we'll be targeting patients with between 25 to 75% fat fraction, and outside of that, patients will be excluded in their upper extremity muscles. Study procedures, after patients are enrolled, they'll have a history of physical exam and laboratory testing. They'll also undergo baseline assessments, including full body MRI. They'll have an open muscle biopsy of the biceps brachii bilaterally. They'll undergo the North Star assessment for limb-girdle, the revised pool test, handheld myometry, and manual muscle testing. They'll also undergo functional testing of the lower extremities at this time for a baseline. Remember, the VTA 100 takes approximately 6 months to manufacture. During that period, we'll have our own natural history study with strength and functional studies as well as MRI. So we'll also be assessing whether these folks may be eligible to receive treatment in phase 2 in the lower extremities. We will not be excluding non-ambulatory patients. In fact, we encourage those folks if they meet the other criteria. Safety evaluations on the first day of treatment will occur every 3 hours. After that, we'll undergo safety evaluations on day 7, 30, and every 3 months afterwards. Patients will undergo routine laboratory testing, including a basic metabolic panel, CBC, PT, PTT, liver and kidney tests such as GGT and cystatin C, as well as CPK. They'll undergo physical exam. We'll also be drawing histopathological inflammatory markers. We'll be interviewing for adverse events or serious adverse events, and patients will also undergo full body MRI. We'll be paying particular attention to locations in the muscles where they were injected, looking for evidence of inflammation and also malignancy. Efficacy evaluations will be performed at the baseline, 1, 3, and 6 months, with the exception of open muscle biopsies, which will only be performed at baseline and at 6 months. We'll be measuring calpain-3 expression by Western blot and also immunohistochemistry. We'll also be using an assay looking for calpain-3 activity as evidence of success. At baseline, there should be minimal to no activity in these patients. We'll be measuring CPKs. We'll be performing the North Star and the pool test, handheld myometry, manual muscle testing, in-call limb-girdle DHI tests. Muscle quality will be assessed by whole body MRI. And again, we'll be looking for progression of disease or lack of progression and perhaps even increase in muscle mass, which we'll be able to quantitatively measure. We'll be able to see the fat fraction, and if there is increase in the muscle mass, this may be reflected by an overall decrease in the fat fraction. We'll be measuring 4-stair climb, 4-stair descend, timed up and go, and 100-meter walk. So that is our plans at current for our trial. We thank you for hosting us today. And if you have interest or questions, please do reach out. We would love to talk to you. Thank you very much.
AskBio is a fully integrated gene therapy company dedicated to developing life-saving medicines. The company maintains a portfolio of clinical programs across a range of neuromuscular, central nervous system, cardiovascular, and metabolic disease indications with a clinical stage pipeline that includes therapeutics for Pompe disease, Parkinson's disease, and congestive heart failure. Today's presentation will be focused on their upcoming clinical trial for limb-girdle muscular dystrophy 2I/R9.
Hey, thank you so much, Kelly, for the introduction. So, good morning, good afternoon, or good evening, everyone. I just want to welcome you to our webinar. So I just want to introduce myself first. My name is Alain Lamontagne. I have the honor to lead the medical affairs organization at AskBio. And actually today, I'm with my colleague, at Eng and Yuan, leading, at our, musculoskeletal program at AskBio. So I just want to say welcome. We're very, very excited, to be here. And obviously what we want to do today is to give you an update on our limb-girdle 2I program, and one of the things that Eng and I were going to do is share the outcomes of our collaboration with over the years with family, with patients, with healthcare professionals, researchers, all involved in limb-girdle. So once again, great to be here, and I hope we're gonna have fun in the next 25-35 minutes. So here's what I would like to do. What we want to do is first of all introduce AskBio. So who is AskBio? What is this organization? As well, I'm a former teacher, so what I'm going to do is I'm going to give you some background about gene therapy, and it's very popular right now, and I just want to make sure that I simplified the different concept, and hopefully you're going to learn a few things about gene therapy today. And obviously the most exciting part of this webinar will be with Hank, where we're going to give you an update of our Lion 101 clinical program that should be ongoing at some point in 2022. So before we start, I just want to obviously add the following disclosures and disclaimers. So Eng and I are speakers and employees at AskBio. We're not here to offer any kind of medical advices, legal or regulatory information. We're here obviously to present public domain informations. We're not here to promote a product or a service, and the information, as I said, are public domain informations, and obviously we own we build the presentation ourselves. Obviously this is for education purposes only. So let's get started. So let's talk about AskBio. So the first thing I would like to share with you is AskBio is one of member of the Bayer family, was acquired back in 2020 from Bayer. And I think this slide illustrates really who we are as an organization. So AskBio is driven by science. And we feel that we are the leader in gene therapy. And why? Obviously, we need to go back in when we founded, our founders founded the company back in 2001. So the company was founded by two people, Sheila McHale, currently our CEO of the organization, and as well as Jude Samulski. And Jude is a well-known, person working, been working for many years in gene therapy. And I like to say, that this year Jude is celebrating 40 years of research dedicated to gene therapy patients. And for me, this is once again is who we are as an organization. So we're the first organization, to clone AAV, associated adeno-associated virus, which is, I'm going to give you a little bit more information. What it is this is a foundational vector for gene therapy. As well, our organization treated, the first company organization to treat patients in the clinic with Duchenne muscular dystrophy, even Pompe with gene therapy. And also, back in in early 2000, deliver, the gene therapy to the brain for some pediatric patients. So this is a bit of foundation of our organization. The other thing that I would like to share with you today is not only we're doing discovery, we're still doing a lot of research and development to understand, obviously, how can we improve, what vectors used for gene therapy, as well try to find, obviously initiate new clinical programs to address different unmet medical needs. But the other thing that I'm very proud to share is not only we are developing products in preclinical stage as well in the clinic now, and I'm going to share more information shortly, but also we have our own manufacturing capabilities within our organization, so which is key as we're going to grow the number of clinical programs. The other thing that I would like to share in terms of manufacturing, I'll give you an example. For example, during the process when we produce our vector, we don't use any animal or human byproducts within a manufacturing process, which is, we believe, is going to be at a faster way, and maybe we'll see later on maybe a safer way to produce vectors for a clinical program moving forward. So as I said, we're very proud. We're in the clinic right now for rare disease like Pompe disease. But as well, when in clinical phase 1 and 2 for other more common diseases like congestive heart failure or even Parkinson's disease. But I think that and I think what is exciting and for you listening to this webcast today is obviously our program for limb-girdle 2R9 program that we're going to share more information today. As well, very exciting. We have other programs. They're going to hopefully enter the clinic at some point in 2022. And also we have, at other programs where we want to look at, other, CNS, at diseases, liver diseases, or hematologic, conditions using, at our gene therapy, platform. So this is enough about AskBio. I hope it gives you a little bit more information about our organization. So let's now talk about, what is gene therapy. And before I talk about gene therapy, I just want to go and set up the foundation about, what are genes? What is a gene? So genes are, I said to, and many, and especially the younger patients, I say, hey, genes it's our DNA. It's our blueprint. They are the instruction for growth and for the health of our cells. And also, why people are a little bit different. Why I'm taller than Hank, for example. Why my eyes are brown. So they are obviously within our instructions within the cell or genes. So the cell, they read genes or the instruction to make the protein, and the protein are used by cell to signal health of a cell and do also many other functions to keep the cell alive. So the protein are a key component of our body. So gene therapy leverage this natural function of cells to make protein. And we use this as a way to obviously use as a medication. So now I give you some insight about what is gene therapy. So gene therapy, it's pretty straightforward. Uses a copy of DNA, a gene, as a new way to treat a disease. So what we do, and researchers, what they do is really to package this information, this gene, and other, regulatory pieces, small pieces, into an empty viral vector that's going to help the LT gene to enter, the cell of impacted with the disease. So, and when, we transfer this information into the cell of the disease, this vector release, the information to the targeted cells, and then the cell can read the information and have, obviously, the desired therapeutic effect. So in a nutshell, this is what is gene therapy. So, and obviously, there are many approaches to gene therapy as a potential therapeutic options. So the goal of gene therapy and what we try to do is really to design for a targeted delivery and continuous long-term activity of the therapeutic, materials or the gene that we want to deliver to the disease cells. So there are three at least different options here. You could be in a situation where a patient, and actually this is obviously relevant for patients with limb-girdle, too high disease. And what we do is in this patient, as you have a mutation in one specific gene, so the fukutin gene related protein, and in the fukutin genes you have a mutation. What we want to do is bring the natural fukutin gene and to correct this information. And what we try to achieve here is to restore the normal function of the gene. This is one option and is really relevant for our audience today. There are other situations where the mutations, it's actually, you have, an expression of a bad gene. And what you want to do, you want to inhibit, the expression of this bad gene. So what we want to do, and we try to inactivate, the function in this specific case. I'm going to give you an example that is relevant. For example, Huntington disease. There's in the patient, they have mutated Huntington, and we want to stop the production of this protein as a potential therapeutic approach. So the third one, finally, is we call it the modified function, where you want to add a gene that's going to enhance a function. And within ASBio, I'm going to give you an example where we have a program for Parkinson's disease where we want to, express, the growth factor GDNF to modify the functions and obviously help the, the neurons in the patients. So in a nutshell, these are the different, the options, that we have in terms of gene therapy, treatment. So gene therapy uses modify viruses to deliver a therapeutic agent or gene. If you look at the last few decades, and one of the most amazing discovery was the research done to illustrate, to understand how we can use an adeno-associated virus, or AAV, as a vector to deliver a gene. This was amazing. The reason why I think it was the most amazing discovery of modern medicine is if you look currently, right now, there's two gene therapy approved product by the FDA and other agency, and are currently using AAV, adeno-associated virus, as a platform for the gene transfer. So why is is a popular platform, and why with ASBio is the vector that we use for gene therapy, and the vector we were going to use, and Hank, my colleague, is going to give you more information for a limb-girdle 2I program, is for the following reason. First of all, AAVs are incapable of replicating DNA by themselves. And the other things that you should know is himself, the AAV doesn't cause a disease. You're not sick because of it. The other thing, as I shared in the previous slides, AAVs can be modified by researchers to deliver a specific gene of interest into living cells and targeting specific tissue. And the other thing that is key on the safety point of view is AAV vectors, they don't change the DNA. They don't out there integrate within your genetic material. They are just there to help to deliver the gene and deliver the gene within the specific targeted cell. And this is the reason why we're using this approach for a gene therapy program. And you can see on the right hand side of the slide an illustration of what is a AAV, which is just a shell per se. And then what we do is we insert our fukutin gene inside of our AAV vector, and this is what we deliver in our clinical program. So in a nutshell, so it works, and I like this illustration here. So what we do in the lab, so we have our our gene, we insert our gene in our vector, AAV-MT vector, And this vector is purified. We have our own manufacturing process, and this material is obviously infused in IV infusion in our patients for limb-girdle 2I clinical program. And this, obviously, after this infusion, there's a release of the AAV, particles throughout the body. The AAV was interesting, is going to recognize the target cells, going to bind the target cell. And after binding of the target cell, going to crosses the cellular membrane, go into the nucleus of the target cell and release the DNA. And what's going to happen is the cell going to use obviously our own machinery to read the information on this on this new gene that is going to be only within the nucleus as a episomal a molecule, so a standalone molecule, and then using the normal machinery of the cell, going to read and produce, as you can see here on the slide, the protein, the fugitin protein. And hopefully we can see benefits from here. So I have another way to explain, and recently my niece asked me a question, say, "Oh, I don't understand. Can you explain for me?" And I can see, I can read, I can look at the slide. What I said to her? I said, let's think about a package. A package is your gene. So what you do with your package, you want to deliver this package to a different house. So what you do is you have your package, goes inside a mail truck. The mail truck is the AAV. And then the mail truck has the information to go directly to the right address. So the truck, the highway, different road, and knock at the door and deliver the package, which is a DNA. This is, using different words, what is exactly we try to achieve, and actually, what is gene therapy? So in summary, what I spent the last few minutes with you is I hope you have a better understanding that obviously gene transfer, gene therapy is a new innovative treatment approach for obviously monogenic diseases which are caused by mutations or overexpression of a single gene. AAV vectors can be used to transfer gene, and what's interesting, can be manipulate, bioengineer to target specific cells, specific tissue. I think why it's so popular, and we see so many clinical trials, currently ongoing. Using AAV is a strategy currently, investigated for obviously many different, diseases, and particularly, monogenic diseases like limb-girdle. On that, I'm going to turn over to my colleague Zhang to give you a little more information about our limb-girdle 2I R9 program. Thank you.
And thanks, Alain. Appreciate your primer on gene therapy. And if you move forward to the next slide, again, my name is Ang Nguyen. I'm the therapeutic lead for the musculoskeletal rare disease programs here at AskBio. So the limb-girdle muscular dystrophies represent about 34 different disorders categorized by a defect in some gene involving either a structural protein or an enzyme that impacts basically the anchoring of muscle cells to one another, and also to experience wear and tear. And depending upon the defect in the gene, one either has slower wear and tear or faster wear and tear, ultimately resulting in some sort of muscle weakness and damage over time. The reason why it's called limb-girdle muscular dystrophy is because the impacted muscles usually are those that attach the appendages to the torso, the shoulder girdle for the arms and the hip girdle for the legs. Specifically at AskBio, we are focusing on a disease, LGMD 2I-R9, and 2I-R9 is caused by a defect in a gene for fugitin-related protein, FKRP. And FKRP is an enzyme which is involved in the glycosylation of a part of that anchoring of the muscle cell, this is called alpha-dystroglycan. And so the glycosylation, the sugar coating of alpha-dystroglycan, is a critical component to help the muscles hook to each other, form muscle fibers, and to move in a synchronous fashion. In addition to all that, it helps as a shock absorber to make sure that you minimize wear and tear. And so without the FKRP, the enzyme, the muscle cells are unable to bind appropriately to each other, and this causes an increased risk for damage, chronic inflammation, and eventually over time breakdown of the muscle fibers, and hence the muscle wasting pattern and damage in this, again, around the shoulder and the hip girdles, slowly progressing, eventually leading to premature death. In the North American and European communities, the most common FKRP mutation is something called L276I, and that's, again, about 70% of the European cases, and about 25 to 30% are basically a heterozygote mutation, and the majority are homozygous mutation. And so I'll let you move to the next slide. Thank you. And so for LGMD2I/R9, there are no approved therapies for the treatment of the disease, and thus the standard of care is really just symptomatic management. This is around, physical therapy, occupational therapy, assistive devices, and then symptomatic therapy for heart or lung disease. As you know, there are new therapies for the muscular dystrophies, and namely exciting times for 2I/R9, be it substrate therapies or gene therapies. And so for us at AskBio, there was a question of, can there be a gene augmentation strategy, i.e. a physiologic replacement of FKRP enzyme performed through the use of gene therapy? And so moving to the next slide, our program at AskBio, and the investigational product is called Lion 101. Again, is intended to provide physiologic restoration of FKRP enzyme. So we're pursuing a series of clinical studies in order to evaluate the Lion 101 therapy for the treatment of 2I/R9 with the proposed indication, as you can see here, that we're using a AAV gene therapy, base gene therapy, for the treatment of LGMD2I/R9 in the young and the younger pediatric and adult population for both homozygous and heterozygous mutations. So we move to the next slide. So as described, there's to be a series of clinical programs or clinical studies in order to evaluate Lion 101. And this year, and we are in the process of doing pre-screening and screening and dosing of the Lion 101 therapy in a first-in-human study. And as you can see on the screen here, there's a series of studies, a pilot, a pivotal, and a post-approval. We're moving into the pilot phase, where the purposes of the first-in-human study are to understand the safety first and foremost, and then the activity of the Lion 101 therapy, i.e. does it demonstrate evidence of a successful gene transfer, as well as expression of the gene, the FKRP gene, and thus increase levels of FKRP appropriately, and subsequently determine whether there's some preliminary evidence that we're getting the effects of the increased levels of enzyme. And from that pilot study, we identified that it's safe, there's a target activity, and also a recommended dose for which we'd further evaluate it in a larger program, the pivotal study, and that would be for the purposes of the confirmation of the safety and efficacy, and for the purposes of marketing, submission, and approval. And so our program is a double-blind, randomized, placebo-controlled study, which means that persons who are enrolled in the program will receive either the Lion 101 therapy itself or a placebo comparator. and to understand, again, the safety and efficacy of the Line 101 product. For people, for persons who receive the placebo product beginning of the study, they will be offered, if remaining eligible, an opportunity to receive the Line 101 investigational gene therapy, and thus everyone who participates in the study would be, again, remaining eligible, would be receiving the Line 101. Next slide. And so if you want to learn more about the Lion 101 clinical program, and namely this specific study is called the Lion CS 101 study, you can get on clinicaltrials.gov, and to learn more about the program, and the clinicaltrials.gov number, as you can see here on the screen. And so moving forward, as we start pre-screening and screening and dosing in this year, we'd like to describe what the pre-screening is. And so in order to receive a gene therapy, we want to make sure that that one does not have an immune reaction to the viral vector itself. And so as Alen had described, we use a viral capsid in an envelope to hold the investigational product. And these are naturally occurring viruses in the community. And so a certain fraction of the community will have innate immunity against the viral vector. And so in order to be eligible to receive the Line 101 gene therapy, one would have to have an absence of neutralizing antibodies, i.e. you don't have an immunity against the gene therapy. And so that would be screened for in a pre-screening process, a blood sample to be checked for your immunity. In addition to that, we would be in the process of confirming the FKRP mutation. And so this pre-screening process would occur prior to dosing, approximately 6 weeks prior to any dosing of the therapy. If the pre-screening is successful, then you would go forward into screening, and that would be the next slide. And so screening basically involves an opportunity to evaluate whether we believe we could see good safety profile and efficacy profile from the gene therapy. And so we'd want to identify what we would consider the healthiest population to see these sort of differences between placebo and the Line 101 therapy. And thus it's based upon genetic confirmation of disease, and then on top of it is an assessment of physical function. So we can see here a 4-stair climb, the ability to complete a 10-meter walk test. And this basic, again, is to assess a physical function for which we believe that we could see differences between the treatment assignments during the course of this study. And next slide, please. And so the exclusion criteria for the first in human study include, first and foremost, is the pre-screening that one does not have an immunity against the viral vector, the Line 101 gene therapy. And in addition to that, making sure that there are no comorbidities that may change the safety profile of the clinical study. And this would include diseases of the heart, liver, kidney, as well as significant pulmonary dysfunction. And ultimately, these are for purposes of making sure that we provide first and foremost safety for people who decide to volunteer and participate in our clinical study. The focus of us as a science-focused organization is to always focus on the patient's safety and a focused development plan, which will provide the best data package to show to the clinical community, the patient community, and to the overall global regulatory authorities that we have a product, again, which is safe and efficacious. And next slide. And so again, if you have questions regarding our clinical program, please refer to the clinicaltrials.gov website under an NCT number. And in addition to that, you can contact us at AskBio at our patient engagement platform called Ask First. And you can see the email here at first@askbio.com, where we can provide questions and answers and clarifications for your inquiries regarding our Lion CS 101 program. And thank you.
Doug Sproul is the Chief Medical Officer of ML Biosolutions. He is a pediatric neuromuscular specialist by training and managed a large clinical and clinical research practice prior to joining industry, first at Genzyme and then with Avexis, where he was a major contributor in the development of Zolgensma, the first gene therapy for neuromuscular disease, prior to joining ML Biosolutions. His career, both as a clinician researcher and industry partner, has focused on expanding therapeutic options and improving care for children and adults with neuromuscular diseases.
Kelly, thank you for that kind introduction. Good afternoon. As she mentioned, my name is Doug Sproul, and I'm the Chief Medical Officer here at ML Biosolutions. Thank you for giving me the opportunity to speak with you today on behalf of ML Biosolutions and our partners at BridgeBio regarding our program to develop BBP 418 as a potential treatment for patients with limb-girdle muscular dystrophy type 2I, also known as LGMD R9 FKRP related. BBP 418 is an investigational product that is still under development as a potential treatment of LGMD 2I. The information we are about to share with you about BBP 418 should not be interpreted that the drug has been demonstrated to be safe and effective as a treatment for LGMD 2I or any other diseases or disorders. ML Biosolutions owes its origin to the patient community and is a wonderful example of how patients and families can help drive therapeutic advances. The McCall Lockwood Lab was created in 2001, supported by a generous grant from the McCall and Lockwood families. And BBP 418 emerged directly from the work of Keelong Liu at the McCall Lockwood Lab at Atrium. ML Bio was created in 2018, and we've proceeded all the way from a pre-investigational new drug application or pre-IND phase all the way to phase 2 in less than 3 years. I would be remiss without mentioning the strength of our partnership with BridgeBio. Together, we share a mission to bring clinically meaningful therapies to patients as quickly and safely as possible. We are dedicated to fostering meaningful relationships with patients and their families by staying connected, listening to their experiences, and applying their insights into our work. And we hope that by developing these relationships with patients, families, and patient advocacy organizations early in the development process phase of our therapies, that we can craft our clinical trial protocols with the patient experience at the forefront. The origin of the ML Bio story is rooted in patient advocacy, and we have held our obligation to those living with LGMD2I at the core of our mission. Remaining connected to the patient community is critical to our work. To that end, we hold regular check-ins with patient advocacy organization partners, and we host periodic focus groups with patients and family members. We spent time attempting to truly understand the patient experience, what it's like from the time that initial symptoms present to the sometimes lengthy and stressful and ambiguous diagnostic journey, to the reality of living with LGMD2I on a daily basis, adapting to the progression of the disease, changes over time, access and getting appropriate care, and coping with the emotional demands and burdens of the disease. Also, we aim to understand why patients choose to participate in clinical trials and what they hope for in a treatment. And these insights help inform our clinical studies. By understanding the priorities and needs of the community, we can employ research methods that consider patient and participant preferences while remaining scientifically rigorous. We understand that the work we do represents hope for the LGMD2I community, and we strongly believe that by listening to and partnering with this community is the best way to move our program forward. And we thank everyone who has taken the time to connect with us over the years, and we thank in advance all those who will continue to partner and engage with us over the years ahead. Limb-girdle muscular dystrophy type 2I, also known as LGMD R9 FKRP related, is an autosomal recessive disease caused by the loss of function of a gene called FKRP. As I'll talk about in greater detail on the next few slides, FKRP is an enzyme that attaches a molecule called CDP ribitol to a string of sugars on a different protein called alpha-dystroglycan. BBP 418 is designed to be a substrate supplementation therapy, meaning we're providing elevated levels of a key substrate, upstream substrate for FKRP to work to increase the remaining activity of that mutated enzyme. Think of a substrate as the fuel that an enzyme such as FKRP will use to do its work. Theoretically, this substrate supplementation therapy helps make the partially functional FKRP gene work better, and thus it's intended to treat the root cause of disease in LGMD2I. BBP 418 is an oral therapy. It's provided in a powdered form and dissolved into water, and we expect that it will be taken twice a day. Our program is moving forward rapidly, and we've been granted Fast Track designation, or FTD, by the Food and Drug Administration, and Orphan Drug designation, or ODD, by the FDA and the European Medicines Agency, which is the European equivalent, as a recognition of the existing unmet medical need for treatments for LGMD2I. These designations are granted by the FDA and EMA as a recognition regarding the unmet medical need that exists and provides certain incentives for companies to develop treatments for serious rare diseases. As you all know, LGMD2I is a progressive disease with a huge need for effective therapies. There's currently no approved disease-modifying therapy for LGMD2I. And while we don't know exactly how many patients are actually affected by LGMD2I, we believe there are several thousand spread across the United States and Europe, and obviously many more in other countries around the world. Most patients with LGMD2I harbor 2 copies of a mutation referred to as the common mutation, or L276I. Patients with this genetic profile are often referred to as homozygotes and typically develop symptoms somewhat later in life and decline less rapidly than patients who harbor 2 different pathogenic mutations of FKRP. The genetic profile of these individuals is commonly referred to as a compound heterozygote. Regardless of one's genotype, the disease will progress, leading to the loss of ability to walk as early as the late teen years in some patients. Approximately half of all LGMD2I patients will ultimately lose the ability to walk. This progression may also affect other muscles. As the heart and diaphragm are muscles, many patients will start to develop breathing problems and heart problems during adulthood as well. For patients with LGMD2I, it's important to understand the complex processes that, when impaired, lead to your disease. For patients with LGMD2I, the problem is that there's a mutation in the FKRP gene that produces an FKRP enzyme that does not work properly. When the FKRP enzyme does not work properly, it disrupts a series of events that are required to form an important shock absorber for muscle cells. This shock absorber is called alpha-dystroglycan, or ADG for short. This is a protein that attaches to the cell membrane of our muscle cells and, together with a bunch of other proteins, protects the muscle cell from damage. For alpha-dystroglycan to work correctly, it must have a chain of sugars called glycans attached. And this is where FKRP comes into the story. This is a process called glycosylation. And FKRP is an enzyme involved in the attachment of one of these sugars, CDP ribitol, to the growing glycan chain. If it doesn't work correctly, then this glycan chain doesn't form properly, and alpha-dystroglycan in turn doesn't work in its shock absorber function for the muscle cell. So the hypothesis behind BBP 418 is called substrate supplementation. And on the left side of the figure is what happens in the untreated disease state. FKRP doesn't work correctly, and this leads to many of the alpha-dystroglycan proteins lacking that fully formed glycan chain. And these hypoglycosylated or underglycosylated ADG proteins do not work correctly in their shock absorber function. And this leads to chronic injury to the muscle cells. And over time, these cells will die off and be replaced by scar and fatty tissue. And this is what we call a muscular dystrophy. On the right is ML Bio's theoretical therapeutic approach. Again, this is an experimental therapy, and the exact mechanism of action is still under investigation. But our premise is that by providing high synthetic amounts of this natural substrate, that this leads to high levels of CDP ribitol in the muscle cell. While the FKRP enzyme doesn't work correctly, it works well enough that with lots and lots of that extra substrate, that extra fuel, that it will successfully attach the CDP ribitol to more of the glycan chains. And this leads to more of the fully formed alpha-dystroglycan proteins, which, if sufficient, should address that root cause problem in this disease. It's a simple, elegant solution to a complex problem. I would note that this strategy is only potentially applicable to LGMD2I and a few other forms of LGMD, such as 2M and 2U, that are caused by deficient glycosylation of alpha-dystroglycan. It would not help treat other forms of limb-girdle muscular dystrophy. While we're still quite early in the development process, we are reassured by how well tolerated BBP-418 has been to date in our clinical trials. And we believe this is because BBP-418 is a synthesized version of a naturally occurring compound. While we are providing high levels of this compound, to date there have been no dose-limiting toxicities, discontinuations, or serious adverse events related to this therapy. It's also quite easy to take. BBP-418 is provided in a powdered form that is dissolved into water, and we expect that the drug, if approved, would be likely taken twice a day. We at ML Biosolutions are proud of what we have accomplished together, and we are excited to partner with the community in our efforts to provide a safe, effective therapy to patients affected by limb-girdle muscular dystrophy type 2I. While I cannot describe everything that's been achieved over the past couple of years, I would like to highlight a number of accomplishments. In partnership with the GRASP Consortium, an ML Bio-sponsored natural history study called the Lead-In Study has been fully enrolled with over 100 patients across 11 sites in the United States and Europe. I want to congratulate our partners at these institutions, particularly Nick Johnson and the team at Virginia Commonwealth University, for this achievement. And data from this effort will be emerging over the next couple of years. We have completed 2 phase 1 trials to establish the safety and tolerability profile of BBP-418 in healthy volunteers, and have initiated a phase 2 trial with 14 LGMD2I patients at VCU in Richmond, Virginia. We recently presented some of the early phase 2 data at the Muscular Dystrophy Association annual conference. We've continued to partner with the patient community in our development program, and we've conducted an online survey of 80 LGMD2I patients and caregivers to collect feedback for the phase 3 trial design, and have hosted focus groups and held patient advocacy leader advisory meetings for additional feedback. And lastly, we've been granted Fast Track designation, as I mentioned previously, by the FDA, and Orphan Drug designation by the FDA and the European Medicines Agency. And we're excited to meet soon with the FDA to discuss our phase 3 study design and to finalize this protocol with input from regulatory authorities, physician leaders, and the patient community. And our biggest looming milestone will be forthcoming later this year with additional details regarding the location and timing of our phase 3 study start. I thank you for the opportunity to speak today on behalf of ML Biosolutions and Bridge Bio. I would encourage anyone with questions about our program to reach out to us at info@mlbiosolutions.com. I wish you a good day, good health, and a fantastic conference. Thank you, and I wish you a good day.
An externally led, patient-focused drug development meeting for 6 limb-girdle subtypes will be held on September 23rd and will focus on the 4 sarcoglycanopathies. LGMD 2C R5, 2D R3, 2E R4, 2F R6, as well as LGMD 2A R1 and 2I R9. Your involvement is critical to the success of this vital initiative for our limb-girdle community. This EL-PFDD meeting will give the FDA and other key stakeholders an opportunity to hear directly from those of you living with LGMD, your family, and caregivers. Your input will provide critical context for the FDA regulatory decision-making process for new drugs and treatment. This virtual meeting will include live speakers, pre-recorded panelists, and throughout the day, live polling and discussion forums featuring you, our viewing audience. The morning session will focus on subtype symptoms, their health effects, and the toll these 6 forms of LGMD take on your daily living. The afternoon session will highlight our community's perspectives on currently available and future treatments and communicate our tolerance to risk. Afterwards, a Voice of the Patient meeting summary report will be issued. It will serve as an educational reference document for the FDA, medical product developers, healthcare providers, federal partners, and the LGMD community. We encourage individuals living with these 6 LGMD subtypes and their caregivers to join our team. So please register at thespeakfoundation.com/PFDD to keep apprised of the planning process and to learn how your voice will make a difference on September 23rd.
I want to share with you a little more about the Believe There Is Hope for a Cure campaign. In 2022 and in 2023, we will focus at the Speak Foundation on raising funds to help further advocacy and research for all forms of limb-girdle muscular dystrophy. Currently, there is a large disparity in research where potential treatments are just not applicable to all forms. With over 30 genetic forms of limb-girdle muscular dystrophy, we want to look at research projects that look at innovative ways to deliver gene therapy to all. We also want to fund research projects where new types of medication could be useful across the board. You can get involved with this fundraising campaign by signing up at believethereishopeforacure.com. You can raise money your own way and help contribute to these research projects that will benefit all forms. Thank you so much.
Dr. Alan Russell is our next speaker. He is the co-founder, chief scientific officer, and a director on the board for Edgewise Therapeutics. Edgewise is a clinical stage biopharmaceutical company focused on the discovery, development, and commercialization of innovative treatments for severe rare neuromuscular and cardiac disorders, for which there is significant unmet medical need. Guided by its holistic drug discovery approach to targeting the muscle as an organ, Edgewise has combined its foundational expertise in muscle biology and small molecule engineering to build its proprietary muscle-focused drug discovery platform. Welcome to Edgewise and Dr. Alan Russell.
Thank you, Catherine. My name is Alan Russell. I am the Chief Scientific Officer and co-founder of Edgewise Therapeutics. We're a small biotech company based in Boulder, Colorado. I'll tell you a little bit more about us and about our strategy over the next 25 minutes or so. So what you're going to hear is a story that's a little bit different from some of the other ones that you've heard. I'm guessing through this series. We have kind of a structural approach to muscle disease, and I'll tell you all about it. And that's summed up in this title here, taking a universal approach to protecting muscle in Duchenne, Becker, and limb-girdle muscular dystrophies. We include here some results from phase 1 trials that we've run in Becker muscular dystrophy and plans for studies in limb-girdle 2I and other myopathies. So I'll give you a little bit of an idea about that. And thanks very much to the Speak Foundation for inviting us. It's a true honor to be able to present our data here. As usual, we are a public company, so we have this disclaimer to put out. And the main point all of these words is to make is that the compound that I'll be telling you about, EDG-5506, is an investigational drug and it's not approved at this stage. All right, so these are the tests that are ongoing that will hopefully show this thing works, but these have not been proved at this point. So just a quick company overview, and then I'll tell you about advocacy at Edgewise. So we were founded back in 2017. Really, we have a key focus towards improving the lives of patients and families suffering from rare muscle diseases. The strategy that we have is to design innovative therapies for muscular dystrophy that provide broad benefit in the real world. So we're trying to devise the easiest possible medicine to take that works in combination with the least number of side effects, that really works to improve your lives. That's the goal. The lead program 5506, I'll tell you about that. It's focused on Becker, Duchenne, and select limb-girdle muscular dystrophies, and I'll tell you why that is. And we're planning for phase 2 studies currently in Becker muscular dystrophy, and I'll give you a timeline of all of the studies that we have ongoing at the end of the presentation. Now, in terms of advocacy, we take this kind of stuff pretty seriously. And the goal for us is to receive input from you guys in the community and then actually take actionable steps based on that feedback to improve the studies that we're running and improve the kind of medicines that we're developing. So we take this stuff very seriously. We try as hard as possible to listen to those comments from out in the community about what you guys need. And we try and take actions based on those feedbacks in terms of designing the therapies that we're making and designing the studies that we're putting together to measure whether the compounds work or not. Now, I'm going to give you a little bit of a background about the dystroglycan complex and then what it really does as we see it, and then why and how we've designed a therapy that addresses certain problems associated with that. So what you're looking at here is a rather complicated cross-section of a skeletal muscle. So this is an example muscle. Maybe it's from the leg, maybe it's from the arm. Skeletal muscle makes up about 40% of your overall lean mass. So it's a lot of muscle. There's a lot of tissue out there. And that really controls movement of the body and general well-being. So if we move about, our skeletal muscles are basically in action. Now, what we focused on here is the dystroglycan complex. And in its most simple terms, what that complex does is it connects all of the stuff inside the muscle that controls contraction of the muscle to all of the stuff outside of the muscle that supports the tissue. So the protein and the collagen, for instance, that surrounds the muscle. So there's a connection here through the membrane via this whole complex. Okay. And I'll tell you about why that is needed over the next couple of slides. So now what I've done is taken a closer focus on the complex and where it's sitting in terms of the inside and the outside of the muscle. So here is the same complex. Here's the dystrophin protein here. Here's all the sarcoglycans. And this whole complex's job, as I mentioned before, is to connect the inside of the muscle, that's this bit, to the outside of the muscle, that's all this stringy stuff. But you can see how we've arranged it here. Here's another complex here. Okay, and this is no accident. So that when this muscle here contracts, and it uses this protein myosin in order to contract, binding to this protein actin, and the reason I'm pointing these out is I'll tell you more about these in the next few slides. When muscle contracts, you can see these connections here to the complex cause the membrane to move along for the ride, and actually also causes this muscle fiber to move along for the ride too. So in doing so, it kind of connects muscle fibers together. So when a single muscle fiber contracts, other muscle fibers around it are also pulled along for the ride and help support that muscle fiber. Okay. Now, many of these types of limb girdles you're familiar with, and many of these are components of this dystroglycan complex, such as these sarcoglycans here, these like smaller units here. There's 4 of those with associated limb-girdle muscular dystrophies. And then FKRP, which I'll be telling you about a little bit more, is associated with limb-girdle 2I, and that holds this whole complex together by amending sugars onto those proteins to really stabilize the complex. And then, of course, dystrophin is talked about a lot in the context of Duchenne and Becker muscular dystrophy. It's where we have most of our preclinical and clinical data, but a lot of it is directly applicable in many ways to the limb-girdle muscular dystrophies, and I'll tell you more about that. So the key feature of dystrophic muscle or limb-girdle muscle, whenever you would have a sarcoglycan complex, the dystroglycan complex, whenever you have that dysfunctional in some way, when muscle contracts, then what you find is that muscle becomes injured, okay? So when the muscle contracts, because you haven't got that connection to the fibers above or below, you get this excessive stress on the muscle fiber, and that causes the muscle fiber to receive injury and damage when it contracts, okay? So it's a hallmark of many of these diseases that when you exercise or when your muscles are active, they become injured more frequently. And that's because when they're contracting, then that stress builds up in that tissue and it results in injury, okay? I'm going to show you it in action. Now, this is a muscle from the mouse that has muscular dystrophy. So this is essentially a Duchenne muscle, but in many ways, many of the limb-girdle myopathies, such as limb-girdle 2I, have a similar feature. When the muscles contract, they receive injury. So this is just everyday use. What we're doing here in this mouse muscle is it's been removed and it's been put into a bath and we're contracting it. And you can see as we go forwards here, you see this muscle fiber here, it's moving when it shouldn't be. It's receiving more stress than it's used to because it's being used essentially, and that starts causing injury. And you can see that injury resolving in these fibers as they start getting snipped up. So the fiber becomes stressed and then it starts breaking down, and you can see the result of that breakdown as these fibers are snipped and they retract here and they start accumulating as these balls of tissue. So you can imagine this happening inside your limbs when you're exercising or active. Small portions of your muscle at any one time are receiving muscle injury, okay? That causes inflammation, and that inflammation is required to repair this type of damage, and muscles can do that. But of course, over time, the body it has trouble keeping up with the ongoing injury, and that's where things like chronic inflammation and fibrosis kick in. And by fibrosis, what that really means is just your muscle fibers start getting replaced with something that's not muscle, either fat or fibrotic kind of protein tissue, and that's where function really starts declining. So our goal is to stop this. We want to provide a benefit to the muscle whereby it can do its usual activity but not fall apart, and I'll show you how we do that. Now, when that muscle fiber is becoming injured, what tends to happen is that proteins that are normally resident inside the muscle fibers leak out, okay? And you saw that in the video, those muscle fibers start degenerating, and as they start degenerating, the contents of those muscle fibers, they spill out into the plasma compartment, into your bloodstream, essentially. Now, a few of those that you may be highly familiar with are things like creatine kinase. All right, so a hallmark of many myopathies, many limb-girdle muscular dystrophies, Becker and Duchenne as well, is that they have elevated creatine kinase, particularly when you're more active, but really all the time. There's a measurable level of creatine kinase. Other proteins also leak out into the plasma, and I'll tell you about those. Those include things like myoglobin, troponin, and other things as well. All of these are just proteins that are enriched in skeletal muscle. They're normally just present inside this tissue, and then of course when you receive the injury, they spill out and they can be measured, and they give you an indication of how unhappy your muscles are. Here's a rather complicated figure to show how we can demonstrate that. So we have an active collaboration with the University of Copenhagen, and John Vissing specifically is the investigator there. And he has devised a scheme whereby you can you can impose activity on people in a controlled way. So individuals come in to the health center there, and he gives them an exercise round. He puts them on a bicycle and he gives them leg weights to press against. And then we measure those muscle proteins that leak into circulation. So in this case, it's a very controlled way of doing exercise in someone to see how much muscle injury is present when you do a specific activity. Of course, you don't have to do this type of activity to receive a muscle injury, but in this case, he just pushes the individuals who come to see him in order for us to measure these biomarkers in a very specific and controlled way. And I have two examples here: myoglobin and creatine kinase. Those are two of them that I introduced back here. So you can see in both cases, now we're looking here at limb-girdle 2I and Becker muscular dystrophy compared to healthy individuals. And you can see in both of those instances, creatine kinase is elevated by the exercise, and as is myoglobin. So this is a controlled way of just measuring how easy it is to injure your muscles. And then that's reflected in the biomarker. And I'm going to return to this later on. There's a reason I'm telling you in some detail about this experimental setup. Now, it so happens that our skeletal muscles are formed from two different types of muscle fiber, so-called fast twitch muscle and slow twitch muscle. So you can see here, we've got the same muscle picture here. These fibers are evenly spaced around in a so-called mosaic throughout the muscle. Okay, so an even balance, about 50% of these things and 50% of these things in any one muscle. And you might ask, why on earth do we need two different types of skeletal muscle fibers in our muscles? And the easiest way of thinking about this is, if you think about normal activity, we do all sorts of things. We pick up very heavy loads, and then we go for long walks that require more endurance and strength. And in many ways, the slow twitch muscle fibers are involved in endurance, so they can keep going for longer without fatiguing. And the fast twitch muscle fibers are associated with strength, shorter bursts of activity to do kind of more active maneuvers, like getting out of a chair or picking up a heavy load or throwing a ball. Now, of course, life isn't that simple. These fibers share each other's loads, but they contain unique proteins in order to do some of these things. And one interesting feature is that these slow muscle fibers also share many components in the heart. Now, one very interesting feature of these different fibers is that they can get injured to different extents in these types of myopathies. And what we have here is we're returning to creatine kinase, which I told you about, and then these troponin proteins. And the reason I'm telling you about the troponin proteins in muscle is that one type of troponin, fast troponin, is really present only in those fast fibers. And one type of troponin, the slow troponin, is only present in slow fibers. And both of them leak out when muscle is injured. But what we found, and this has been reported for many years as well, is that while you see high levels of creatine kinase in things like Duchenne, lower but still elevated in Becker, and you could put one of the limb girdles into here as well. You can see these two are elevated compared to a healthy control. You only see the fast troponin and not the slow troponin in any of these individuals. So when I showed you those muscle fibers contracting and that muscle injury and the leak of proteins, that leak of proteins appears to be much, much more in the fast fibers than the slow fibers. And our goal is to really protect these fast fibers while retaining their functionality and allowing these slow fibers to do their normal work. So really just kind of normalizing how muscle works. So as I mentioned, fast fibers are more susceptible to damage due to the lack of functional dystrophin or a functional dystrophin ligand complex. We've made this investigational therapy, 5506, that's designed to protect these fast muscle fibers from damage. And we're going to do that by modulating myosin. I'll tell you a little bit about that in a bit. In animal models of muscular dystrophy, 5506 protects these susceptible muscle fibers and prevents long-term development of damage. So you can see things like improved fibrosis and the like. And I'll show you some examples of the compound at work. So this is returning to this graphic. This muscle has no functional dystroglycan complex here. It's been destabilized by mutation. So when this muscle fiber contracts normally, it stresses the system, and that causes the injury that we showed you already in the video and the graphics. Now, what we're doing here is we apply a modulator of myosin. That's the protein that regulates contraction of the muscle. And what we found is that small amounts of the compound that do that decrease the stress in the muscle so that now the muscle can contract normally without causing that stress effect that causes the injury. Okay? And here's the easiest way of seeing it in action. So what you're looking at here is the same video on the left-hand side. So this is the mouse muscle from a dystrophic mouse. It has Duchenne, it has no dystrophin, but this would be applicable to not having a dystroglycan complex 2. And you'll see again, when this muscle contracts, it's going to fall apart. So it's going to receive injury, and then it's moving around when it shouldn't be, and then you'll start seeing these snipping events as these enzymes start clipping up the proteins inside the muscle and the fiber falling apart. Now, what we've done here is we've applied small concentrations of 5506. That's binding to the myosin in the muscle. That's changing the stress in that muscle so that when the muscle contracts, and you can see it contract in a sec, there you go, that stress effect now does not cause the injury that you saw before, because we've kind of normalized that stress response so that it's not injuring the fiber. So this thing is contracting as normal, but instead of having this really profound injury response, it keeps on trucking. So in a way, what we've done is we've not replaced any of the dystroglycans. What we've done instead is taken a structural approach to normalize the stress in the muscle rather than changing the genetics of the muscle itself. So as such, you could use this compound for a whole variety of different myopathies where there is a stress response to activity. Here's example number 2. It's a much more complicated model. And again, this speaks to Duchenne and Becker muscular dystrophy, but it's the information that we gathered here is highly transferable to the limb-girdle muscular dystrophies. So what we did is we did short-term dosing in a model of Duchenne muscular dystrophy in dogs. These are golden retrievers. It's probably the most severe muscle disease model that you can get. So these golden retrievers tend to be smaller, and as they get older, the muscles become quite sick. So the muscle breakdown that I showed you in the video keeps going, causes inflammation, causes muscle loss, and the dogs get quite weak and inactive. So what we did is we took older dogs that had already had that muscle degeneration, and we gave them 2 weeks of therapy with 5506, just as an oral dose once a day. And what we did is we measured every day, we measured creatine kinase, and this is the average of the 2 week dosing period, so before and after the dose. And you can see when the dogs took the medicine, their creatine kinase went down almost 50%, something around there. At the same time, what we did is we heard reports that the dogs were more active. So we did another study just looking at activity in these dogs, and you can see the opposite. So when the dogs took the compound, they were more active, and this is measured with an activity collar, just measuring habitual activity. So you can see those dogs were considerably more active, about 30%, while taking the compound. So this is kind of a proof of concept that when you take the compound, it allows the muscles to work normally, but it decreases how injured they are when you're active. So this is just a review about what we know so far about 5506. It's been into a number of studies in healthy volunteers, both in single and repeat doses for 2 weeks. And we gathered a lot of information there that we can kind of use to tell you more about how the compound works and how tolerable it is. In terms of the dosing for 2 weeks in these healthy volunteers, 5506 is well tolerated. There were no serious adverse effects. It was a well-tolerated drug. So it's a relatively safe drug so far in studies. How's it given? It's well absorbed. You can just take it orally. It's a once-a-day type of medicine. You can take it with or without food. So it's a very practical medicine to take. Does it get to the muscle? What we did is in those studies, we took muscle biopsies, so small chunks of muscle, and just measured how much compound was in there. And what we found is that 5506 is highly concentrated in muscle compared to the bloodstream. It actually, you take the compound, it goes into your bloodstream, and it actually comes and finds the muscle, and it sits there to do its work. So that's a great thing to know, that the compound actively partitions and gets into your muscles. Now I'm returning to biomarkers because I'll tell you what they do in the context of Becker. If you remember, I've told you about creatine kinase, and I've told you about fast troponin being a biomarker of fast skeletal muscle breakdown. And you can see again that Duchenne individuals have very high troponin and CK. Becker have high, but intermediary levels of troponin and CK, and then healthy people have either none or low CK. Okay, and this is just a reflection of how unhappy your muscles are and how much they're leaking their contents into the bloodstream. Now, what you see here is what happened in Becker individuals who took 5506 for 14 days. So 2 weeks of therapy. And what we did is at the end of the 2 weeks of therapy, we measured blood creatine kinase or the fast troponin. And you can see here's what a healthy individual looks like. Here's what a Becker individual looks like, creatine kinase. This is what happens when you take the placebo, and this is what happens when you take the regular compound 5506. And we saw an approximate 71% decrease from baseline with the therapy. And the same story is true for fast troponin. In this case, we've got an 83% decrease with the compound. So a nice drop in those injury biomarkers when individuals took the compound for 2 weeks. So just a proof of concept, very similar to those dog studies, but now, of course, in individuals with Becker muscular dystrophy, there appears to be protection from the compound when you take it as an oral medicine for 2 weeks. So here's the therapeutic review for 5506. The main goal is to preserve and potentially improve muscle function in Becker, Duchenne, and other limb-girdle muscular dystrophies that have activity-dependent muscle injury. So when you use your muscles, they get injured. It has the potential to be used alone or in combination with other therapeutic approaches. Now you can imagine this is kind of a structural approach. We're just changing the internal stress of the muscle. So we should be complementary with gene therapy approaches, anti-inflammatory approaches. These are all very different types of drugs than the one that we're developing here. And it's really designed to stop the damage where it begins. We're really getting in at the core cause of dysfunction in these muscles by uncoupling the ability of the muscle to injure itself. And that's right at the very top of the disease cascade. So we hope to have a fairly big effect in terms of improving muscle health. And as I've mentioned before, it's a novel mechanism of action. No one has tried this type of approach before. It'll be really exciting for me as a researcher to see how this story goes. And I'm really hoping that we can make an improvement in people's lives with this type of approach. Here's some review of the clinical timelines that we have ongoing this year and next. So right now we have a 12-month open label study ongoing with participants with Becker muscular dystrophy who were also in that 2-week repeat dose phase 1 study, where I showed you the creatine kinase and the troponin. So this is ongoing. It's going for a year. We're a few months into this study. It's going to roll all the way through 2022. At the same time, we have a natural history study initiated in individuals with Becker muscular dystrophy to get a better idea about how the natural course of the disease takes hold. And that's in a broad age category from 8 to 65. There's the clinical trials number for that one. And that's really to set the table to understand more about how the disease changes with age and how a medicine might impact that. We have a second study planned in Becker muscular dystrophy. Now, this is a more traditional phase 2 study. This is to start in the second half of 2022 in individuals 12 plus. This is a much bigger study than the long-term extension. So we're excited to get that one going. And then we also have a study using the exercise challenge setup that I described back in the presentation in limb-girdle 2I and McArdle as kind of examples of different types of muscle disease that are separate from Becker and Duchenne. Okay, so this is a study based in Copenhagen. It's going to start second half of this year. It'll be a small study looking at this controlled exercise and then long-term outcomes after that. So it's really just an idea to see if I do a controlled exercise in an individual with limb-girdle 2I or McArdle, do I get those biomarkers changed? And then does the therapy, if I give the therapy for 3 months, change that response to the injury? Okay, so it's a very simple yes, no answer, and then we'll do a long-term follow-up on that study to see if that compound has any functional improvements with longer-term dosing. So we're really excited for that study to kick off towards the second half of this year. Now, of course, quite often when you give these presentations, it inspires 1,000 questions. So if you do have any specific questions, there's an email there for you to follow up. Just send us an email with your questions, and hopefully we'll have a good answer for you. And then you can go visit our website as well, which is constantly being updated, just to give you a better idea of where we are in the clinical development pipeline and what the results are so far. So thank you very much for your time, and have a good rest of the day. Thank you so much.
Rita Perlingero, PhD, is a professor of medicine at the University of Minnesota. Her laboratory has been studying stem cell transplantation and skeletal muscle regeneration in mouse models of muscular dystrophy for more than 15 years. The Perlingero laboratory was the first to demonstrate functional improvement after the transplantation of mouse and human pluripotent stem cell-derived myogenic progenitors in dystrophin-deficient mice. Rita has 90 peer-reviewed publications, from which more than 30 are on regenerative medicine for skeletal muscle. The NIH has funded her laboratory uninterruptedly for nearly 15 years. Rita is a co-founder of Myogenica, a muscle cell therapy startup company.
Well, thanks, Kelly, for the nice introduction. I would like to also thank the Speak Foundation, Catherine Bryant, for the invitation to speak in this important event, and actually having the pleasure, the honor to wrap up the limb-girdle muscular dystrophy Global Advocacy Summit. Today, I'll be sharing our recent developments in optimization studies to enable phase 1 clinical trial of iPS-derived myogenic progenitors to treat muscular dystrophy. Before I begin my disclosures, I representing today the University of Minnesota, but I have recently co-founded Myogenica, a startup which is in the process of licensing the patents from the University of Minnesota. So to the science, pluripotent stem cell derivatives actually have been used quite extensively in recent years. Many studies have been applying safety and efficacy trials for the treatment of macular degeneration, Parkinson's disease, and also solid tumors and hematological diseases. So far, these trials have shown safety, and many have demonstrated success or improvement in these different diseases. One aspect in common for these 3 diseases that I'm showing you here, that are more in the pipeline, is that for these diseases, you are generating a specific cell type that's important for that particular disease, just the final differentiated cell type. In case of macular degeneration, we are looking for the pigment epithelial sheet. In case of Parkinson, you are looking for the dopaminergic neurons. And in the case of solid tumors, hematological diseases, we are looking for the generation of natural killer cells, which are then injected with the idea of destroying the tumors. So all challenging, but what is important here is to generate this particular cell type, lots of this differentiated cell, and that's it. With muscular dystrophies, it's a little bit different. As you might be familiar, this is just an example for disease progression for Duchenne muscular dystrophy. Many other types of muscular dystrophy have a similar progression. As we are looking here on affected biopsy, we have the myofibers, we have the nuclei in the periphery. This is a healthy biopsy. In the early stages, you start seeing that the architecture changes. You see inflammation, you see fibrosis. And one feature that I want to bring your attention here is these myofibers here, that you might observe that the nuclei is actually in the center. And this is actually an indication of regenerating new myofibers, which is the indication, was actually one of the early observations that led to the discovery that the skeletal muscle actually has muscle stem cells. So in the beginning, in the early stages of the disease, the muscle stem cells, they work hard to compensate for the muscle degeneration, and if make these new myofibers. But over time, it's not enough. And as you can see in many types of muscular dystrophy, you see the muscle architecture is gone, you have fat deposition, and you basically don't have any more myofibers. Of course, this varies from muscle to muscle, disease to disease. But the message from this slide is that for the treatment of muscular dystrophies with cell therapies, we want to replace the tissue with the disease tissue with muscle fibers, myofibers, so the functional tissue, the final cell type that you need to create a healthy muscle, but also muscle stem cells, because this is going to be important to allow for long-term regeneration, because as you can imagine, this process of regeneration is continuous. But as you have healthy muscle stem cells provided to this muscle, these cells should be able to keep contributing to the formation of healthy myofibers. So how do you do this? Well, it's not trivial for any of these therapies that are ongoing in the clinic right now. It starts with the generation of large numbers of the cell of interest. And when we started this project many years ago with the first proof of concept, this was more than 15 years ago, even before induced pluripotent stem cells, with this rationale, this premise that you really needed to generate not only muscle fibers, but also stem cells, our strategy was to modify the pluripotent stem cells, the cells that have the capacity to generate any tissue in the body, with PAX3 or PAX7, which are proteins. These are genes that are very important for the development of muscle and the maintenance of muscle stem cells. So we modify cells, the pluripotent stem cells, with these genes, and you give a signal to induce the expression of this, which is dependent on a drug called doxycycline, and this is conditional. So we give this drug, and the cells respond and start proliferating PAX3 or PAX7 myogenic progenitors. They act the same way. They're very similar. And this allows for the expansion, significant expansion, and you're going to see in a few slides, in the dish. And once you transplant these cells in different animal models of muscular dystrophy, which you're going to see in the future slides, we see muscle engraftment. In this slide, I'm showing an example for Duchenne muscular dystrophy. These mice lack dystrophin, so you can see that the mice who were transplanted intramuscular, they have dystrophin positive myofibers that are also positive for green fluorescent protein. In this case, the cells were labeled with this color. In addition to the muscle fibers, we see muscle stem cells. They are located in the periphery of the muscle fiber. This is a donor-derived fiber, it's green, and you see the presence of PAC7, which is a marker of stem cells colocalized with the donor marker, so indicating the presence of muscle stem cells and muscle fibers. And obviously very important is to see that this transplantation actually results in muscle that's functional. So this is a really summarize this slide. We have done these transplantations in early studies, intramuscular, IV, and also intraarterial. And in all the different cohorts, we see an improvement in function in the muscles that were transplanted with cells. Later on, we evolved and we combined IPS technology and genetic modification using the more severe animal model of Duchenne muscular dystrophy, the double knockout for utrophin and dystrophin, and show that you could make IPS cells from these mice, put microutrophin back, generated the myogenic progenitors using PAX3 as I showed here before, and you transplant and you see large numbers of donor-derived cells. And again, even this animal model that's extremely severe, you are able to see improvement in the contractility of the muscles that have been transplanted. We also transplanted these mice IV, and we also were able to see the presence of donor-derived myofibers in different muscles. One question that's very important, because again, as you think about muscular dystrophies, you have this genetic defect, and the degeneration, regeneration is ongoing. We'd like to recapitulate that in the mouse model, which you have mouse models for different types of muscular dystrophy, but it not necessarily recapitulate exactly to the same level. And also it would take a long time to follow up this mice. So one way to do that, to see how the cells respond to multiple rounds of degeneration and regeneration, is to induce injury with a toxic substance that in this case is cardiotoxin. So we label the cells with this call with lentivirus that we see luciferase, that's what you're seeing here. So this leg didn't receive cells and this leg received cells, and you can see right after transplantation, the signal is very strong. We inject 500,000 cells there. And then the signal starts to fade because the cells start to differentiate and making muscle. Some are just eliminated. And by 4 weeks, the muscle is just stable. And then you come with a second set of injury, and all the mature fibers, all the fibers that are there, they get eliminated with this injury that we are provoking. As you can see, the signal goes down, and as you can see, start very low. But what happens is that the signal comes back up. And what this means is that the cells that you transplant generate stem cells that upon this injury are responding and making new myofibers, because this injury doesn't affect the muscle stem cells, just the myofibers. So when you look at the histology, you can see that all these green fibers here, they express embryonic myosin heavy chain, which is an indicative of newly formed myofibers. So this shows that the cells are able to respond to injury. So we got a little bit greedy. It's like, well, we did just once. What happens if you do repetitive rounds of injury? Because, trying to, even though it's an artificial system, can we mimic? Can you allow how hard the cells can work in terms of regeneration? And as you can see here, we did 3 rounds of injury, and as you can see, the signal comes back up. And this is how the muscle looks like at the end point at week 20. So all these are donor-derived myofibers. So this shows really the cells have the capacity to contribute to long-term regenerative potential and actually come back and contribute over multiple rounds of degeneration. So this was mouse work, but how about human? Of course, this wouldn't be relevant if you are not able to see the same kind of results with human pluripotent stem cells. And this work was actually published 10 years ago. Time flies. So we are able to recapitulate the same system I showed before. So at this time, we put PAX7 and RTTA lentiviral vectors, and we generated these myogenic progenes that proliferate significantly and make myotubes in the dish. Kind of a quality control. When you transplanted these cells in an animal model, here, this is, I think, my last slide with MDX, in a mouse model of Duchenne muscular dystrophy, that doesn't have the immune system allowing for the transplantation of human cells. You are looking here specifically for human dystrophin positive fibers and also for another marker of human cells. So you can see the engraftment is similar, and we are able to see, again, an improvement in the contractility of transplanted muscles. And here we are looking at the green bar compared to the yellow bar that were muscles just injected with saline. And this is how a dystrophic non-treated mouse looks like, similar to PBS, and this is a control. The same way you look for the presence of donor-derived muscle stem cells in that location that I told you earlier, and here they are expressing a human marker, PAC7, and in the right location. They are there. Are they responding? So one way of looking at that is to leave the mice sit, because this is also very important to look for safety, right? The cells are making muscle, they are improving the function, they are making muscle stem cells, but are they making anything that could be unsafe? So we kept a cohort of mice for a long time, almost 1 year, which is equivalent to many years in a patient, and we saw no tumors, no ectopic formation of muscle in any different tissue. And we could see that the engraftment was persistent. So that was very encouraging. So that put us in a situation that you looked at. There are two ways that you could envision a therapeutic, the development of a therapeutic application from pluripotent stem cells. One, you have iPS cells generated from a healthy donor, as I showed you before. With those cells, you reprogram to make iPS, you generated the transplantable myogenic progenitors, and you inject into patients with muscular dystrophy. And also you can envision, because you generated these pluripotent stem cells from the patient, you could generate patient-specific iPS cells, which could be used for autologous transplantation. Obviously, in this case, this would require the genetic correction of the specific disease. So before I go into in our pipeline for the first in-human clinical trials, I'd like to show you what we have done in terms of showing the proof of principle for the autologous transplantation. And in this case, we actually were looking specifically in limb-girdle muscular dystrophy. We studied two types. We studied limb-girdle muscular dystrophy 2A associated with calpain-3 mutations, and also limb-girdle muscular dystrophy 2I associated with fukutin-related protein mutations. So starting with calpain-3, we had it in the lab, iPS cells from 3 patients, 3 independent patients with different calpain-3 mutations. These cells, we use our approach, they are able to generate the myotubules in the dish. You know, the patients do have muscle, so they differentiate fine. And before going the gene correction, we actually show that this is the control. We have calpain-3 expression, while the patient-specific iPS cells don't. Then we came up, because of time, I'm not going to discuss the details, but this work is published, so I'm happy to discuss further if anybody's interested. We use CRISPR-Cas9 mediated genome editing to correct the mutation. And as I'm showing you here, this for one of the line 1915, we have 2 clones that are corrected and uncorrected is here, no calpain-3. The same for the others. So you see detect calpain-3 only in the corrected clones. Then we transplanted this in an animal model, immunodeficient animal model, knockout for calpain-3, and you see the presence of human-derived myofibers more in the corrected clones, but you could not really look at the protein because this is very hard to detect. The calpain-3 antibodies are not good for immunofluorescence, but what we did, we looked for calpain-3 mRNA in these transplanted muscles, and as you can appreciate it here, you see no signal for the muscles that were injected with saline, and why you can see transcripts for human calpain-3 and human DMD in the transplanted muscles. Similar studies were done with fukutin-related proteins. So in this case, we also had 3 patient-specific pluripotent stem cell lines, different spectrum of the disease. We had the WWS, a limb-girdle muscular dystrophy 2I, and one of the limb-girdle CMD kind of phenotype. So when you look at in the dish here, what we are looking for to detect the correction, are looking for 286 staining. So this antibody here, it recognizes functional alpha-dystroglycan glycosylation. So in the lac in the mutant iPS cell line. They all differentiated fine, but in the mutant, uncorrected, you are not able to detect 286 staining, which would be red here, as you can see for the control myotubes. But upon the gene correction, you can see that this same line upon correction, looking at clone 40 here, you can see nice staining for 286. And you look at this by immunofluorescence and also by Western blot, and what you're looking here is the presence of the protein here. And you're also able to see that these myotubes now are able to bind to laminin, which is not happening in the mutant FKRP myotubes. What happens if you transplant? So we have the limb-girdle muscular dystrophy 2I model, which we have generated the immunodeficient version, which allows for the transplantation of the human cells. And you can appreciate it here, well, in the non-transplanted muscle, you have no human signal, as you would expect, and no immunoreactivity for 286. But in muscles that have been transplanted with the corrected clone 40, you can see the presence of human dystrophin, and just to indicate that the muscle fibers here, and also the human marker, human lamin AC, which is green, human specific, and these areas are also positive for 286. So this shows that actually the cells that have been corrected and transplanted in an animal model of limb-girdle muscular dystrophy, which is devoid of functional glycosylation of alpha-dystroglycan, you are able to rescue the phenotype. And this is the enumeration of the quantification of engraftment. And in these muscles, we are able to see the presence of donor-derived muscle stem cells. So, okay, what's next? So we show that we are able, theoretically, both allogeneic and autologous transplantation could represent therapeutic alternatives to treat muscular dystrophy. But as we were planning, what would be the next, how to begin the road for translation, we started discussing this. There are many questions. Purification, what would be, one way is to manufacture cells in the lab. Another way is to manufacture in a lab that's for clinical production, to cells to be given to a patient. So there are many, many steps that have to be taken in consideration and conditions that the cells have to be maintained in. Safety, scalability, and obviously delivery. So there were many questions. This is very, summarized the list of questions we had. And since if you went to autologous route, this would be the generation of one cell product. It would be good for just one patient. We thought we had so many questions that you need to address, they would make more sense to begin with the allogeneic transplantation, being that you generate a cell product that will be extremely well characterized, and this can be tested in several patients, right? That's the idea of the allogeneic transplantation. You have one cell product, and this can be used for many patients, many diseases, many types of muscular dystrophy. And that's how we started. So this initiative started 2016. I can't believe it's 6 years ago. This is the group. We had the privilege to have experts in many different areas that are important to evolve this basic research to a clinical trial. So skeletal muscle expertise, cell therapy expertise, clinical production expertise, our neurologist colleagues, and people with expertise in regulatory affairs. So this is the molecular and cellular therapeutics facility here at the university, where we have our own CGMP production facility. So we work very close with Dr. McKenna's group to enable this, clinical translation of our myogenic product. So when you began, this is just, again, this is a very summarized list, but there were lots of things that we needed to address. Start with the methodology. Is this really the best methodology? And we tested different strategies out there. And one key aspect of this technology is that you really need to generate very large numbers of cells. And these cells have to be somewhat homogeneous. And you want early myogenic progenitors. As we discussed in the beginning, we don't want a more differentiated cells. You want cells that you can generate large numbers of these early progenitor, when you transplant, give rise to stem cells and muscle fibers. Another very important point is the purification. So pluripotent stem cells, they have the capacity to make teratomas. So we need to make sure that the cells that are finally, the lineage has been specified, and even better is if you are able to purify your cells of interest. So anything that's not the myogenic cell is going to be eliminated in this purification step. We have to generate a GMP compliant. We have to make sure the scalability. Well, the list is very long. I'm not going to read all of it, but I think you can appreciate that we have just completed the CGMP, the clinical product has been generated. We have lots of vials in the freezer, and we are getting up now for the GLP preclinical studies, which is going to allow us to be ready for IND filling in several months. So this is a scheme of the strategy. So we have CGMP iPS cells. We transduce this with the CGMP third generation lentiviral vectors, which allows for the generation of what we call the IPAC7 master cell bank, that we have several or 100 vials. These will give rise, we start each vial, give rise to the myogenic production, which after several days in culture, we have this purification step for CD54. And after several days of expansion, we have what we call myopaxon. That's the myogenic cell product that we plan to transplant into patients. This is an example how these cells look like after transduction. This is in the iPS stage. They have normal karyotype. They make nice myotubes in the dish, and more importantly, they still have the capacity to make myofibers once transplanted in animal models. So I'm making a very long story short. As you can imagine, there was significant delay in different points of this journey because of all the aspects related to COVID in the last 2 years, and you name it. But you have been able to nicely transfer our technology to the clinical grade facility, and you have optimized the purification, being little columns in my lab, to this Clinimax, which is this instrument that you use for clinical application. That's a closed system. The cells don't get exposed to the air or to the operator. And using this strategy, and after optimizing the media composition that's compatible with clinical application, we are able to validate that the cells have the same potential for expansion. Easily, we can obtain billions of myogenic progenitors. These cells maintain the expression of key surface markers, and regardless of passages in culture, they maintain their capacity to make myotubes in the dish. But more importantly, they have the capacity to contribute to muscle regeneration when transplanted in animal models of muscular dystrophy. You can look here, human engraftment after 2 months, and this is after 6 months. So the engraftment is persistent, as we have seen before. And now, just out of the oven, we are transplanting these cells in different animal models of disease. And I'm happy to share with this audience that we have recently transplanted the cells in the mouse model of limb-girdle muscular dystrophy 2I. And you are able to see that this particular batch of myopaxon actually is able to generate improvement in maximal force and specific force. So we are very excited with these results. So where we are, so this is a very summarized slide, how, what is necessary to go to the clinic. We started really here, although we had a lot of proof of concept. We had a lot of work to do to make sure that what we had generated was compatible with clinical application. So we have been doing proof of concept, validation, toxicology, all the characterization of the product that's required for the FDA. We had our pre-IND several years ago, because we want to make sure the FDA was in agreement with our plans, and we were. But there was a long list of testing that need to be done, and some have been completed, and some we are in the process of starting now that we have the final product, CGMP. So in terms of our plans for the first in human phase 1 trial, we are focusing on individual muscle injections. The main objective of this first clinical trial is the feasibility and the safety of intramuscular injection. That's our priority, is to make sure the cells are safe. We are anticipating a small number of patients of 7 or 10, and as of now, the idea is they're going to be Duchenne muscular dystrophy patients. And as soon as we have the proof of concept that the cells are safe, our plans is to move future trials and expand the patient population to include the limb-girdle muscular dystrophy and also FSHD patients. And we'll be focusing on additional muscle injection sites and in the future, the systemic treatment. But as the FDA made it very clear, first we focus on the intramuscular, and then in another package, we focus on the systemic. So one thing at a time, but these studies are running in parallel. And I would like to end by thanking this amazing group that I have the privilege to work with. Current lab members, Jim has been instrumental, Jim Keeley has been instrumental in optimizing the protocol and transferring the technology to the clinical grade facility. Karine has been instrumental in validating the myogenic cell product every step of the way. Alessandro is no longer with us, but he was instrumental in getting this project out of the ground and, being part of the team in the early years of transferring the technology to the clinical grade facility. I also like to thank Neha, who is the scientist who led the studies on the gene correction of limb-girdle muscular dystrophy 2I. I should circle here too, but Sridhar was the one leading the work on calpain 3. I also would like to thank the colleagues here, as I mentioned before, a group of experts that without them we wouldn't be able to reach to the point where we are now and move forward with the plans we have to move into the clinical trial. We have collaborated with the Indiana University, Kenny Cornetto, with the generation of the lentiviral vector cGMP and the validation testing. We received mice from our colleagues from the Carolina Medical Center for the limb-girdle muscular dystrophy 2I, Melissa Spencer for the 2A, and we received IPS cells, patient-specific IPS cells from Dr. Adolfo Arregui in Spain, and also from NBank in San Diego. And of course, I'd like to thank all the funding agencies. And here I including all the agencies that have funded us over the years, over more than 15 years, because, even though these are the current funding, without this funding in the early years or over the years, we wouldn't be able to be at this stage where we are now, to be getting ready for a clinical trial, preparing for the clinical trial. So I would like to really deep thank for the funding agencies, and hopefully we'll be making an impact in the near future. Thank you.
Thank you to all of our presenters and speakers. And thank you most of all to those who are living with limb-girdle muscular dystrophy. As someone who is living with LGMD 2I, I understand the difficulties every single day of this disease. My hope and prayer is that we see treatments come to fruition very quickly and safely. If you would like to donate to the life-saving mission of the Speak Foundation to help us continue to do programs and projects that help the LGMD community, please go to www.thespeakfoundation.com. We want to thank you for being with us today.